So our first speaker this morning will be Dr. Leo Chang. So Dr. Chang?
Yes, yeah.
Are you going to give us some welcoming and introduction address?
Yes.
So Dr. Chang is the Associate Director of State Laboratory of Marine Pollution and a visiting Associate Professor from Department of Biomedical Sciences, CTU. So he has very broad interest in ecology and biodiversity of marine ecosystem. So Dr. Leo Chang will give us some welcoming address.
Yeah, thank you, Harry.
Yeah.
So on behalf of the State Laboratory of Marine Pollution at the CTU University of Hong Kong, I would like to welcome all of you to participate today's symposium. So as you know that yesterday we have a training workshop, following by today's one-day symposium, which will feature a series of talks on recent advances in marine environmental research, including the latest technology of pollution monitoring and control, occurrence and impact of emerging chemical concerns, problem and imminent stressors such as hydroxia and algotoxins, and also novel approach in ecological restoration. Particular attention will be placed on toxin and man-made substance that pollute the ocean or affect its living biodiversity. Participants will have a chance to express their view on the challenging marine environmental research during the panel discussion chaired by Professor Rudolph Wu. So Henry already introduced me. I'm the Associate Director and also the Director of Shenzhen Key Laboratory for the Sustainable Use of Marine Biodiversity. As Rudolph is here, the State Key Lab is built upon the excellent leadership of Professor Rudolph Wu, established the Area of Excellence Center of Marine Environmental Research and Innovation Technology in 2004. And then Rudolph is continue taking the lead in organized Tennessee Regional Center for Excellence in Marine Pollution over the last, I think, 12 years, Rudolph, right? And the State Key Laboratory, the major reason is to be a key international research center in advancing marine environmental research that contribute to the protection and management of the marine environment and generate positive societal impacts. We have three major missions to protect marine environments of Hong Kong, South China, and Asia Pacific regions. So that's why we organize the training workshop and today's symposium for the purpose of putting together more interdisciplinary expertise through high quality multidisciplinary research and innovation relevant to pollution monitoring and control, environmental risk assessment, ecosystem response to stressors, and ecological restoration, and also to build capacity by nurturing and training environmental scientists through yesterday's workshop and today's symposium, and manager and industrial entrepreneur in the regions, and also to support Hong Kong SR government and the Chinese central government in the management of environmental quality and protection of marine ecosystems. So the uniqueness of MURRI STMP is its inter-university, multidisciplinary, inclusive, always emphasized by Professor Wu to pull together all the expertise in Hong Kong to conduct collaborative research that gather together many minds to become greater together, to be inclusive, not exclusive, to tackle grand challenges, create impacts together. So we have three strategic research fields. The first one is led by Professor Zhang Tong, the first speaker, and then Dr. Guo Zhenji, and he will give a presentation in the later morning. And Professor Wang Wenxiong lead the second strategic research field, eco-safety and environmental risk assessment. And Dr. Henry He to be the deputy leader for this direction. And also Professor Chu Jianwen is also here. leading the ecosystem response and ecological restoration with my assistance. So there are two pieces of good news from SKMP. SKMP will lead UN endorsed project, the UN Decade of Ocean Science for Sustainable Development Framework, proposed by Professor Kenny Wu, the director. And also we are in application, a regional training center on pollution assessment and monitoring. So thank you all for your attention, and I pass to Henry for chairing the following sessions. Thank you.
Yeah, thank Leo. Yeah, thank you very much. So now we are just going to start our first morning session. So the first speaker will be Professor Tong Zhang. So Professor Zhang is a chair professor in water and environmental engineering from Department of Civil Engineering, University of Hong Kong. His main research areas include wastewater engineering and biological treatment, anaerobic digestion, environmental microbiology and biotechnology, environmental bioinformatics, and environmental toxicology related to wastewater treatment. Today, he will present mechagenomic insights into antibiotic resistant genes in environments. So Professor Zhang, the podium is yours.
Thank you, Henry. Thank you for kind introduction. Hello, everyone. I'm Zhang Tong from the Department of Civil Engineering, the University of Hong Kong. My laboratory mainly works on environmental microbiome. Since 2007, we have done some research about antibiotics, the chemical pollutants, and the antibiotic-resistant genes, the biological pollutants. Today, it is my great pleasure to have this opportunity to share with you our study on ARGs using metagenomic measure. As you may know, in 2015, the World Health Organization, WHO, listed antibiotic resistance as one of the biggest challenges facing human health in this century. According to a report published by ONU in 2016, by 2050, if no action taken, the problem of antibiotic resistance cannot be effectively solved. 10 million people will die due to the diseases as related to drug resistance every year, exceeding the deaths due to the cancer. Antibiotic resistance, of course, is a medical issue first, but it is not only a medical issue. It also an issue having environmental dimension. According to the widely accepted scientific concept, One Health, the health of human, animal, and environment are closely connected. Antibiotics used by human account for 30%, and the remaining 70% is used by animal. After use, 80% of the used antibiotics will be discharged into the environment. At the kind of the selective pressure, it will become the important factors in promoting the spread of resistant genes in the environment. At the same time, the drug-resistant bacteria and the other resistant genes produced in the course of different use will enter the environment with the charge of sewage and pollutants and become the main sources of the resistance in the environment. The environment is the key node in the spread of antibiotic resistance. Bacteria in soil, river, lake, and seawater could develop resistance due to the drug-resistant bacteria, antibiotics, and other chemical substances used by human beings, such as disinfectant, heavy metal, and so on. Human and livestock will be infected by more drug-resistant pathogens through food, water, and air contact. And the antibiotics used in response to these infections will begin another new round of the drug resistance evolution cycle. United Nations the environment, United Nations Environment published the Frontier Report 2017 and pointed out that antibiotic and resistant genes have become environmental issues of serious concern. There are different measures to analyze the resistant genes in the environment. The basic method is to isolate the bacteria on selected media and then determine whether they carry resistant genes or not based on the phenotype. That means which antibiotics they are resistant to. Molecular methods such as PCR and sequencing have been used to study the genotype of resistant bacteria for quite a long time. And the quantitative PCR can detect the concentration of resistant genes in different environments. If we can design suitable primer and probe, then we can use high throughput quantitative PCR or DNA chips to study hundreds of different resistant genes at the same time. With the rapid development of second and third generation high throughput DNA sequencing technology, metagenomic methods are becoming a widely accepted broad spectrum quantitative detection method. Of course, there are other methods also could be used to study ARGs together with the above method. Compared with the metagenomics, the throughput of qPCR method is low since the number of available primers are limited. Additionally, qPCR methods are affected by many factors, including the reaction system, qPCR instrument, the matrix effect, and the training of the operators. It is very difficult to standardize. Since the resistant genes are a global issue, scientific research and environmental management need to have more systematic and comparable data set to identify the important pollution sources and compare the overall level of resistance in different countries and regions. This requires a relatively standardized analytical method. At this stage, the metagenomic method as a method with a high throughput, more standardization, and also better openness has been applied more widely. It not only can analyze the known resistant genes, but also may help us discover new types of resistant genes if we combine it with the functional screening. More importantly, the unknown resistant genes in the previous DNA sequence dataset could be retrospectively rechecked for reanalysis and reminding when we have knowledge of the novel resistant genes. In terms of the detection sensitivity and customization, the qPCR method and still it's much better than the metagenomic method. So in summary, currently the qPCR method and the metagenomic method have their own strengths and complement with each other. But to study the resistome, that means the whole collection of antibiotic resistance genes in a complex environmental system, the metagenomic method is much more preferred considering the above benefit, which I mentioned. The database is the core of the metagenomic detection method. The completeness of the database will significantly affect the accuracy of the detection result. If the database is not that complete, the abundance and the diversity of resistant genes in a sample will be underestimated. So the key technical bottleneck of metagenomic method is how to effectively and accurately expand the existing resistant database. At the beginning, we merged two widely used resistant gene database and then produced a more comprehensive one. Based on that, we made some improvement and developed the online ARG-OEP analysis platform for metagenomic analysis using the second generation sequencing dataset. This analysis platform could be used to comprehensively analyze the resistant genes in environment sample. It can simultaneously detect 1,200 resistant subtypes, cover 24 types of antibiotics. In the second version of this database, we add hidden marker model to perform high throughput screening of new resistant genes. Currently, this database and the platform become a widely used tool for the analysis of environment antibiotic resistant genes. The website of our tool has been visited for more than 25,000 times up now. Using the established metagenome measure, we analyze the resistant genes of 10 types of typical environment samples and study the entire chain of the water environment system, including the sediment, soil, river water, drinking water, sewage treatment sludge, human gut and sewage influent, livestock manure, and farm wastewater treatment system. The total abundance of resistant genes in different environments is closely related to the degree of human influence. From the natural environment to the livestock, the total abundance of resistant genes has increased by 3 orders of magnitude. The main resistant genes include aminoglycoside, beta-lactam, quinolone, sulfonamides, and the tetracycline resistant genes, which are highly consistent with the type of widely used antibiotics, showing that antibiotic use affects the distribution of the resistant genes in the environment as the major driver of spread. The influence of the sewage treatment plant could be used as the representative samples of resistance in the gut of human population of 1,000 people from that catchment area. Wastewater treatment plant is an important ARG source in the environment, especially the widely used activated sludge since 1950s. Due to adsorption, antibiotics enriched on the surface of sludge floc could be a significant selective pressure, plus other factors such as high bacterial diversity, higher biomass, short distance between the cells, continued mixing, providing a very favorable condition for the horizontal gene transfer of ART, so-called HGT. This ecological environment is completely artificially created and developed in the recent 60 years. It does not exist in a natural environment. The average generation time of bacteria in active sludge is about 15 to 30 days, which is much faster than the growth rate of bacteria in natural water and soil. This means in active sludge, the enhanced horizontal gene transfer, HGT, together with rapid growth of bacteria, will further enlarge the impact. At the end, the resistant bacteria will be discharged into the environment together with treated wastewater. 1 cubic meter treated wastewater usually carries up to 1 billion total bacterial cells. The continued discharge of a large amount of wastewater definitely will increase the resistance in water and sediment. In addition to the snapshot sample from different environments, long-term sampling tests of the sample of typical environment are also very important, because only in this way we could fully understand the spread and evolution of resistant genes along time. So far, there are limited studies based on long-term sampling of active sludge. We have conducted sampling for 14 consecutive years monthly at the wastewater treatment plant of Hong Kong. And here are the results of 9 years from 2007 to 2015. The results showed that the abundance and the profile of ART changed significantly every 2 to 3 years. Additionally, compared with the soil resistance, the resistance of active sludge had higher horizontal gene transfer potential. Although wastewater treatment plants are considered as critical points in disseminating ARGs, there is a limited study about assessment of the resistance and the metabolism in effluent in a quantitative way. we proposed an absolute quantification metagenomic method by considering the mass of sequenced DNA and also the total mass of extract DNA from a sample. Of course, we assume 100% extraction efficiency and to assess the resistant and also mobile genetic elements in the effluent in the cities of North America, South America, Europe, and Asia. The metagenomic absolute quantification results are comparable to those obtained using qPCR. We propose a risk ranking system based on ART abundance in the unit of copy of ART per cell. Hong Kong wastewater effluents were ranked in the moderate level, that means level 2 and level 3. Using the same method, we established a public platform. Actually, it's an interactive map for researchers and the government officers, all the general public, to search online for the information of the resistance in the wastewater treatment plants of different cities, and to understand the potential risk level based on the global metagenomic dataset of ART. Here is an example, shows the ART profile in the city of Japan. Analyzing the occurrence and the types of resistant genes in different environments is only the first step. More importantly, we must understand its dissemination and HGT mechanisms in the environment. This requires answer to a series of questions, including the host of the resistant genes, the associated mobile genetic element, MGE, and so on. To obtain this information, short sequences are not enough. We need advanced metagenomic measure using long DNA sequence. This is the current focus of attention. Here are some results of our application of the third generation sequencing, nanopore. First, we developed a process based on the nanopore long sequence to analyze the genetic background of ART, identify the host. The result obtained not only give us a new understanding of the MGEs, such as plasmic and integrative and conjugative elements, ICEs, but also provide an important reference to further analyze the development of resistant bacteria and the transfer mechanism of ART in the environment. A total of 10 potential resistant pathogens were detected in our research. Most of the pathogens carry multiple resistant genes, covering 6 types of resistance. By tracking and monitoring this potentially pathogenic bacterial group, we found that 5 of them were present in both influent and effluent of sewage, not being completely removed. Resistance genes associated with MGE, mobile genetic elements, have higher HGT potential, horizontal gene transfer potential, and then high risk than those on the chromosome. Our results shows that most of the ARTs in the wastewater treatment plants are carried by plasmid and integrative and conjugative elements, so-called ICE. The ICE carry 13 subtypes of ARTs, indicating that ICE may play an important role in the spread of ART. 23 ART subtypes carried by plasmid existed in the influent and effluent of wastewater treatment plant of Hong Kong, indicating that this mobile and risky ART were not completely removed and could be an important source of ART in the receiving water. Thus, we should further strengthen the wastewater disinfection treatment. With the wide application of the high throughput sequencing, the whole genome of different bacteria exponentially increased in the public database. Deep mining of this big genomic data and machine learning could help us reshape our knowledge of HGT, horizontal gene transfer, and code selection of ARTs and other resistant genes, and guide our analysis and interpretation of environmental metagenomic dataset. Here, I would like to share 3 projects of us for your reference. In the environmental field, HGT, horizontal gene transfer, is a major mechanism of resistant generation and got a lot of attention. Plasmid play an important role in HGT, but the previous studies on ART associated with plasmid were mainly based on the plasmid of pure culture. This limited our knowledge. In addition, little is known about the interaction between the plasmid and other mobile genetic elements, such as insertion sequence, so-called IS. We developed a tool for plasmid classification, ART annotation, and visualization, and then use this tool to systematically analyze a large number of bacterial plasmid and the genome. Our result shows that most of the ARTs carried by plasmid closely relate to the conjugated plasmid, and there are also a large number of ART transfer network associated with the insertion sequence, IS, indicating the significant role of the joint interaction between the IS and the conjugated plasmid as the universal mechanism of HGT. In addition to this in silico analysis, we also verified the interaction of the IS and the plasmid and the important role in ART horizontal gene transfer by some laboratory experiment. Another example is the type 1 integron, which is an important ART carrier. It is almost 100% distributed in the gamma class of the Protobacteria phylum. Based on our analysis of 120,000 bacterial genome in the public database, especially they enriched in those bacterial families in the human gut microbiome. The 8 categories ARTs on the type 1 integron are related to the aminoglycoside, beta-lactam, and so on. We also analyzed abundance and the genetic association between ARTs and metal-resistant genes based on the big data analysis of bacterial genome. The results show that the bacteria related to human have more obvious co-selection of ARTs and metal-resistant genes. And the relation between the ART and the metal-resistant genes is also much stronger in the pathogenic bacteria. This explains the co-selection of ART due to the metal pollution in the environment at the genetic level. Of course, this in silico big data analysis need to be further validated by the experiment and the real environmental data set. The source of ARTs in the environment mainly comes from hospital, pharmaceutical factory, wastewater treatment plant, and animal farm. Then a question to ask is, could we use metagenome measure as the management tool to determine the source of ART contamination in an environment sample? In other words, is it possible to track the pollution source of ART? We combine the big data analysis of ART with machine learning approach and propose a new method to track the source of ART. First, we performed the feature analysis of 650 global metagenomic dataset of human feces, animal feces, and wastewater natural environment, using the random forest algorithm to establish the predictive model and then configure the samples and adjust the parameter to optimize the model and use simulation data for validation. After that, we conduct MST, microbial source tracking analysis for the real environment sample in Hong Kong and Pearl River Estuary and South China Sea. Yeah. For 5 of the 12 Hong Kong marine sediment sample, the contamination of ART from wastewater exceeded 10%. Such result match the pollution level of this sample very well. In our another study, a large portion of ARG in the sediment of 2 sampling sites at the Pearl River estuary came from wastewater. With most studies on ARGs, people have realized that risk assessment of ARG in the environment is more important than just recording the concentration and the distribution. There are 2 major types of risk of resistant genes. The first one is the risk of antibiotic treatment failure in the medical field, in the hospital, and the other is the risk of the spread and transfer of resistant genes in the environment. Currently, there's no widely used model for environmental risk assessment of ARGs. In a recent paper, we propose an assessment framework based on 3 major factors, including whether the ARG is enriched in an anthropogenic environment, GT potential of this ARG, horizontal gene transfer potential of the ARG, and also the third one is whether the ARG is carried by the pathogen. This framework proposed a basic risk classification and also provide a practical measure to identify those high-risk ARGs and take the first step to move forward from the surveillance to risk assessment. Within this framework, based on the big data analysis of a large collection of both genome of bacterial culture, metagenome of environmental sample, we proposed a draft list of ARG to be controlled with high priority in the environment. Of course, this list could be further periodically updated when there are more bacterial genome and more environmental data set and more knowledge about HGT, horizontal gene transfer, and MGE, mobile genetic elements. Finally, I hope this could be the take-home message. Metagenomic measures are a powerful approach for surveillance of ARG in environment to understand the genetic mechanism and environmental driving factor for the development of management tools and also for the assessment of risk. At the end, I would like to thank the hardworking team, the collaborators, and the funding agencies. Thank you for your listening. Thank you.
Thank you, Professor Zhang. Is there any question that from the audience? So Professor Zhang, may I ask one question?
Sure.
Yeah. So I'm not very familiar with the ARG. But I used to have some strange ideas. So I take a web of my dog, take this tissue to for running antibiotics. And I did find several kinds of different antibiotics. And the thing is that I never use these kind of drugs either in my room or any member of my family use. So could you suggest where those antibiotics could come from?
You mean antibiotics or you mean antibiotic resistant genes?
Antibiotics.
Okay, you mean there are some residue of antibiotics?
Yeah, very strange.
In the sample of your dog? In a sample of your dog?
Yeah.
Okay. Did you check the dog food?
They are, I did. They are not originated from dog food or dog drugs. No, they are human antibiotics.
So I mean, did you take some sample and for the analysis, using your dog food as the sample for the analysis to see whether there are some antibiotics already in the dog food?
Yeah, I did, but nothing in the dog food.
Okay, so that's not the source.
Yeah, food is not the source. Yeah.
Then I do not know what could be the source. You know, if they're not from the food, and also you didn't use antibiotic for a dog, then I really do not know where they come from. If you
Yeah,
talk
yeah.
about the ARGs, then they could be carried by pathogen, by bacteria. Then that's easy to some other transfer exposure pathway. But for the antibiotics, the chemical, if not from the food, and also you didn't use that, not in the water, and we don't know where it come from, because most of them they will not be transferred in the air. Yeah.
Yeah, probably very difficult, right?
Yeah. No possibility, I guess.
Okay, yeah, skip very strange.
Yeah.
Yeah. Thank you for your answer for sure. Yeah. Okay. Yeah, the antibiotics and ARG could be very interesting topic. Yeah. So our next speaker is Professor Jiangwen Chu. Professor Chu is a professor in Department of Biology, Hong Kong Baptist University. His main research area includes deep sea biodiversity, genetic connectivity, and genome level adaptation to extreme environmental conditions. Today, he will present stress responses in corals, winners or losers under climate change. Professor Chu. Yeah. Great. You've already shared. May play your slide at full mode.
Thank you, Chair. Can you guys hear
Professor
me?
Chu, you are using the presenter mode, so we can see every like next slide. Do you use the
I'll
screen?
change. How do I?
Probably you should end the sharing first. End your sharing first. Oh,
let's see.
swap. Yeah. We try this one. Yeah.
So this,
Yeah.
is
Good.
this
Good. Yeah. We are good. Yeah.
Okay. Thank you, Chair. For the introduction, I do deep sea research, and but I'm interested in shallow water research as well. So in recent years, I have been studying coral health, especially predation on corals by sea urchins, and as well as corals' responses to bleaching. So we conduct field surveys, and we conduct laboratory experiments. Today, I would like to report our work on bleaching, coral bleaching. So for those from Southeast Asia, I'm sure you guys know better corals than we do, because you have more well-developed coral communities. You know, reef corals, they are distributed throughout the tropical and tropical oceans, and they support high biodiversity. In South China Sea, we have more than 500 species of hard corals, and they are very important ecologically and economically. However, the status of coral reefs worldwide is not in a good situation. So over the last 50 years, there has been more than 50 years, there has been a decline of global coral coverage by more than 50%, and fisheries by 60%, and over 63% of coral reefs biodiversity have declined. According study of more than 3,000 reefs around the world, there has been dramatic changes of decline of coral baselines over the last several decades. So with the predicted increase of sea temperature by this century, it is predicted more coral deaths will result in the future. So I'm sure you guys all know about this coral and symbiotic relationships. They are the foundation of this high productivity in the coral communities. So this symbiosis is very important for the normal function of corals. However, environmental stressors such as temperature, high temperature, low temperature, hyposalinity, high light intensity, pollution, and sedimentation all have been known to cause coral bleaching, resulting in the breakdown of symbiosis. So there has been several large-scale coral bleaching events since the 1980s. So the most recent one is the 2014 to 2017 global bleaching event, which has affected many coral communities. So I'll focus on the bleaching of Hong Kong corals. So I'll briefly introduce the environmental settings and the 2017 bleaching event we experienced in Hong Kong, and also the implications for the fringing communities in Hong Kong, fringing coral communities. And also, I would like to share with you our follow-up studies. So I'll focus on reporting this bleaching event. First, Hong Kong is slightly south to the tropical of Cancer. So the climate is subtropical instead of tropical. So we have great seasonality with a wet season where temperature can be, water temperature can be 25 to 29 degrees, which is ideal, I think, for coral community development. However, in winter, in the dry season, temperature can drop down to 14 degrees. So during the cold season, you don't expect corals to grow. So they are under marginal environment for coral development. So in Hong Kong, we don't have true reefs, but we do have 84 species of hard corals. So this is a typical situation for the fringing communities of the northern South China Sea, compared to Taiwan, which has roughly 300 species of corals. Our diversity is relatively low. Nevertheless, we can find some corals, such as petit dryer, going to a height of 2 meters above a rocky substrate. So in some places, we still see relatively good coverage of corals up to 70%, 80% substrate cover. Coral bleaching has not been a serious problem in Hong Kong due to the subtropical climate. So as we would not expect this kind of environment to experience heavy coral bleaching. So there has been only a few anecdotal reports, not very detailed reports, since the 1980s. And most of these damages caused by bleaching were relatively local, very small scale, not regional scale, although there's been a massive bleaching event in 1997 during this strong El Nino year. But this is, again, this is not a whole Hong Kong event. In several locations, the impact was quite high, but most of the other sites were not heavily affected. And also in 2014, there was a small scale bleaching event affecting 8 sites. So we conduct a field survey, and we also looked at the recovery, and we found the recovery was relatively good, with more than 90% of the colonies we tagged fully recover after several months. So what I'm going to report is a breaching event discovered in 2017 when we undertook a project with a number of collaborators in different universities, including collaborators in CTU, Dr. Leo Chang, who is co-chairing this meeting. So we surveyed 33 major coral communities in Hong Kong. And during the summer, we found these 6 sites with breach corals. So when we discovered this coral breaching, we conduct video transit surveys. Basically, took videos in the field to reduce the time spent in the field. So after we got the videos, we analyzed these videos in the laboratory using 2 methods. The first method is to use the 5-point sampling method, which means we play the video on the computer screen and we stop the video at a certain time period, and then we overlay the computer screen with several red dots, and we determine the substrate under each dot. For example, whether it's a breaching coral, whether it's sand, whether it's mud, etc. For corals, we determine whether it's healthy or breach. So that's one method. And another method is that we count all the corals that can be found, all the coral colonies that can be found on the transect. Most of these are larger than 10 cm in diameter. We identify them to genus level, and we score each colony as breach or healthy. So just to show you some of the most commonly affected corals. So this is a Physiological under bleaching, and as you can see, this Pavola is not breached. And in this photo, the Pavola is breached, and this is Echopora, and this is varieties. And these are all very common genera of corals in our areas, as well as in South China Sea, I believe. So we found coral breaching in this 6 sites, and affected corals vary from 5.9% to close to 60% of coral cover. So for instance, at this Ayawan, this several genera, Petridrala, Politis, and Echopora, they were responsible for 25%, roughly 25% for these 2, and then 2.4 for this Echopora. Breaching. And then for Barazai, this Echopora and Montipora, these are prey corals. They were responsible for most of the breach area. So as you can see, basically several coral genera, they are responsible for most of the breaching we discover in Hong Kong. And only 6 of the sites had the corals undergoing breaching among the 33 major coral communities we surveyed. So that's the 5-point method. Using the colony count method, we found a similar result. So we found roughly 20 to 56% of colony breach in this 6 sites. And in Barazai, where the prey corals are dominant. The breaching was most serious. And at the coral breach inside Hoi Hawan Marine Park, most of the breach came from Pavona. So that's the observation of the field situation. After this observation, initial observation, we conduct tagging. So we tagged roughly 200 colonies, less than 200. And for each breach colonies, we use a steel bar with a label to tag the colony. And after 1 month and after 3 months, we went back to the site to check the recovery to see whether they have died or they have recovered. So this is the situation. We tagged Pavona, Patridrala, Goriopola, Turbinaria, Ecopola, Poritis, and Lepistria. These were all the coral colonies that had suffered from breaching. And then after 1 and 3 months, we went back to check the recovery. So this shows the recovery of this different genera of corals. So overall, we see most of them have, most of the coral colonies had a good recovery of close to or over 90%. Only Goniopola had a relatively low recovery rate, and that's because probably due to the few colonies available for tagging. But overall, as you can see, the recovery was quite good. So we tagged roughly close to 200, but we only recover 137 in the follow-up surveys. Among this, 80% have fully recovered, and roughly 20 have partially recovered, and roughly 3 had suffered from mortality, which means they died after breaching. And noticeably, those that eventually die, they are mainly ecopolar and some multipolar. So these are pericorals. So just look at the before and after photos of this coral colonies. So this is a Pathogens before and after recovery, and Poritis as shown over here, and then as shown over here, recover. And ecopolar breach colony, and then over here, a recovered colony, and Pavona as well. So these are partially recovered colonies. So as you can see, this was a breach colony, but only a small patch of the coral remain, but this is fully recovered, all right, this part. But the other sections were missing after breaching. So after they die, they probably become very fragile. And also the same for this colony over here. Only this part recover, and this part is dead. So we would like to know what factors trigger the breaching after observing this phenomenon of breaching and recovery. We didn't have environmental sensors at this size, but in Hong Kong, Environmental Protection Department had a network of environmental monitoring stations. We chose one that is relatively close to our site, and we look at several environmental parameters: salinity, chlorophyll, and DO, and temperature. we compiled the data from 2007 to 2016 on this side, and the 2017 data on the right-hand side. As you can see, we didn't see much changes in the chlorophyll. So we look at the changes in different months. We didn't see much changes in the chlorophyll, much differences between this particular year, 2017, and 2007 to 16. We didn't see much differences in chlorophyll, DO, and temperature, but we saw a lot of differences during the summer. Surface salinity dropped from roughly 30 parts per thousand to close to 15. That is a lot of fresh water. So there was heavy rain before the salinity drop was recorded. And as you can see, the data is not very dense. So every month there's only one data. So we might not have captured the lowest salinity happen during this particular year and before the discovery of coral bleaching. So this led us to conclude that maybe salinity was the culprit. Low salinity was the culprit of this particular event. So from our study, we found very mild impact of coral bleaching. However, if you recall, in 2014, we had a similar small-scale impact. And in the future, based on the prediction of the Hong Kong government, there may be more extreme rainfall, extreme temperature event, high temperature events. So this kind of cumulative impact may not, should not be ignored. And also, we found that there's selective bleaching of several coral species, as well as selective mortalities, especially the playlite or ecopola, as well as some multipolar colonies. So this kind of cumulative impact should not be ignored because they have the potential to shift the community from a more diverse community to a more monotonous community dominated by, for instance, Pavona or the varieties. So we feel that in the future, if this kind of events happen again and again, there could be winners, losers, and losers in among the existing corals of Hong Kong. So this is the field study. But during this field study, we also collect some samples. We collect the healthy sample, we collect the normal sample, normal looking sample in a breach colony, and we also look at the breach. We also collect the breach tissue, and we also look at the recovery tissue. So we collect this kind of tissues. We also run transcriptome analysis to determine the pattern and in gene expression to look at what happened at the metabolic level, at the immunity level. So recently we have published a paper on the host and symbol transcriptome responses during the natural breaching and recovery in Pavona. And we are analyzing some more data on Pachydryla as well as Montipola. These are very different species. And also they are different in their responses to breaching as well as the recovery, to look at whether their metabolism are different, whether their immune system responses are different, and whether they are under different levels of oxidative stress. And also with the support of UGC Hong Kong, as well as ECF, Environmental Conservation Fund, we have established a laboratory system. This is one section of the system where we have individual tanks that allow us to do laboratory culture of corals and conduct laboratory experiments to determine temperature and salinity stressors to corals. So we measure the algal concentration, we measure chlorophyll, we measure also transcriptome of the host and symbol, and we also measure the bacterial composition to see how this kind of different patterns of the symbiosis are broken and doing a breaching. And also we use 9 species of corals to do this kind of experiments. We have just complete this kind of experiments. We haven't got time to do the comparison, but our initial results shows that branching corals, they are very susceptible to temperature, high temperature, but all of the corals, they are very resistant to low salinity. We use low salinity down to 21 parts per thousand, but most of these local corals, they can survive under this kind of low salinity conditions for up to more than 1 month. So that's very surprising. So I hope in the future, I will, in the near future, I'll be able to report the results. So also with this kind of study, we can gain some insights into how different molecular pathways are involved in their differential sensitivity, underlying their potential fate in face of global environmental changes. So that's all I would like to report. I would like to thank the funding bodies as well as my lab members who have contributed to this work, Yanjie, James, and Yolanda and Jack. Thank you very much for your attention. So
yeah, thank you, Professor Chu. Is there any question from the audience? Oh, actually, we do have a question from Professor Cabansat from University of Philippines, Visayas, Tacloban College. The question is, there are already studies focusing on high temperature resistant species. Would you recommend these species to be transplanted to reefs adversely affected by bleaching? Why or not?
That's yeah, I have read some papers considering the transplant of high temperature resistant species. But in Hong Kong, we are in the beginning to understand which species may be more susceptible to temperature and salinity stressors, and which one may be very resistant. I hope after we discover this kind of phenomenon, we can consider this in the future transplantation experiments. Currently, I think the restoration program is rather cowboy style. I think they only consider which species grows faster, and they tend to use Echopora. And in our experience, all species of Echopora we test in Hong Kong, they are very susceptible to high temperature. So if you don't consider this temperature susceptibility for a restoration program, then there's a high likelihood that your project would not be successful. So thank you. I'm not sure if I have answered your question.
Yeah, thank you very much. Yeah, I do have a follow-up question. So besides the temperature resistance, do we need to consider other factors like the suitability of the coral species? I'm not very familiar with this area, but
yes,
probably,
Chair. I think we only look at the phenomenon right now, but actually there could be a genetic basis for their differential temperature resistance as well. And also in this coral transplant restoration program, we also need to consider the genetic background.
Sure. We have another question. Have you assessed if previously bleached recorded corals have same reproductive capacity as unbleached?
Well, this is a very good question. We haven't had the opportunity to look at their reproduction, but I hope we are considering this collaboration with a colleague in Chinese U, Dr. Abel Tree, who has conducted some coral reproductive experiments. So hopefully we will have data. I think their reproduction must be compromised, but we don't have data. Thank you.
Thank you very much. We have probably the last question due to the time issue. So another question will be from the presentation. The recovery rates were noted after 2 to 3 months. How is this compared to recovery rates from other areas?
I think will be comparable. Most in depends on the if the condition improves, some corals may be able to recover. But if the condition, the worst condition, the worst environmental conditions persist, then corals may die. So maybe quite difficult to compare recovery rate from one location to another because the situation may be different. So you need to look at the environmental conditions during and after bleaching. So we were quite lucky. Probably in our case, the salinity, low salinity event did not last very long.
Okay, thank you very much.
Thank you, Chair.
Yeah, thank you.
And thank you for the audience. So I'll stop sharing.
Yes, please.
Okay.
So our next speaker is Dr. Leo Chang. So I've already introduced Dr. Chang. So his presentation topic today will be the recent progress and future research on Cicoteria causing marine benthic dinoflagellates in Asian Pacific regions.
Yeah, thank you, Henry. So
Thank you.
you can see my PowerPoint, right?
Yes.
So no problem, right?
Yeah.
Yeah, so I don't need to introduce myself. So I will go to my presentation. So first of all, what is Cicoteria fish poisoning? I think for most of the friends come from Southeast Asia, they may know that. So Cicoteria fish poisoning is a foodborne illness caused by the presence of cicotoxins in the flesh and visceral of tropical and subtropical coral reef fish. More than 400 species of bony fish at all trophic levels have been identified as cicotoxic. Cicotoxin is produced mainly by dinoflagellates that are usually attached on macroalgae, sand, coral rubbles, and seagrass in the sea, especially in tropical areas. That means the Southeast Asia. Organisms in the group of benthic dinoflagellae have been implicated as the causal organism of CMP, such as Porocentral, Curia, Ostratus, Gambidisca, Fukuyu, and Aphidina. However, many of these species have been reported to produce various types of toxin, but it is believed that only toxin from Gambidiscus and its closely related species Fukuyu reveal the symptomology corresponding to the ciguatera fish poisoning illness. So this is the SEM and then the live photo of Gambidiscus. So they are sized just as the diameter of a hair. So in fact, if you have good eyesight, you can see them under the sea. So there are more than 18 species in the Gambidisca and Fukuyu families. They produce gambiotoxins and a suite of ciguatoxins. So what is ciguatoxins? Ciguatoxins produced by dinoflagellates, right? I already mentioned that from Gambidiscus and Fukuyu. So more than 50 analogs of CTS has been identified. So they are polycyclic eaters. So they have a leather-like structure. They are very stable, resistant to heating and freezing. So you cannot get rid of them in the fish. So they also resist mild, acidic, and basic conditions. And they are lipophilic, higher concentrated in fatty acids. So if you intake the ciguatoxin, they will stay in your body forever. So from our many years study, we start the whole food web study from Gambidiscus, and then to herbivore fish, to carnivore fish, and then to seafood consumers. But today I only will focus on the benthic dinoflagellates. So at the beginning, it's very difficult to get the ciguatoxin fishes. So I went to the Republic of Kiribati and sample along the most endemic area, and then we have ciguatoxin concentration with the location. So after we get this map, then we dive into the sea, and then we try to isolate dinoflagellates. So in fact, these fish are our detective. So let us know how the distribution, abundance, and density of Gambidiscus in the Republic of Kiribati, mainly the western side of the island. So from the tree, we isolate a lot of Gambidisca highly toxic species, and you can see this species is very toxic compared to other species. So in collaboration of Professor Choi Peng Liu and then Ludo Ding from Malaysia and China respectively, we identify a new Gambidisca species type 6 as the Gambidisca balachi. So most of my students are now working on this species. So you can see the ciguatera is an issue of local, regional, and global importance. In the past, CMP was only endemic in the tropical and subtropical region between latitude 35 degrees north and 35 degrees south. And ciguatoxin, according to their distinct structure and geographical distribution, are classified into 3 groups: Caribbean ciguatoxin, Indian ciguatoxin, Pacific ciguatoxin. So this make the ciguatoxin research more challenging because they have a diverse structure and toxicity. So now there's widespread of ciguatera fish caught from the endemic region to other parts of the world because of the high demand for coral reef fish for seafood. So which lead to the inter-regional trade of large volume of these fishes into different areas around the world. CMP so now become increasingly prevalent with the effect of globalization as well as the climate change. So now the ciguatoxin is widespread everywhere. So you can see this is the global distribution of ciguatera fish poisoning. So global warming may also lead to an increase in CFP cases. So an observable positive relationship between CFP incidence and average sea surface temperature has been discovered. Why? Because the temperature is the primary environmental factor regulating growth. Can be discussed tends to growth at better at higher temperature. And Professor Chu already mentioned there are global bleaching due to global warming. And in fact, more coral bleaching provide more surface for the proliferation of these toxic can be discussed species because they need to attach to those turf, grow on those bleached coral or dead coral. So this is a study by Marina China, my collaborator in French Polynesia, and she discovered that coral mobility may be the factor in the genesis of natural occurring growth of Gambidiscus by providing more new surface for colonization by this opportunity macroalgae that are ideal hosts for Gambidiscus. So these observations support the hypothesis that CFP outbreak can occur in response to disruption of reef. So more disruption, more toxic fish. So in the future. So the time lag may be only a few months for them to grow. So you can see that the hot season, the coral bleaching, and then later you find the high maximum cell abundance and toxic production found in this area by the Gambidiscus toxicus. So my collaborator, Professor Lin Boti and Cui-Ping Liu also find that these disturbances that alter benthic substrate type can have profound effect on the composition of species present. The highest Gambidiscus cell abundance was detected in the microhabitat dominated by algal turf associated with coral rubble fragments. And this is the summary. The climate change, local stress, like overfishing, pollutant, will cause disturbance to the healthy coral reef. And then this degraded coral reef, they will be the best surface to host these beta species, the benthic dinoflagellates. And then their toxicity will cause the shift of the alteration of the trophic transfer, and then will change the species diversity in that coral reef. So don't underestimate the ability this tiny dinoflagellate to the whole coral reef ecosystem. So this is my prediction 10 years ago. The number of CFP may further increase because the overexploitation of coral reef fish, that continued degradation of coral reef due to global climate change and other anthropogenic disturbances, as well as the expanded distribution and elevated abundance of toxigenic Gambidiscus globally. So my prediction is that cicatrice fish entering market around the world is going to increase. So in 2017, my collaborator, Professor Lin Boti and Cui-Ping Liu, and also my Vietnam collaborator, Ha Wit Tao, they already discovered some local fish, the snapper, are identified contaminated with cicatrice. So this is not import from other area. This is a local find. And also, Dr. Tao from Vietnam, the Institute of Oceanography has carried continuous surveillance on the CFP, and she discovered from the 2008 to 2018, within these 10 years, there are many local CFP incidents has been identified, and mainly from the red snapper and the moray eel. And she found that CTH1B is responsible for snapper poisoning in Vietnam. So this is the map showing the Pacific region, the distribution of Gambidiscus species. So after literature review, we find that there, in fact, a lot of Gambidiscus identification in Japan, Korea, Taiwan, Okinawa, China, Hainan, Vietnam, Malaysia, Indonesia, etc. And then in Japan, the Okinawa has found different kinds of Gambidiscus species. And Professor Leo Chang from Chilean University has conducted systematic sampling, and he found that 3 species has identified as Gambidiscus in Chinese waters. So, so we are able to confirm some Gambidiscus from Malaysia and Curibus, and with several new records in collaboration with Professor Liu and Limbote. So in response to the ciguatera fish poisoning in Vietnam, we actually lead our research to go to conduct some benefit dinoflagellate sampling in Vietnam, and we discover there are many Gambidiscus species and other benefit dinoflagellate in the degraded coral reef. And in collaboration with international academia, especially I think from the Southeast Asia, the Vietnam, Malaysia, Indonesia, we will conduct a future research to investigate the bioactive potential of benefit and antiphatic toxic dinoflagellate and their associated microbiotas. So you can see that there are many people research on pelagic harmful algal blooms, but there are still not many scientists working on benefit and epiphytic toxic algae. Unlike planktonic microalgae blooms occur often in the upper part of water column, so you can see them. There's no visible environmental phenomenon such as blooms and red tide can be easily linked to benefit dinoflagellate, and then outbreaks are often overlooked. So this is when we go to study the benefit dinoflagellate sampling, we always observe this kind of underwater blooming just above the coral and cause coral bleaching, massive death of invertebrates. So we develop our unique sample procedures. We design some kind of in situ auto sampler we can bring down to sample the target dinoflagellate. And this is a short video showing our so everyone can see the video. So we use this sampler to conduct our trial survey sampling in Hong Kong November 5th, and then we find 5 species of this benefit dinoflagellate species. So the identification are still ongoing. And once we get this single cell, we can mass culture to study their toxicity and other kind of applications. So we will develop into 2 kind of SOP. First is purification of toxicity, and then the second part is species identification. And eventually, we hope that from this dinoflagellate, we can produce a low price toxicity to support high impact biomedical pharmaceutical research. So this is just a brief summary of our recent research. So at the beginning, we start the ciguatera research for food safety monitoring. And then we purify 74 micrograms of cigotoxin from the marine eel, and it costs 1.73 million. So the toxin standard is very expensive, $3,000 per 1 microgram. And then we developed 2 rapid, robust, and reliable analytical methods for toxin analysis. And then now we move on to the identification and characterization of bioactive secondary metabolite from those bioactive dinoflagellates. So use molecular networking to identify new cigotoxin analogs. As well, for drug development, we also use the molecular networking to isolate secondary metabolite for pharmaceutical applications, such as the control sensing anti-inducer and inhibitor discovery from marine microalgae derived bacteria. And also, most of people focus on cigotoxin because they are lipophilic, so they will transfer along the food web, but not much research are on metotoxin. So our research also focused on the metotoxin. They are water soluble. Firstly, we discovered 7 super carbon chain compounds, and more than 10 metotoxin analogs were discovered, and their structure formula were now elucidated by MS spectrum. So why this is exciting? Because most of CTS analogs are new compounds, and these macromolecules are not protein-based molecules, which make this discovery very exciting and meaningful. So I just show you some of our new discovery, metotoxin macromolecule. Some are known compounds, some are new compounds, and some. So this is all exciting results from this. So based on our 10 years research experience, we also provide training workshop and introduction of scientific diving for marine dinoflagellate sampling and processing. So we conduct a training workshop in September 2018 for UNESCO WestPac. And now, thank you for your attention and acknowledge the funding organization. Thank you.
Thank you very much. Thanks, Leo. So before our break, ladies and gentlemen, would you mind turning on your camera so we can have a group picture again for all the participants? Thank you very much. So yeah, great. So Grace?
Yep.
Yeah, would you mind take the pictures?
Of course. So let's give, say 15 more seconds for people to turn on their cameras and get ready, and then we can take the photo.
Great.
All right, so as usual, I'll also take a few. So on count of 3, 3, 2, 1, cheers. Okay, and another one. 3, 2, 1, cheers. Well, because we have a few more participants, so I'll have to take 2 more photos. Okay,
Yeah,
sorry.
sure.
3, 2, 1, cheers. And one final one. Ready? 3, 2, 1, cheers. Okay, thank you so much.
Okay, so, sorry, we have a question actually for Leo. Yeah, so the question is, in the late 1990s, I've been working with Dr. Fukuyo and Dr. Mari Singat isolating ciguatera benthic dinoflagellates, which we've deposed in central Philippines, Seguros and Reef. So is it possible to collaborate? So that's an offer for collaboration for Leo.
Yes, Francis, certainly, I think this is the purpose of today's symposium. And we want to share our latest research, and then we also look for collaboration, especially in Philippines, because we have a collaborator in Thailand, in Malaysia, in Indonesia, and we don't have a very solid collaborator in Philippines. So definitely, we are very pleased to collaborate with your team or other Philippine researcher. As you can see that we train people along our sampling. So whenever we go to Vietnam, we go to Thailand, we will train local scientists. And also we will try to develop some kind of international standard for this kind of sampling method for comparison, comparative study and collaboration. So thank you for your kind offer.
Yeah, we have several raising hands. So how can I open up? So do we need to open up? So Dr. Jing Ling, do you have any question?
Yeah, Leo, thanks for an inspiring talk. So your current focus is on the benthic and epiphytic, right? So I'm wondering, is there any dynamic interaction between the pelagic and the benthic system in terms of those algae and the algal toxin, those things? Yeah, thank you very much.
Yeah, I think those benthic dinoflagellate have some kind of stage they will become pelagic.
Okay.
So I think, and also they also secrete this lipid philip toxin into the water column. So this toxin will be traffic transfer to zooplankton, etc.
Okay, so they are not strictly isolated. So there are still some dynamic interactions.
Yes, still some kind of linkage.
Okay,
Yeah,
good.
But they prefer benthic, right? Yeah, but sometimes maybe if the condition is not good enough, so this is why you see those dinoflagellate can transport to other part of the world through the water current or the ballast water.
Okay, thank you very much.
Yeah, thanks, Nathan. Lenny, do you have anything to say?
Yeah, I just want to say that there is already, and we actually find many benthic diatoms and dinoflagellates related to ciguatera shellfish poisoning, but not very. Since the Philippines is mostly focused, and the big problem here in the Philippines is paralytic shellfish poisoning caused by Pyridinium bahamense. That's why the focus of most of the researches in how harmful algal blooms are in Pyridinium, but we do see many of these in our samples, but not so many people working on it. Although there was one research before in Bolinao, working on Gambierdiscus, they set up a mesh in the bottom so that it can collect it can attach that mesh there in the bottom so they can collect those kinds of dinoflagellates. So yeah, it's a good actually, since there are not so many people working in the Philippines on Gambierdiscus.
Yes, I'm not deliberately left out Philippines. I try to link up with Philippine researcher in a harmful algal bloom many times. I approached Professor Rajada, right? And then it seems that all of them are interested in pelagic research, like the paralytic shellfish poisoning, DSP, etc. Right? Not many people are interested.
Yes, because that's the main problem. Yeah, and paralytic shellfish poisoning is just really causing deaths here, and that's why
when you see those kind of PSP or DSP, they are water soluble. So in fact, if you use separation, they will be cleaned off.
Depurated, but
will be more problematic in seafood poisoning in the future.
Yeah, but there is still until now deaths in paralytic shellfish poisoning, and
Yeah.
the system is not still very good. How the early warning system, like the warning system. So that's why many are focused on that. But yeah, I agree that we should not forget other harmful algal species that are there, that we also have here in the Philippines. There are a lot, and even in Ciguatera fish poisoning, there was actually a case in Iloilo of Ciguatera fish poisoning already, but again, nobody is pursuing this area.
Yeah, we will be very pleased to collaborate with Philippine scientists, and then we can go there and then sample with you and then collaborate. So I go to Philippines for diving training many times, but only for training, not for scientific research. Yeah, so please keep in touch and email us if you are need our help, because I think in the regional.
Yeah, we would. Actually, I know Dr. Lim Potin and Dr. Chin Liao, so I thought already covered in that area. But if you are looking for other samples in the Philippines,
yeah, definitely. We are eager, looking forward. Yeah.
Yes.
Yeah. Thank you. Yeah, Lini.
Yeah, thanks, Lini. Probably we will have only 1 minute short breaks. So we will come back in 1 or 2 minutes, and Dr. Leo Chang will be the following chair for the remaining session. So I will see you pretty soon.
So let me Dr. Jimmy actually is our old friends in CTU. I think Jimmy graduated from CTU, right, Jimmy?
Yes.
Yeah. So Dr. Jimmy is an assistant professor in Department of Civil and Environmental Engineering in the Polytechnic University. In fact, I also graduated from Polytechnic University. So his main research area includes air pollution and human health, airborne and foodborne transmission of antimicrobial resistant and pathogens, and pollutant-induced immunosuppression in Chinese white starfish. And I also understand Ginny is also working on the lipophilic toxin in dolphin now, right? So
yes,
an area.
Yeah, hope to collaborate with you as
Definitely,
well.
we can collaborate in the future. So today, Dr. Jingling will present implication of antibiotic resistance in marine culture for coastal environment quality and seafood safety. So let's welcome Jingling. Yeah.
Okay, thanks, Leo, for the kind introduction. So today's topic, I think that today's topic would concern a lot to our Southeastern Asian friends. And okay, so we are looking at the global trend of the marine culture farm and antimicrobial use. Okay, so now to achieve this is really due to the driven by the increasing demand for high quality food, okay, supplied by the marine from seafood and including marine culture or marine capture. And you can also see over the 2 decades, right, past 2 decades, the marine culture production has steadily surpassed the marine culture and becoming a dominant supplier of the seafood supply. Okay, now this is also very evident and increasing trend of the antimicrobial consumption in aquaculture, and in 3 years ago, and all the way projected to 10 years later. Okay, so this really concerns our Southeast Asian countries, including Indonesia, Malaysia, Thailand, Cambodia, whatever. Okay, and on the global scale, on the global context, okay, so the antimicrobial consumption will still be increasing, okay, and to support this kind of marine culture activities, okay, over in the next decade. So one important thing, as Professor Zhang Tong in the very first talk, he has given an paranomic view on the antimicrobial, the antimicrobial resistant genes, antimicrobial resistant pathogens, all those things, right? And which can be shaped by human activities, one of which is the aquaculture activities or marine culture activities, okay, freshwater culture and marine culture. So as we can see from these examples, even from the Baltic countries, okay, so the aquaculture really changes the profile of antibiotic resistance and those mobile genetic elements, okay, associated genes in the surrounding aquatic environment. And this is another case of like a very intensive fishery and marine culture industry, okay, industrial marine culture production, okay, very intensive. So the fish meal application really induces the antimicrobial resistant gene propagation, okay, in the surrounding water, in the sediment, for example, in this case. So which in turn, the feedback is the global rise of those antimicrobial resistance pathogens, okay, the increase as an emerging threat to seafood, okay. So there's a two-way. One is influencing the surrounding environment, okay, through release of antimicrobial resistant genes and as well as their carriers into the environment, which may in turn through the feedback loop from seafood to the coastal consumption groups, okay, which pose this kind of emerging microbial biological food safety threat. All right, so on the global scale, okay, so according to the WHO, the foodborne and the waterborne diarrhea diseases, okay, is an emerging, it's a very serious issue, okay. So every year 2.2 million, over 2 million deaths, okay, occur due to this kind of foodborne or waterborne diarrhea diseases, okay, around the world. So in China, for example, foodborne disease are represented by almost 300,000 individuals affected and more than 2,000 deaths during 1994 to 1994. This is an old record, okay, and among which 40 to 50% are caused by predominantly zoonotic microorganisms. So from a One Health perspective, okay, so the environmental community and the clinical community need to work together on a concerted efforts to tackle this kind of One Health issue, okay, of the antimicrobial resistance spread along the human animal environment loop. So in which marine culture is a section, okay, so that we really want to reverse this kind of projected trend of the antimicrobial resistance infection and the antibiotic failure, okay, project up to 30 years later, okay. All right, now antibiotic resistant genes are the basic unit, okay, the genetic basis of antimicrobial resistance. But this is not like not really like synthetic or human-induced pollutant, okay, chemical pollutant. So it is really naturally occurring. It is really naturally occurring as a kind of host defense from microbes because they also need to compete with other microbes, okay, other competitors in the environment. So antibiotic resistant genes, okay, is an ancient and then natural occurring. Okay, you can find in the cave, right, back to many thousand years ago. And also our previous research comparing the pristine down to 4,000 meters deep in the South China Sea, okay, in the South China Sea from their sediment, compared to the sediments in the more urbanized estuary, the so-called Pearl River estuary, okay. So we did this kind of metagenomic analysis and compare them. So we can really see this kind of a gradient, okay, and through this kind of from pristine to the anthropogenic impact, the increase in anthropogenic impact really drifted this kind of natural antibiotic resistome, right, increasing their diversity, alter their diversity and abundance, as well as their mobility and carriers. All right. So now down to this topic, the marine culture in today's topic. Okay, so we really want to ask a few key questions. So specific to marine culture, how does marine culture alter the structure and the properties of antibiotic resistance and its associated microbiomes in coastal waters compared to pristine oceans as a global baseline? And may such changes have significant implications for coastal environmental quality and in turn for safety? So to this end, we conducted a sampling, a very comprehensive field sampling in one accredited fish farm, okay, under our Hong Kong government so-called Agriculture Fishery Conservation Department, okay, AFCD accredited fish farm in the east coast of Hong Kong. This is called Cyclone. Now compare this side of the sea is relatively clean now in terms of water quality, and compared to the western part, which is more influenced, subject to the pressure from the Pearl River estuary, as we shown in the previous slide. Okay, from the mainland China, the land-based runoff. So we sampled a few common economic fish species and also the surrounding environmental samples like sea, surface water and sediment. Okay, first we did perform the phenotypic screening. Oh, no, we basically isolate the bacteria, okay, from these fish species, and we subject those isolates to Sanger sequencing and PacBio sequencing for their whole genome analysis, right? So to understand their identity and also the genome structure, and also phylogenetic relationships with known pathogen isolates, okay, strains in human or in other matrices, okay. So we also conducted the antimicrobial susceptibility testing, okay, to see their phenotypic resistance types, okay. And at the same time, we also conducted the genome type assessment, just like Professor Zhang Tong just mentioned before, the metagenomic sequencing, right? So basically, we extract the genomic, again, we extract the genomic DNA, okay, from these samples, right, and purify them, okay, and purify all sorts of things, then conducted the metagenomic sequencing, okay, to understand the structure of the microbial communities, the host taxa, the potential mobility of the ARGs. So, and also we included one site, okay, from North China, okay, from northern China, so in coastal city Yantai, okay, which also houses a conventional industrial marine culture system without recirculation unit. Now then we try to compare, okay, now we need to find a baseline, okay, from the global pristine ocean. Now this kind of Terra Ocean expedition and provided a very good platform, okay. This is a global expedition in major oceans. So they sampled a lot of surface water samples as well as other as well as other samples. Okay, and submitted all the metagenomic sequences. Okay, onto the website. It's available online. So they include those surface water layer and the deep chlorophyll maximum layer, as well as the even deeper, okay, mesopelagic zone. Right. So then we compared, okay, we compared our own we compare the pristine surface ocean, okay, globally as a baseline with our marine cultural resistome and bacterial communities. Now we can very clearly see that the marine culture really all activities altered composition of the bacterial communities in marine culture waters and the surrounding environment. Okay, as well as we can see this kind of marine culture induced activities. Okay, also really suppress the biodiversity, suppress the diversity of bacterial communities in marine culture waters compared to the global ocean baseline. And in terms of a lot of indexes like species richness, Shannon and the Kell index. Okay, and we also seen this kind of ARG profiles also were altered in the marine culture waters compared to the pristine surface water. Okay, so and we also see one interesting thing is the richness of the ARG host. So which means those bacteria, okay, taxa, carrying the ARGs, their richness did not change, but their identity shifted in marine culture impacted coastal waters. All right. So, and we also observed the increasing proportion of ARG carrying contacts with MGs, okay, which means the MGs in this case, which means that the ARGs are the potential mobility of the ARGs are increasing. Okay, so maybe more easily transmissible to other species. Okay, so the overall, okay, so we can really see compare the genetic context between the marine culture resistome and the pristine surface ocean, and we can see that in the marine culture, the ARGs are more so marine culture environments more harbored more by in the marine culture environment, okay, were more carried by the pathogenic hosts, and the frequency and the co-occurrence with the mobile genetic elements are increasing, indicating high potential for horizontal gene transfer compared to the pristine surface ocean baseline, which in turn means higher environmental health risks. Okay, so for the phenotypic, okay, because we just talked about the genotypic analysis, then we really isolate certain species, okay, resistant species, potential opportunistic pathogen species belonging to the Staphylococcus, okay, so has been isolated from those fish edible tissues. Okay, so they're carrying really multiple drug resistance, and we use this a online pipeline, so-called MetaCompare, to rank the resistant risk, okay, between the fish edible tissue and our also ongoing study subjects like the urban air, right, inhalation, also drinking water ingestion. So the preliminary results really show that the resistome, okay, risk is maybe the ingestion of this kind of fish edible tissue may represent a more important role, but whether the cooking would reduce the risk or not, it's not clear. So maybe we can also have a further testing on that. So in short, for a summary, okay, so the pristine oceans are featured by naturally occurring stable antibiotic resistomes. Okay, so no matter how deep, okay, the ocean is, the naturally occurring antibiotic resistome, their profiles are relatively stable, okay, across the ocean gradient. And also their potential for horizontal gene transfer is quite lower. And also there's the host ranges of most non-pathogenic bacteria is very narrow, okay? And the marine culture really induced a significant loss of bacterial diversity. Now, just like think about antimicrobial use, okay? And the industrialized, westernized lifestyle really reduced about microbial diversity in our human gut. So we think this kind of mimic, okay, marine culture induced a significant loss of bacterial diversity and a contrasting increase in mobility potentials and the pathogenic host ranges of ARGs. And also resistant pathogens in marine culture products may have some implications, okay, for foodborne health risks via seafood consumption to our coastal consumers. All right, but we still have a lot to do, okay, to substantiate our claims. All right. So eventually, I really want to thank our Stakey Laboratory of Marine Pollution to provide this, to support this collaborative research. Thank you very much.
Yeah, thank you, Jamie, for this very interesting talk and important talk. I think the overfishing is a global problem. So mariculture will be very important for provide nutrition to human, right?
Yes.
Yeah, but I think now the mariculture is very dense.
Yeah,
and
very
with
dense.
a lot of antibiotic.
Mm.
Yeah, so how can we solve this kind of problem in the future to provide safe and seafood?
Yeah, I think maybe one possible option is using two possibilities. Okay, one they're actually arousing a lot of interest. One is using probiotic to control this kind of pathogens, right? Because fish disease, parasites or pathogen, the fish pathogens are also a threat to those seafood species. That's why use of antimicrobials is currently is a must. Otherwise, the fish farmers will lose a lot of fish supply. Right? So, but in the future, an alternative could be like probiotic. I think James, are you also working with Kevin? Right, Kevin in our ABCT department, right? Is kind of doing this kind of pilot trial. And another control strategy may be using the so-called phage therapy. Phage therapy is actually an ancient tool to control the bacteria, which in this case to control the pathogens. You use their natural enemies. Yeah.
Yes, thank you, Jamie. So maybe we don't have time. So
okay.
So maybe we can left those discuss in the discussion session.
Yeah, sure, of course.
Yeah. So our next speaker with Dr. Gordon. Let's thank you, Jamie, and then
thanks.
Welcome our next speaker, Dr. Gordon Gi. Dr. Gordon Gi is an associate professor in Department of Chemistry, City University, Hong Kong. His main research area include RNA biology, chemical biology, nucleic acid, gene quadruples, gene regulation, and atoms. Today, he will present universal biosensors for pollution monitoring control. Very useful. Welcome, Dr. Gao.
All right. Thank you, Leo. So since I hope you guys can hear me well. So my internet is not that good, so I will let Phoebe help me to play the video that I pre-recorded yesterday in office.
Yes, please wait a moment.
Thank you.
I think, yeah. Is the sound good?
No. I think you might need to share sound when you share the video too.
Yeah, I'm sharing the video. Can you see the video?
We can see the video, but no sound.
But no sound. So I play from the beginning.
There's still no sound.
No.
Maybe, Dr. Can you present?
Yeah, let me try on my side, Phoebe. I maybe I know how to fix it very quickly. If not, then I can present to you.
Yeah.
Let's see. Yes, quick. Okay. Might be a little bit tricky. Let me know if you hear the sound now. Let's see. Thank you for the opportunity for me to speak.
Okay,
So my name is Dr. Choral. You can call me Kit. So today I'm going to talk about the universal biosensors for pollution monitoring. So there's a big problem in here. So globally, 80% of the wastewater flows back into the ecosystem without being treated or reused. And most of the problems relate to water qualities are caused by intensive agriculture, industrial production, mining, and untreated urban runoff and wastewater. And you can see these few articles and websites from here. And since the 1990s, water pollution has worsened in almost all rivers in Africa, Asia, and Latin America. And about 1.8 billion people use a source of contaminated drinking water, putting them in high risk. So the cost of water management are greatly outweighed by the benefits to human health. And looking into Hong Kong, so the sources of these waste of water pollutants are mostly coming from sewage and including antibiotics and chemical contaminants. And then they go into the seawater and river. Wastewater also can be caused by the agricultural livestock, and that these provides the nutrients for this algae in here, and these algae can, overproduction of this algae can generate the toxin, which could also be problems to the seawater and river, and then to the marine animals. And we are in particular interested to monitor the antibiotics using that as a initial chemicals to interact with, to detect. So antibiotics. So what's antibiotics? So antibiotics have been used for decades as human medicines and veterinary drugs to treat bacterial infections. The majority of the antibiotics are resistant to biodegradation in nature, so meaning that they cannot be eliminated completely in these sewage treatment plants. So they will be existed in the marine environment for a prolonged period of time. And this could cause a lot of problems, including the AMR. And these are some classes of antibiotics that we will talk about. And I'm sure I don't need to say too much about AMR, and just a few key things listed in here. So it has already cost 700,000 humans' lives worldwide annually, and is expected to increase over the next 20 years if we don't do anything about it. And it's worth to mention that the WHO have declared in 2020 that AMR will remain one of the 10 biggest threats to public health in the world. So despite the substantial interest from environmental scientists, only a few studies work on the occurrence, distribution, and fate of antibiotics in Hong Kong. The deficiency in this study can be attributed to two major causes. So one is there's a lack of efficient methods for antibiotic detection, and there is also an absence of practical tool for on-site analysis. So what our project is aiming to develop some efficient technologies for continuous and effective surveillance of antibiotics residues. In Hong Kong, Victoria Harbour almost receives 20 billion liters of wastewater discharge from the surrounding sewage treatment plants every day. And as mentioned previously, many of these antibiotics that are detected are resistant to biodegradation in nature. And from the study shown in China, it shows that many of these antibiotics actually cannot be removed in sewage treatment plants. So the efficiency of removal is less than 60%. In the situation in Hong Kong, these few classes of antibiotics are the highest, most poorly removed in these sewage treatment plants. So this poses a problem that these antibiotics might have in the marine environment, and if we eat those seafood, then we have those problems as well. So this causes a significant threat to public safety and health. Traditional or current technologies to detect these or monitoring these antibiotics, either chromatographic in nature or electrophoretic in nature, right, such as HPLC and CE, shown in here. So this method have severe limitations. So for example, tedious sample separation, involves specialized personnel to handle this expensive machine, and they cannot be used on site, and they have to be a high maintenance cost to operate them. There's also techniques that use immunoassays to do it. However, there's also limitations such as cross-reactivity between antibodies, and the production of antibodies usually require intensive labor, and they normally have a huge batch-to-batch variation. And because they're antibodies, so they always have to be in the refrigerator or freezer. So the transportation and storage will be a big issue. So we hope to develop an optical assays that can provide some technical simplicity, rapid response, low cost, and potentially high throughput and on-site analysis. So we have 4 aims of our objectives in this project. So first is to establish the capture select platform to generate new antibiotic binding aptamer. So that's the first aim. And then we hope to optimize and stabilize the aptamer that we develop that can help to bind to our antibiotic of interest strongly and specifically. Then we want to develop structure switching sensing platforms to detect antibiotics and compare it with those traditional or current technologies. And if everything goes well, then we'll go to detect the antibiotics in different sewage plant or even in the seawater. So let's talk a little bit on aptamer first. So aptamer is referred to chemical antibody. They are normally single-stranded DNA and RNA selected through a process called SILAX. It's a technology developed in 1990s by Jack Shostak, Larry Gold, and Gerald Joyce. Aptamer has comparable binding affinity and specificity with antibody. However, it has additional advantages such as low cost, high stability, scalable synthesis, and then flexibility to incorporate chemical modification. So apta switch, so meaning that when it binds to the aptamer antibiotics, then it will change in structure and create a response. And those response we can couple it with, for example, fluorescent detection or visible light detection. And it's possible to recognize antibiotics specifically by having different types of secondary structure. For example, shown in here, the stem loop structure has these GA-GT loops, and it's known that these GA loops in here can interact with one of the antibiotics, SDM, shown in here. And conventional SILAX process suffers from one of the main issues, which is the target itself is relatively small. So if we need to incorporate biotinylation into it, then it might affect the aptamer selection process. So in order to overcome it, we develop the capture select process that can allow the demobilization to be on the oligonucleotide rather than the target itself. So this way we can then select for antibiotics without modifying the antibiotics in the first place. And this is a scheme to show this approach, and I'm going to explain it slower in here because it's quite complicated. So first you have the DNA library shown in here. So N30 is the randomized sequence, N10 is the randomized sequence. And then we have a fixed region. Then the first thing we do is then we immobilize the oligos and hybridize it, attached with biotin. We have then the magnetic beads that can capture with strep afidin coated magnetic beads shown in here. And then we will add our antibiotic of interest shown in here. If there's no interaction between the antibiotics with the aptamer candidate, then it will have no reaction. However, if the antibiotics will bind to this N30, N10 region, so then they will be released from this biotin and break these hydrogen bonds, and they will be eluted from the magnetic beads, and these eluted aptamer sequence will then be able to go through the PCL amplification and then next to the next selection round. So normally you perform around 8 to more than 8 rounds, so then you can enrich the aptamer of interest. Then at the end, once we decide, oh, maybe the 8 round is enough, then we will add the next generation sequencing adapter to it, and then we submit that to a high throughput sequencing library. And then after we get the aptamer candidate, then we can test the aptamer to see if it really binds to the antibiotics and how strong they bind. We can modify the DNA or trim the DNA to be shorter so it will save costs. And then we can further do chemical modifications to make the DNA more stable and resistant to different kind of environment or pH conditions. And we have done this first few candidates. So for example, we have SDM, SMC, and tetracycline and other antibiotics, and we've done 12 rounds, and these are some of the potential candidates that we have generated. And now we are in the process of testing and verifying the binding. So after that, I think we can then use some of these aptamer sequence to create the apta switch that can, once bind to the aptamer, we can generate the response. And to generate or to create the apta switch, so this is some of the strategy that we are planning to employ, and we have done some experiment on it, and it seems to show it works quite well. So without going into all the details in the technicality, so basically we use the gold nanoparticle, and normally they will absorb with the aptamer sequence or single-strand DNA. When there's no antibiotics, so they will absorb, they will show the color. When the target, for example, SDM antibiotic shown in here, then they will bind to the aptamer, and then it will not absorb to the gold nanoparticles, then it will change in color. And then once we have this, and then we centrifuge, then remove the gold nanoparticle, and then we can take these things to perform cascade reactions to generate the fluorescent signal or visible light signal. And this, we have designed 2 approaches to do so. So we hope that this approach can be used for all different types of antibiotics, because gold nanoparticles should absorb to
all these single-strand DNA aptamer. Again, without going into detail of how this approach works, so this is approach 1. So basically, we have different regions, A, B, C, D, and what happens is that when there is the aptamer, when there's the antibiotic, it will bind to it, and then we will have different concentration of these antibiotics, and then that will allow us to trigger different amount of response after the cascade of reaction. So, so we will get a dose-dependent fluorescent signal depending on the concentration of the antibiotics that are available. Okay, so this is approach 1, and this is the preliminary result, and we show that we can theoretically generate a large fold enrichment in the fluorescent signal, and then now we are testing to see if this in a more complex solution with containing the antibiotics. So we also have the approach 2 is still caused or mediated by the destruction of the tetrahedron nanostructure, the TNS. So it's similar approach, but the fact that we will not require any enzyme step in this approach. So we have a few different types of oligos, and that will create this tetrahedron nanostructure when there's no antibiotics. When there's antibiotics, then it will cause a change in the tetrahedron nanostructure, and then again it will generate the fluorescence based on the amount of the antibiotics present, and then this will destroy the tetrahedron structure and generate this fluorescent signal. So again, by a dose-dependent manner, we will have different response for the fluorescent signal. So again, we have done some very preliminary data, and it shows that there will be full enrichment. And now again, we are testing by doing different amount of antibiotics. Do we still see enough response to get a detection signal? So just a quick summary for the 2 approaches. So approach 1 is isothermal, which is good. So we don't really need a PCR or thermocycler to do it. And as you can see from the preliminary data, it has much better sensitivity and full enrichment. However, the limitation for the approach 1 is that it's longer incubation time. You will need an enzyme to do this reaction, and these are other features of this approach. For approach 2, it doesn't require an enzyme, which means that it might be able to be cheaper, and also can be done without refrigeration of the enzyme. However, the sensitivity is lower in this approach. So we're still working on all the other aptamer as well on these 2 approaches. We will probably have more data to show you next time. Before we end, so we hope that later on, if the approach works, we can then test it really on the sewage treatment plant, rivers, and other sites. And with that, I would like to thank the groups, especially Andrea, who has been optimizing the AptaSwitch platform, and also Beatrice and Danyang for working with the Capture Select platform and the chemical modification on the aptamer. And also like to thank the Safety Lab for the support of the funding, and especially also Kenny and Phoebe for all the support and knowledge to me, to a newcomer to the field of me. So with that, I would like to thank and take any question.
Yeah, so I think due to the time, it's a little bit overrun, so we only allow 1 question. So any question from the audience? Yeah, if no question, then we thank you, Dr. Ko Chun-Ki, inspiring and innovative talk. Thank you, Dr. Koh.
Thank you.
So let's welcome our next speaker, Dr. Water. Dr. Water actually is my PhD student, and he is now a postdoc in Department of Applied Biology and Chemical Technology, the Hong Kong Polytechnic University, under the supervision of Dr. James Fair. His current research focus on coral in situ metabolism and physiological response to the changing climate by underwater respiratory and experimental manipulation. Today, Water will present non-invasive methods for assessment of coral health in situ. Let's welcome Dr. Water.
Thank you, Dr. Chan, for the introduction. Yes, I'm currently a postdoctoral researcher at the Hong Kong Poly U, and today I'm going to talk about non-invasive methods for assessment of coral health. And this is actually a project still going on, and it's actually taken a few years for the development, actually involved my PhD project. In this presentation particularly, we are going to see, I will go very briefly through introduction of coral metabolism, and then we will see what are the main techniques used and widely distributed to assess the coral health. And then we will see how we can apply these techniques through in situ observation of metabolic rate of the coral Platygira carnosa. And then we see some conclusion. So as already introduced before, corals has coral reef in general are a high biodiversity hotspot with an enormous economical global value. And this value is coming from mainly from fishing. If we think, for example, from South China Sea or Southeast Asia, fishing and seafood products are the main economy and the main food for population living along the coastline. But also, we know that people enjoy sport water activities and also scuba diving or anything related to the ocean, like outdoor activities related to the ocean. So there is a lot of economical income from tourism activities. But one of important services also for the coming from the corals is about the coral coastal protection. So basically, corals and the reef in general can protect the coastline from erosion, from wave energy and tide movement. So, but actually, in this presentation, I want to focus more on the metabolism of corals. So we need to understand what corals do to live and how they can acquire energy. So in a very simple schematic representation, we know that corals is a combination of symbiotic, actually symbiotic relationship between the animal, which is the polyp side, and endosymbiotic zooxanthellae, which are the algal symbionts. So the main energy is coming from the sunlight, and it can be used from the zooxanthellae to make the photosynthesis. And one of the results of the photosynthesis is the release of oxygen and production of sugars or carbohydrates. And this molecule can be used from the polyp side, from the animal, to make all the basic metabolic processes, for example, the respiration process, and can in turn release the CO2 that can be reused and recycled through the photosynthesis. And we have a production of energy which can support the uptake of dissolved inorganic carbon and calcium ion to make basically the calcification and to build the coral skeleton. So if you take these three main biological processes, take in mind this, but actually coral is something very much more complex organism. So this is what we call coral holobiont. It's a meta-organism, so it's not only a combination of polyp and endosymbionts, but also we have bacteria, we have viruses, we have other microorganisms living together in this microenvironment. And together they have basically they can recycle the energy, they can recycle the organic matter through into inorganic nutrients. And so all these organisms and microorganisms can support the biogeochemical cycles around and inside the corals. So we have been doing, so basically if corals can have the energy, can acquire energy from these two sides and address this energy for coral growth and standard metabolic processes. Actually, we need to consider that we have some global impacts affecting the corals, such as increasing the greenhouse gases, which are causing warming of the ocean, so rising ocean temperature, and also reducing the pH of the ocean through the ocean acidification. But also we have a combination of severe climate phenomena like El Nino or cyclones, or if you think, for example, typhoon, especially in this area, in the subtropical area. And these global impacts, we need to also correlate with the local impacts affecting the corals exactly in the place where we are going to study. So locally, we have a combination of coastal development and sewage treatment, which are releasing pollution or increasing the turbidity. And we saw from the previous presentation, for example, releasing of microbes in the water. And this can increase the eutrophication of the seawater. We can affect the dissolved oxygen in the seawater. So the combination of these can be detrimental for the coral survival. But we have been doing literature review of techniques to assess coral physiology in situ under global and local stressors. So we find out that from 90s to nowadays, most of the studies focused on primary productivity and physiology of corals under stress, while with minor studies, only few studies focusing on light limitation or photoinhibition inhibition of corals, specifically under, for example, under turbidity events or deeper corals. And how these techniques evolved through the years. So the main methodology were always been fluorometric techniques. We think about, for example, diving pump, with which we can measure the fluorescence of endosymbionts and the photosystem, specifically the photosynthetic efficiency of the photosystems, and also using submersible chamber and benthic chamber. So these are very common technique now that we can use to monitor the coral physiology, but also the sampling scale evolved. So if we know a lot about polyp endosymbionts interaction and what happens around the colonies, the scientific interest now is evolving more towards community studies. So we want to know more exactly on the coral reef in general, not only focusing on coral specifically, but also including the seagrasses or the role of fish, for example, in the energies of a reef ecosystem in total. So to talk about coral physiology, we have been developing specific protocols and techniques for this underwater respirometer. So this is particularly called KISME, is an acronym for Community In Situ Metabolism. It's an underwater respirometer. It is actually a small benthic chamber that you can see here in the video that we can attach on the coral, and we can measure dissolved oxygen and pH through dark and light incubation. So we can measure the respiration, the photosynthesis, and the calcification as like main biological processes, as I described before. And so we use this system combined with most common diving pump and photographic analysis to measure several parameters on the corals. And specifically, we want to know at the beginning what are the physiological limits of the corals. We used Platygira carnosa. Previously, Professor Chiu mentioned that Platygira carnosa is one of the flagship corals. He's believed to be stress-resistant coral, and this coral is distributed in Hong Kong water and widely distributed in Hong Kong water, so easy to find. So we try to do a single stress experiment at the beginning to see what happened to corals when we stress them to high temperature from, in this case, 25 to 32 degrees. And we observed an increase of respiration, reduced photosynthesis, and reduced calcification. So in total, we had a reduced of energetic availability indicated by this PR ratio. But when we stress coral under low salinity, actually, we only observed reduced calcification because obviously we have a reduced amount of inorganic carbon or salts in the water, but increase of whiteness. So corals start to become pale, but not exactly bleaching at the bleaching level. So if you see this level are pretty much extreme, okay, for the coral survival. But we only could find that some stress events, but not affecting the bleaching. This is in the short-term stress. So later on, we applied these techniques in situ. So we went directly underwater to see what these corals are doing. So we went to Chek Chau, and this was study lasted 2 years from 2018 to 2020. And we want to see if this coral Platygira carnosa, how is modulating, how is doing through the year, so with a seasonal change. So if we mentioned before that in Hong Kong and in general, in the subtropical region, we have 2 distinct seasons. For example, summer, which is the wet season, and winter, which is the dry season. Actually, I want to highlight that we can find transition between these main 2 seasons. So I split the data between spring, summer, autumn, and winter. So we have been going to Port Island every month for 2 years, and we collect both environmental data and coral physiology data. So starting from environmental conditions, you can see that temperatures go up during spring to summer, and then go down around autumn and winter with a very low temperature. Here in Chek Chau, we never reached 14 degrees as previously said, but we had 18 degrees to about 30 degrees. And also salinity, we can have a drop of salinity, localized drop of salinity due to rainfall. And also a very high fluctuation of pH, and also the oxygen in the water column. So if you see also the chlorophyll was very high sometimes, but this actually I want to highlight that this area it is enclosed bay with a very shallow water. So occasionally we can expect this extreme value, but it's very interesting that corals actually are still performing pretty well in the water and still looking good. So how they can actually survive in this environment and how they can modulate their physiology. So this is just few data that we have submit, actually still under reviewing coral reef. We can see that through the season, coral can change and they can modulate their energy, and they can allocate some energy for the standard metabolic processes and for example, the PR ratio, or sometimes they can allocate more energy for the calcification, so they can grow better in one specific region, season, and also change the color. The color also keep changing. The color is a mean of chlorophyll content and then symbionts content. So we can actually take this data and correlate with environmental information. And we can see that not only temperature and salinity affecting the corals, but also the pH. This is very simple. In red are negative correlation and in blue are positive correlation. So pH and temperature are negatively affecting the coral survival, the coral metabolism in general. But if we see the positive correlation, we have the turbidity and the salinity positively correlating with the energetic allocation. This means that turbid waters, which means probably more supply of food, and high salinity are actually good for the coral metabolism. And then we can also plot this data with here, for example, with the temperature salinity parameters. And again, we can see that the energy here is mostly allocated when or mostly produced when the temperature is low and the salinity is high. But calcification instead is following another trend. So calcification instead is like the around 25 degrees is probably the best temperature and condition for coral growth. So all these data are available online. So in the Pangea repository, we had also both the data from the underwater respirometer published in Pangea, and also this environmental data and coral metabolism data. So you can access and you can see this data online. And then more is yet to come, because as you can see from this picture is published on a Italian diving magazine, and we have been collecting also the coral mucus from these corals. And we conducted this study also in Hong Kong marine parks, but at this moment I cannot show you the results, but the work is in progress. And also we are working in collaboration with the CTU colleagues on proteomics, on tissue and endosymbionts. And here I wanna have a call for collaborators. If anyone has data from metabolic rates, water quality, I would like to develop a model for energetic performance. So if anyone willing to share, you can contact me, and we can have a study together. So some major conclusion very briefly, we see that actually these corals, these techniques can be used to measure the metabolic and biogeochemical properties of corals under different conditions. And then we saw that in situ survey, underwater survey, highlighted the salinity and pH affected the calcification and whiteness level. So this may be a reason of why Hong Kong corals in general are slow growth coral. And this finding can explain the high resilience pattern of coral. So maybe it can explain why these corals are still there and why they're looking still good. But actually what we are missing is the long-term monitoring and the real-time information. So of course we cannot be in the water for 365 days per year, but we need to improve the resolution of these data collected directly from the field. So I would like say thanks to all collaborators to these studies, from State Key Lab in CTU, the Chinese University and University of North Carolina, which were the inventor of KISME, and with special thanks to Professor Wells from University of Maine. And then thank you all for your attention, and yes, open to your question and your comments. Thank you.
Yeah, thank you, Walter. I think you have already two questions from the audience. So the first one is from Lily, I think. Do you have data supporting how various coral diseases affect coral metabolism already?
Unfortunately, not.
Cambenza, not Lily. Yeah, first question.
We don't have data on coral disease. We didn't observe any disease from underwater from these corals. So we don't have this data. We don't know much actually. But yes, will be interesting to see if there is any information on how the microbes in general, and if there is any pathogen affecting the corals. This is very common topic I think in a more tropical area and in Caribbean, but I would say so far not in Hong Kong, but yeah.
Yeah, I think that this is a chance for your collaboration with colleagues in Philippines or Indonesia. I think there are more coral diseases discovered, right? And then the second question is, do you think the correlation response of coral metabolism on the abiotic parameter study similar among coral species?
This is a very good question. Actually, I think that the response is not the same with all the species, because different species have different metabolism. Some species, for example, rely more on the endosymbionts. More species rely more on the food, on getting the energy from the food particles outside. So they can have different amount of, for example, fatty acid or lipids content. So they can respond differently to different stress. Usually we say this, we define stress-resistant corals and stress-sensitive species. And this in case Platygira is a stress-resistant species. So there is a difference, depending on the species, they can respond differently.
Yeah, thank you, Walter. And
thank you.
I think the time, I think, limitation, so we don't have more questions.
Sure, thank you.
Thanks for your presentation. Yeah. So our next speaker is Dr. Rick Lai. He is a postdoc in State Key Laboratory of Marine Pollution, City University of Hong Kong. His main research area include marine toxicity, nanotoxicity, environmental risk assessment of different emergent contaminants of concern. Today, Dr. Lai will present the influence of environmental temperature and salinity on the toxicity of zinc oxide nanoparticles. Let's welcome Dr. Lai.
Yes, thank you for the introduction of Dr. Leo Chang. Since my network is not very stable, I will have Grace to help me to share the pre-recorded presentation for you. Thank you. Can Grace help me to press the next slide?
Yes, we are moving on to your presentation. Thanks.
Due to the small size of engineered nanomaterials, they have larger surface to volume ratios compared to their bulk counterparts, and therefore they generally will have advanced properties than these bulk particles, and are becoming more and more popular in commercial products. According to a survey in 2014, there were more than 1,800 products from 32 countries that use engineered nanomaterials. Consequently, it is expected that global market value of nanomaterials could reach 100 billions by 2025. However, such an expansion of engineered nanomaterials will inevitably lead to the potential concerns about their pollution in the environment. In the subsequent studies, we will use zinc oxide nanoparticles as our target nanoparticles because in our review we found that it has relatively higher production and it's popular in the commercial products while it has relatively higher toxicity than other nanoparticles, and there is relatively fewer research efforts on it. The COP26 on climate change has just recently ended. The latest IPCC synthesis report suggested that the current global ocean surface temperature has increased by 0.88 degrees Celsius in the last decade, and they expect the global surface temperature is going to increase by 1.5 to 4.5 degrees of Celsius by 2100, suggesting that global climate change and warming is a critical problem. Likewise, in Hong Kong, the number of extreme hot days is increasing over the years. The precipitation is also recording record high. Such extreme and frequent weather events will inevitably lead to variation in temperature of salinity in the ocean environment of Hong Kong. Temperature has been the most commonly studied environmental factors about their impact towards the chemical toxicity. According to a previous literature review, the authors suggested that the toxicity of over 80% of the chemicals could follow two different models. In Model 1, they suggested that the chemical toxicity generally increases with increasing temperature. In Model 2, they suggest that the chemical toxicity should be the highest at the two temperature extremes. The authors speculated that such variation in toxicity would likely be associated with the physiological responses of the test species. For example, in Model 1, at low temperatures, the test species usually will have lower intake rate and metabolic rate, and hence they will take in less chemicals and are not affected. Alternatively, in Model 2, since the species are at the temperature extremes, they will spend a lot of energy to cope with this heat stress, and therefore they will have less energy to cope with the chemicals and then become more vulnerable to the chemicals. Indeed, according to a previous study that used single-cell nanoparticles, the authors suggested that the toxicity of single-cell nanoparticles also increased with increasing temperature towards two of the three studied test species. On the other hand, salinity has been a less commonly studied environmental factor. However, based on existing literature review, it seems that salinity would have a similar effect as temperature towards the chemical toxicity. In a recent research, the authors tried to model the toxicity of 5 metals under different temperatures and salinities. They found that the metal toxicity would be the highest at the salinities at two extremes. They suspected that this is because at these extreme salinities, the test species would likely experience osmotic stresses and hence become more vulnerable to the metal. Alternatively, for studies of single-cell nanoparticles, the authors found that the toxicity of single-cell nanoparticles generally increase with decreasing salinities towards all the test species. Apart from the potential osmotic stresses that may be faced by the test species at low salinities, the authors suggested that at low salinity, single-cell nanoparticle would have smaller agglomeration and hence increased release of zinc ions, which would consequently increase their toxicity. Based on the above literature review, we could notice that both temperature and salinity could affect the physical-chemical properties of the chemicals, as well as the physiological responses of the organisms, and therefore affect the toxicity of the chemicals. However, there is only few studies that can investigate the combined impact of temperature and salinity. In one previous study, the authors found that temperature and salinity could join and interact to affect the toxicity of single-cell nanoparticles. They suggested that the toxicity of single-cell nanoparticles was the highest at extreme temperatures but decreased with increasing salinity. However, only one species, a marine diatom, was studied in the previous research. Therefore, it is necessary to evaluate and investigate if other test species also have similar responses to single-cell nanoparticles under the joint impact of temperature and salinity. There would be 3 different research questions in this study. Firstly, we want to know whether temperature and salinity would affect the physical-chemical properties of single-cell nanoparticles. Second, what would be the toxicity of single-cell nanoparticles under different temperatures and salinities? Lastly, would different species respond differently under these factors? The major hypothesis is that both temperature and salinity could modify the physical-chemical properties of single-cell nanoparticles and hence affect their toxicity to the test species. Test temperatures and salinities were selected based on the situation in Hong Kong waters. As shown in this graph, the temperature in Hong Kong marine water generally range from 15 to 30 degrees of Celsius. And another 35 degrees of Celsius was added based on the prediction of the IPCC report as a worst-case scenario. The 3 selected test salinities also cover the common salinity range in Hong Kong marine water. In order to evaluate the impact of temperature and salinity on single-cell nanoparticles, we put single-cell nanoparticles and their bulk particles and ions into incubator and incubate them at different temperatures and salinities for 7 days. Afterwards, we measured the physical-chemical properties, including ROS production, agglomeration size, ion release, and surface zeta potential, which are closely related to their major modes of action according to the previous literatures. The physical-chemical characterization has generally proved our hypothesis that temperature and salinity could modify the properties of single-cell nanoparticles. We found that for agglomeration and ROS production, they generally will increase with increasing temperature and salinity. Meanwhile, the zinc ions released from single-cell nanoparticles will reduce with increasing temperature and salinity. These findings lead to another question, that is, how these modified physical-chemical properties of single-cell nanoparticles could affect their toxicity. To answer this question, we exposed the incubated chemicals to different model organisms. In the first experiment, we exposed them to Tigropus japonicus, which is a marine copepod. It is a dominant taxa in the aquatic zooplankton communities, so it has an important ecological function, and is a benthic species at intertidal areas where they will experience abrupt changes of temperature and salinity. These animals will be previously acclimated at the specific temperature and salinities before the experiment, and the test applied is the standardized 96-hour acute toxicity test with chemicals renewed in the middle of the exposure, that is 48 hours. This is the toxicity results of single-cell nanoparticles along different temperatures and salinities. In our findings, we found that the toxicity of single-cell nanoparticles increase with increasing temperature but decrease with increasing salinity. Such results agree with the previous studies about single-cell nanoparticles. However, this did not agree with any of the monitored physical-chemical properties in these studies. For example, for release of zinc ions, which has always been attributed as the major toxic mechanisms of single-cell nanoparticles. Its trend actually decreased with increasing temperature and decreased with increasing salinity, which is different from the toxicity of single-cell nanoparticles. This suggested that the observed toxicity in this study could not be explained by any of the single physical-chemical properties of single-cell nanoparticles. Apart from the potential impact for the joint influence of several physicochemical properties of single-cell nanoparticles at the same time, the physiological responses of the copepod may also play a role. For example, at low temperature, we observed reduced mobility of the copepods during the experiment. This may suggest that they may have entered a dormant stage which has been defined and observed by the previous studies. At this dormant stage, they may have reduced metabolic rate and intake rate, and hence they are less sensitive to the chemicals. Another question is whether such response to single-cell nanoparticles would be consistent among different species. Therefore, in the next experiment, we again expose the incubated chemicals to another model species, Xenostropus securus. It is a marine mussel, and it is living in a relatively polluted area in Hong Kong and has relatively high tolerance in temperature and salinity. Most importantly, they are a unique target group of nanoparticles. As shown in this figure, they are able to filter out single-cell nanoparticles within 1 hour. The experimental setup is very similar. The animals are first acclimated to the specific test temperatures and salinities within 1 week. After that, they are exposed to the chemicals for another week, after which we will check different physiological parameters of the marine mussel, including survival, clearance rate, lipid and protein stocks, etc. The experimental results suggested that temperature, salinity, and chemical concentration significantly interacted with each other and induced toxicity towards the marine mussels. To better illustrate the results, we present our results in the following PCA diagrams. First of all, we found that at environmentally relevant concentration of single-cell nanoparticles, they were not inducing toxicity to the marine mussels. However, at a higher test concentration, we found that single-cell nanoparticles were inducing adverse effects compared to the control. Therefore, in the following results, we will focus on the difference between the control group and the treatment group at higher test concentration. For temperature, we noticed from the control group that high temperature alone could already induce adverse impacts to the marine mussels. In the treatment group, there was a similar pattern, but this time the high temperature seemed to be more separated from the lower temperatures. This may suggest that single-cell nanoparticles may have deteriorated the performance of the marine mussels under high temperature. This is consistent to what we observed in the test using the marine copepod Tigriopus japonicus. Likewise, for salinity, we noticed that low salinity alone could induce adverse impacts to the marine mussels in the control group. However, interestingly, in the treatment group, we found that it was the high salinity that induced adverse impacts to the marine mussels. This is not only different from the control group, but is also different from the observation in the experiment using the marine copepod T. japonicus. This suggests that different marine species may indeed respond differently towards the joint interaction between temperature, salinity, and single-cell nanoparticles. In the next step, we try to correlate the physicochemical properties of single-cell nanoparticles with the responses of the marine mussels under different temperatures and salinities. We noticed that at high temperature and salinity, the adverse response of the marine mussels may correlate with the agglomeration of single-cell nanoparticles. Therefore, we suspected that the adverse impacts may likely be due to the significant agglomeration of single-cell nanoparticles and the increased filtration rate of the marine mussels under high temperature and salinity, which eventually result in increased uptake of the single-cell nanoparticles. Therefore, we try to evaluate the bioaccumulation of zinc in the marine mussels after the exposure, and we found that the zinc level of the marine mussels indeed increased with temperature and salinity, which may suggest they have intaked more single-cell nanoparticles under these conditions. This study managed to answer 3 different questions. We found that temperature and salinity could modify the physicochemical properties of single-cell nanoparticles, including agglomeration, ROS production, and ion dissolution. And we found that single-cell nanoparticles could have different toxicities under different temperatures and salinity and towards different species. Such difference would likely be a consequence of the modified physicochemical properties of engineered nanomaterials and the physiological responses of the test species under different temperatures and salinities. Therefore, the take-home messages are: during the assessment of the risk of the engineered nanomaterials, different environmental factors should be considered. And the physicochemical properties of the engineered nanomaterials should also be evaluated so that we can correlate their toxicity with these properties. We are particularly interested in modeling the potential toxicity of engineered nanomaterials under different environmental factors and different species, so that we could accommodate the rapid development of the future nanomaterial market. This is the end of my presentation. I would like to give special thanks to my supervisor, Professor Kenneth Leung, as well as Professor Lee and Professor Jurisic. I also need to thank the General Research Fund given by the Hong Kong government to support this study. And also great thanks to the audience, and questions are welcome.
Yeah, thank you, Dr. Lai. So I think there's one question from Dr. James Fair. James Fair, would you like to ask in person, or you want me to ask on your behalf?
Do you hear me?
Yeah.
Yes, thank you, Dr. Lai, for the presentation. I would like to ask what was the exposure concentration of your nanoparticle and how is it compared to the environmental level, please?
Yes, we have 2 experiments. One is on the marine copepod, one is on the marine mussel. For the marine mussel one, we have tried the environmental concentration, environmentally relevant concentration of single-cell nanoparticle, which is around 10 microgram per liter in the water. But for the marine copepod, because it is an accurate test, we use a relatively high concentration, which can up to 15 milligram per liter. But in the copepod experiment, we try to model the toxicity response of the copepods under different temperatures and salinity, and we found that at the most toxic situation, that is the high temperature and low salinity, the chemical toxicity could actually be lower than the the existing water quality guideline of zinc in the environment. That means that this experiment also showed that there is a potential risk of single-cell nanoparticles under the current regulation of the water quality guideline.
Okay, thank you, Dr. Lai. I do have another question, if I'm allowed it. We know nanoparticles tend to aggregate at higher temperature and higher salinity, which is higher ionic strength. One of your conclusions is that this aggregation can induce the toxicity to marine mussel. Would you please further comment on it, how aggregation can induce toxicity?
Yes, this is another area that we would like to further study, but we don't have any concrete support for this. But we just think the nanoparticles, because every different mussels will have their own filtration size range, they may filter certain size of these particles. And so we think that the agglomeration of these particles may fit in the filtering size of this marine mussel, and that's why increase their accumulation in their body. And this at this stage, we only check the biofiltration, the bioaccumulation rate of zinc in the marine mussel, and we did find such kind of response under the high temperature and high salinity.
Okay, I see. Thank you.
Yeah. Any other question from the audience? Yeah, Dr. Lai, I have one question. So you expose those nanoparticles to the mussel, right?
Yes.
And then do you think they will accumulate in their body, or deparation will get rid of this nanoparticle if we put them in a clean water for a certain duration of days?
Yes, thank you for your question. It's also a very interesting and important aspect to be studied. What I can provide you to now is that we have checked the bioaccumulation of zinc level in the muscles after the exposure, and we did find they have increased accumulation of zinc along different conditions of temperature and salinities. That means they should be able to accumulate zinc oxide nanoparticles in their body. For deprivation, I'm not sure, but we have in our preliminary test, we have tried to expose a high concentration of zinc oxide nanoparticles to the marine mussels, and we noticed that during their filtration, they are actually be able to produce some of the feces. That means that they are releasing some of these nanoparticles out of their body. So I think deprivation may also be possible for the marine mussels.
Yeah, it depends on how much they can release, right?
Yes, yes.
Yeah, if they release only a little bit, but accumulate most of them, then maybe those muscles cannot be sold or they are no longer edible, right?
Yes, it can be because we did find a high accumulation of zinc level in their body, but we haven't checked whether if we put them back in the clean water, will they be able to reduce? How long will it take for them to reduce to a normal level?
Yeah. Okay. Yeah. So, Dr. James Fair has some comment, right? Dr. James Fair.
It's okay. It's a communication with Dr. Lai because we are working on a similar experiment.
Okay. Yeah. Yeah. So, any question from the audience? If no more questions, then now come to the end of the morning session. I would like to thank all the participants for your attention, and also Dr. Henry He, my co-chair, and then the 6 excellent speaker for the very interesting and inspiration talk. So maybe Grace or Phoebe, do you have any announcement for the symposium in the afternoon?
Yes, just one. Please come back at 2:00 p.m. In Hong Kong time or 1:00 p.m. Cambodia time.
Okay, thank you, Phoebe. And also we have a panel discussion chaired by Professor Wu. So please also stay until the panel discussion session. Okay, so goodbye. Thank you. Have a good lunch. Bye-bye.
Now it's 2:00. Good afternoon, audience. Welcome back to the symposium. This is James Fang, the chair of the afternoon first section. The first invited speaker in this section is Professor Rudolph Wu, who is a chair professor currently in the Department of Science and Environmental Studies at the Education University of Hong Kong. Professor Wu is our world-leading expert in marine ecotoxicology, in particular in the research area of aquatic hypoxia. Many of us, including me, have been students of Professor Wu. Today, we are fortunate to have Professor Wu here to share his insight into hypoxia in aquatic environment. Professor Wu, please.
Thank you. Thank you for the introduction, James. Now, in this talk, I try to discuss the topic about hypoxia. And then if you look at the topic, you'll be maybe surprised that nothing could be worse. I hope within this 30 minutes, I'll be able to convince you that hypoxia is the most important environmental stressor in our environment today. Now, first of all, what is hypoxia? Hypoxia is defined as the dissolved oxygen of the water, which is less than 2.8 milligram oxygen per liter, which is equivalent to 2 milligrams oxygen per liter. Now, we shouldn't blame everything to pollution. Hypoxia may be a natural phenomenon or due to pollution. For example, stratification due to this density difference, like with a thermocline or a halocline. That means a different temperature or different salinity of the water can block the oxygen diffusion, and that causes the hypoxia. And probably more importantly, in recent years, human activities add a lot of nutrients to the water. We all know that the limiting factor of this phytoplankton growth is nutrient. So we put excessive nutrient into the water that causes a phenomena which we all know about. This is eutrophication. And also because we discharge a lot of organic matter in the water, and then the bacteria, when they degrade these organic matters, require oxygen. That would consume a lot of oxygen in the water, making the dissolved oxygen low and become hypoxia. Now, in actual fact, in the last 30 to 80 years, decrease in hypoxia occurred all over the world, in USA, China, Scandinavia, United Kingdom, Sweden, Germany, Denmark, Black Sea, and Adriatic Seas, all over the world, in actual fact. Now, nowadays, globally, there are more than 400 is what they call dead zones, which is associated with eutrophication. That is to say, is these 400 dead zones are due to human activities and causing this eutrophication. And the total area add it up together is more than 245,000 kilometers square. For those who are not familiar with what the total area means, the total area is much larger than the United Kingdom add up together. And then one of the dead zones in the Gulf of Mexico, for example, which occur every summer. And then the size of this dead zone is similar to the whole area of the whole state of Virginia. So this gives you some indication of how bad and how large it is. Now, what's more is that if you look at the number of dead zones, and you see a dramatic increase from something like 1960s to 2000. And then in actual fact, these data is a bit outdated, and then that number of dead zone and the total area of dead zone keep on increasing ever since. Now, because of hypoxia, a lot of major change in structure and function of marine ecosystem over a very large area. What I like to emphasize is over a very large area has occurred all over the world. Something like in China, in Europe, in Japan, USA, and similarly some in Australia. Now, there's well-documented data showing that hypoxia actually causes a lot of animals to die, what they call mass mortality. So it's not uncommon to see lots of fish are killed by this low oxygen. Very simple, not enough oxygen, and they all die. And this shows you some picture that's very common. And let alone that they also have for this natural population of fish. For those who culture fish, again, they suffer a lot because hypoxia can kill off all their sort of culture fish within several hours. And then, of course, this hypoxia also causes death in the natural animals, like coral, and then like a lot of these animals sitting on the ground of the seashore. Now, in terms of function and structure of the ecosystem, and hypoxia has caused major change in species composition. They actually change the species composition, and later on I'll talk about this and in greater detail. And then also they then decrease in the biodiversity and species richness, and change in the trophic structure and the food web. So that's a very fundamental changes in, as I said before in yesterday's lecture, the ecosystem function is utmost important, and actually is much more important than the structure itself. So it changes the trophic structure, change the species, change the whole function, and change the life cycle and species distribution. And what's even more long-term is that they actually alter the bioirrigation. And some of you may not know that a lot of the ants animal, like the polychaete worm and then bivalve and things like that, and they're living on the sea bottom, and then they keep sort of stirring the sediments, stirring up the mud, and then but these sort of activity, what they call the bioirrigation, can turn some of these microbial animals and microbial organisms, and then into active form, and then they are sort of responsible for a lot of this essential biogeochemical process and sediment water exchange. So if you kill off this animal or change the animal composition, and then you expect these sort of major biogeochemical cycle will be disturbed. Again, I'll show you some scientific data that we collected later on. Now, long time ago, we have been interested in this and the hypoxia phenomena because we regard this as one of the most important, if not the most important environmental problem globally. So compiling literature and some of the data, we find that during normoxia, for example, and then you got lots of animals which are suspended feeders. But when changed to hypoxia, basically a lot of these animals change into deposit feeder because suspended feeder need more oxygen. Deposit feeder because they do more deposit and they survive better. So again, there's a major functional change in the functional group of these animals. And then they change from demersal fish under normoxic condition. Demersal fish means fish living on the bottom. And then change to pelagic fish when the water turned into hypoxic condition. Again, that's a fundamental change, mainly because this demersal fish which is living on the bottom, well, because the bottom oxygen depletion would be more serious than that in the water body. So they change from demersal fish to pelagic fish, and from high diversity to low diversity, and then from large body size to smaller body size, mainly because smaller body size can be more able or more efficient to take up oxygen from the water. So again, what these hypothetical changes and is summarizing, there's again a major change in species group and also affecting the function of ecosystems. Now, more recently, funded by the State Key Lab of Marine Pollution, we conduct some study, and for the first time, and we demonstrate that actually hypoxia can actually decrease the sediment carbon fixation. As shown in this figure, what we do is that we transplant the sediment from the hypoxic site to the normoxic site, and from normoxic site to the hypoxic site, and so on. And you see that after several weeks here, and then we transplant the sediment and has decreased in the carbon fixation. In contrast, the hypoxia actually increased the sediment phosphate fixation after 10 weeks. So you see that this, again, is very fundamental. We all know that the carbon, nitrogen, and phosphorus cycle are most important biogeochemical cycle in the marine environment, not only in the sediment, because in the sediment fixation will affect the water nutrient concentration as well. Now, these shows for the first time that hypoxia can actually affect the geochemical cycle by experimental evidence. Again, some 15 to 20 years earlier, when we start interest in the problem with hypoxia, we actually discovered for the first time that hypoxia is an endocrine disruptor. And the potency of endocrine disruption is comparable to the most toxic EDC in the world. Now, what we did is we put the fish under hypoxia condition for several weeks, and then we find that testosterone, which is male hormone, decreased dramatically, while the female hormone, estradiol, increased dramatically. And then, so we said, oh, is it only the carp? So we tried this out in different species, try and silverfish, the same happened. And then also we find out that this hypoxia actually affects this spermatogenesis. That is to say, the development of the male gonad. And you can see that under hypoxia, both the silverfish and common carp, and they all share the commonality is that they actually delay in their sort of the spermatogenesis. You got less spermatic, which is the mature sperm. And then you got more sort of primordial sort of germ cells like the spermatogonia and spermatogonia. And then for the diameter of these lobule, and then under hypoxia, the common carp actually sort of decrease in the diameter of this male lobule, showing that the male reduction and spermatogenesis process has been affected. Then we look at the sperm motility. Sperm motility is a very good indicator on the sperm quality. And then, so you show that the swimming activity, different velocity of the sperm of the fish also decrease. And then we use other models like the zebrafish and also find that the ovarian cell stage, similar to the spermatogenesis, has been sort of inhibited. So you got less sort of the mature oogonium, the oocytes, as compared with the sort of the premature one under hypoxia, showing that hypoxia actually inhibits the development of this ovarian. Now, at that juncture, a guy came from the USA, said, oh, come on, and this is all laboratory experiment. How do you ensure that it also happens in nature? So he said, oh, I don't know, because as you may probably aware, doing this field validation is very expensive and very tedious, but we can't afford to do that. And then Peter Thomas in Texas actually did similar experiment in the field of Gulf of Mexico, collecting fish, Atlantic croaker, from different places with different hypoxic level. And then he shows exactly the same thing in Atlantic croaker, and showing that in Atlantic croaker is the same. And then the sort of development of oogenesis are inhibited under hypoxia, under field condition. Then, if you put things together, we'll find that under hypoxia, the fertilization rate decreases, mainly because you got decrease in spermatogenesis and you got decreases in oogenesis. So egg is not very good, the sperm is not very good. This is why you decrease in your percentage fertilization from almost 100% to only 55%. Then with this sort of substandard fertilized egg, the hatching rate also dramatically reduced from something like 100% to less than 20%. So even the larvae has been able to hatch, the 14-day larval survival again decreased dramatically by 1/2. Now, if you take everything together, something like compare the sort of normal egg developed to larvae under normoxic condition with the egg produced by the fish affected by hypoxia, from egg to larvae, it decreased from something more than 90% to 5%. So that is this shows that hypoxia has a very dramatic and serious effect on reproduction. Something like only 5% of the original sort of egg to larvae ratio can be obtained. Now, the second thing that we demonstrate is that hypoxia is teratogen. Teratogen is a chemical that can make something lead to malformation, abnormal development. So initially, we find that they actually delay the development and then delay the larval development. So that's the larvae after 24 hours under control condition. And then after 48 hours, they got tail and things like that. But if you put the zebrafish embryo under hypoxia, well, for the same period of time, 24 hours, you can see that the development has been delayed. There's no tail yet. And then after 48 hours, this guy can almost swim and is still like embryo. Now, then we look at the malformation, primary skeletal deformity, and abnormal development. And then you can see that it manifests after 96 hours. That's the control. That's the treatment. That is to say, under normoxia, normal condition, the malformation is less than 10%. Whereas I mean, Now, after 98 hours, the malformation goes up to 15. After 160 hours, and then the normal malformation rate is only 10%, while this increased to almost 20%. Again, we use the fluorescent dye to look at the apoptosis. Apoptosis means means when the cell is trying to, to kill off itself, because the cell during development, they can ask that normal cell to sort of go away. Now, that's what we call the apoptosis. Now, hypoxia actually enhance apoptosis in the head, but not the tail, as shown by these fluorescent dyes dyeing. And apoptosis are identified by this staining, and from the head, number of apoptotic cells increase, which means the apoptotic cells in the tail decrease. We all know that apoptosis is an important process in determining the normal development of any animals. So this shows that they actually disturb this process, and then therefore leading to this and abnormal development. Now, then again, we talked to a number of scientists over the world. So these guys raised a lot of questions. They said, oh, come on, of course, under hypoxia, a lot of these animals, regardless, will reduce their biological activity, like under not condition, which is suboptimal, they decrease in their activity. So this is why they decrease in the hormone secretion, decrease in the growth, and then sometimes decrease in the development. So maybe it's just as simple as that. Now, and the other guy said, well, the reason why you find a smaller gonglet in the hypoxic fish, and also find the reduced hormone production, lower hormone production or disturbed hormone production, is due to reduce energy intake and reduce growth, because under hypoxic condition, the fish will feed less. So in that case, they got less energy, and this is why they got a smaller gonglet, and nothing to do with disturbance. Okay, and then the other so endocrinologist said hypoxia affects sterogenesis. Now you provide us the end result showing that spermatogenesis is disturbed. You also provide data on hormone is disturbed, but how do you know that sterogenesis is disturbed? And then the other guy asked, does hypoxia also affect the GnRH and gonadotropin, not only the sex hormone? Now, it would be very difficult for us to sort of test this condition, mainly because if you expose the fish to hypoxia, well, everything will be affected. So we use another approach. We use an in vitro study using a human adrenocortical carcinoma cell line, and then expose this cell line to hypoxia. We actually show that 3 genes regulating sterogenesis were affected by hypoxia, but more importantly, not the others. Now, this is the sort of the sterogenesis pathway from cholesterol all the way to testosterone to estradiol, the male hormone and female hormone. And these are the enzymes that regulate all these processes. We actually find that they affect specific process rather than specific enzyme like this. They affect the CYP11A, 3-beta-HSD, and then also CYP11B1, and then CYP19. In particular, we demonstrate that they affect CYP19, so this is why testosterone cannot be transformed in the estradiol, and this is why you got a higher level of testosterone than estradiol. So these demonstrate specifically that under in vitro condition, which is only the gonglet is affected by these hypoxia, and then they affect specific process, but not the others like CYP21 and CYP20B1 and so on and so forth. Now, then we look at the problem further. We find that hypoxia actually affects sex differentiation and alters sex determination. So again, we use zebrafish as a model. So after growing the zebrafish for 2 generations under hypoxic and normoxic condition, under normoxic condition, well, you got about 62% male, and then, but under hypoxia, you got almost 75% male. Then again, we look at the hormone again, look at the hormone level in the female fish and the male fish. We actually compare the testosterone to estradiol level. Now you find that basically the testosterone to estradiol, that means to say the male to female hormone ratio remain unchanged in the male, but changed in, or increased in the female. That is to say, they actually affect the female by increasing the testosterone level as compared with the estradiol level. So the female fish got more male hormone than female hormone, and this is why they direct their sex differentiation into male. Now, we all know that in this world, women and female are much more important than male. And then because they are actually the key factor controlling reproduction and hence the future population. So no girlfriend, big problem. Now, then you can see that if you in the population, you got more and more male and less and less female, eventually you can predict that the population will extinct, right? Because you got not enough female. No matter how many female you got, does not matter. Now, then we ask the question further. Is the observed male bias phenotypic or genotypic? Because we find that actually they actually affect the hormone only. So is it phenotypic or genotypic? Then I talked to a medical doctor when we are sort of showcasing our research in the UGC's format. And then they said, oh, we said, oh, maybe it also occurs in human beings. Nonsense. Human being, in the sex, human being is determined by the genotype, which has got XX chromosome, XY chromosome. So it's not controlled by the hormone, okay? Or even controlled by hormone is to much less extent. So I asked the question, will the same happen to other species with sex determining chromosome? So the medical doctor said, oh, no, come on, unlikely. And then, so I tried to get one of my previous students to work on the problem. So we can't work on human being, but so we can only select a fish with a sex determination chromosome, just like human being, okay, which is the, which we treat that, which is, we treat this fish and the, with under hypoxia and normoxic condition. And then we look at the sex reversal. And we find that even for the genotype XX, the sex reversal is 77%. And for the genotypic XX is 54%. Whereas the genotypic XY, they remain unchanged. So this shows very clearly saying that even you have a sex chromosome of XX, the sex reversal can still occur. Now, then we go back to the medical doctor, and then I also did some more study because, as I said, we couldn't work on human being, no capacity of working human being. So we searched the literature. We find out that patients suffering from sleeping apnea. Sleeping apnea means you got a blocking nose, and this is why you snore when you're sleeping. And then, so this guy is always hypoxia because they don't have enough oxygen. They have lower sex drive and lower male hormone. And then we find that due to this according to this epidemiological evidence, sex ratio in human depends on this sex hormone level of father and mother during the conception. And then the higher testosterone level during conception would favor the subsequent birth of sons. That is to say, if you have apnea, that affect your sex hormone level. The second point is that if your sex hormone level is lower, and then they favor the birth of son, which is actually totally in line with the experimental evidence that we got from fish. Then we go further to ask the question, saying, will this pass to the next generation? Just like this cartoon. Say if the grandma and grandpa were subject to hypoxia, and then how about the son and the grandson? Just like this grandson said, grandma and grandpa, I'm innocent. I've never exposed to hypoxia. Don't pass your problem to me. Will that happen? So we asked another PhD student to do the work. Oops. Now, what we do is that we put the fish under normoxic condition, normal fish, grow them for 4 generations, F0 to F3. And then the other group we put under hypoxia. And then after 1 generation, we split this group. And then one group were also subject to hypoxia for the subsequent 3 generations. Whereas when this is important, we take out some individuals which are subject to hypoxia for the F0 generation, put them back in normal condition, normoxic condition for another 3 generations. Then we try to compare their physiology, their sex, hormone, and so on and so forth. And we find out, in actual fact, that the hypoxia actually reduced the sperm motility in F0 and F2 for the transgeneration group. That is to say, the sperm motility of this, their son and the grandson decreased, despite the son and grandson have never been exposed to hypoxia, only because of the grandpa has been exposed to hypoxia. So this is what we call the transgeneration effect. Then we look at the percentage of mature sperm, spermatics, percentage of spermatic, and the same occur in the F2 generation, the transgenerational group. And then we look at the fertilization success. Again, the F2 are affected. I want to emphasize these guys and even the father have never been exposed to hypoxia before, and only the grandpa and grandma has been exposed to hypoxia. Then we then look at the epigenetic and transcriptomic and proteomic changes. We provide this evidence showing that in actual fact, they actually affect the fish, the F0 generation, affect the epigenome and change the epigenome, and then leading to this reproductive impairment in the F1, F2, and F3 generations. Now, then we ask the question, is that will the situation get better or worse? In fact, hypoxia is pretty simple. Everybody knows it, and it's because we produce a lot of these nutrients and put them in water. We produce lots of waste in wastewater, put them in water in the marine environment. It's extremely simple. We know about this problem about 30 or 50 years ago. But the problem in reality, if you want to remove nutrient, it's very expensive. To give you an example, in the Baltic Sea, in the Scandinavian country, which is very rich, they reduce 50% in the N and P going to the Baltic Sea, which is hypoxic, and cost them something like almost $4,000 million every year. Whether it is affordable to some of the developed country, but definitely not affordable by the developing country. And then if you look at the growth globally, the growth of these treatment facilities are likely to catch up with the growth of population and industry, particularly in the developing country. And the other problem is that the contribution from atmospheric fallout non-point source are significant. Just like in the US, everybody knows about where the problem is, but because it's come from agricultural runoff, and then it's very difficult for the government to control agricultural runoff, because how do you know the nutrient come from me? And let alone at least half of the nitrogen will go up to the atmosphere and come down. So they are not directly discharged in the river. So again, this causes major problem in controlling. And let alone transboundary issue are difficult to resolve. If Germany is producing this nutrient and then affecting France, we expect sort of Germany will sort of put in a lot of money to rectify the problem. the problem is not only with Germany, it's with the other country. Now, that's not the end of the story. Hypoxia, I'm sure you agree with me, is an evil, is it? And then when the meeting of the two evils, when hypoxia meet with climate change, the situation would be even worse because climate change increase in temperature would increase in freshwater runoff, increase in freshwater runoff, increase in water stratification, then enhance hypoxia. Freshwater runoff would increase nutrient flux, increase the productivity, and therefore also enhance hypoxia. The increase in the temperature would lower the oxygen solubility in water and also increase the metabolism, again, add on the demand of oxygen and making the problem of hypoxia even worse. So overall, the risk assessment of hypoxia present to you is the environmental consequence is very serious. Area affected will be very large, economic loss very high, number of people affected very large, probability of occurrence very high, lasting effect, long lasting, takes months to years to recover, and the trend is getting worse. So we try to argue at the United Nations Forum and several years ago, and then we argue and saying that hypoxia is the most important problem, environmental problem faced, and so affecting our marine environment worldwide. And eventually we won. And then our work has been featured in the Science for Environment Policy by the EU, and also reported by all sorts of public media in US, Australia, and Nature, and worldwide. Then, to conclude, hypoxia is an old problem with new scientific insight and calling for an urgent reassessment. So for the first time, we discovered that hypoxia is an endocrine disruptor that can affect selected gene, specific gene, not every gene, receptor and neurotransmitter along all the HPG axis, and therefore causing reproductive impairment. Hypoxia is a teratogen leading to delay in development and malformation through disrupting apoptosis, affecting sex differentiation and sex determination, leading to a male biased population and poses significant threat to the sustainability of natural population. Affects the epigenome, causing epigenetic changes of transgenerational reproductive impairment in subsequent generations. So given the fact that hypoxia occurs over a large area worldwide, and with this endocrine disrupting effect, teratogenic effect, transgenerational disrupting effect, hypoxia is the most important stressor in our marine environment, and it's difficult to control, likely to get worse, present a major threat to us worldwide in coming decades. And this is a research team from my previous research student and working together with my research assistant, Professor Paul Lai, and then my colleague, Richard Gong. I present this talk in various occasions, and I said, if anybody can challenge me saying that hypoxia is not the most important stressor in the marine environment, I'll give them $500. I'm still keeping my $500. It's still valid. If anybody can challenge me and tell me with evidence saying that hypoxia is not the worst, well, you can get this $500 from me. Thank you.
Thank you, Professor, for the wonderful lecture. We have received a few questions from the chat box. Because of the time limit, I've selected 2 of them to ask. The first question is, do hypoxia exist on seagrass habitat? And what are the impact of hypoxia on marine plant? The second question is, can fish really swim away from hypoxia?
The first question is that whether they occur in the marine park?
In seagrass habitat.
In seagrass habitat, I must say, I don't know. And it's a good question because seagrass itself produces oxygen itself because it's produce a lot of oxygen that may counteract the effect. And nobody have done similar things before, I would say. But I would say that hypoxia, because it's so generic, they may affect the associated fauna and flora with these and the seagrass. But I don't know. For the second question,
can fish swim away from hypoxia?
Again, this is a good question. Well, if hypoxia occurs in a small area, the fish will swim away. But as I point out, hypoxia occur in over very large area. The fish cannot swim away. Like say, even in Hong Kong, if they occur in Total Harbor, the whole Total Harbor, there's no way the fish can escape. Let alone that occur in the dead zone, all these dead zone, as I said before, the dead zone in the Gulf of Mexico is the size of Virginia. If the fish want to swim away from the circle, hypoxic circle like Virginia, takes a long time, and then they'll die before they be able to do that thing.
Yeah, thank you, Professor Wu. The
Thank
next speaker is Dr. Henry He, Assistant Professor in the School of Energy and Environment at the City University of Hong Kong. Dr. He is an aquatic ecotoxicologist who studies molecular and cellular toxicology using different aquatic invertebrate and fish models. Today, Dr. He will present a very new and interesting topic on liquid crystal monomers, which represent a class of emerging contaminants related to electronic waste. Dr. He, please.
Thanks, James. Thank you very much. Can you see my slide?
Yes, I can.
Yes.
Sure, no problem. Good afternoon, everyone. My name is Henry. So today I'm going to present a relatively new finding from our group called liquid crystal monomer, the LCMs, which is a class of emergent pollutants related to e-waste. Oh. So nowadays, I'm pretty sure everyone has a phone or phones, and together with a couple of electronic devices with LCD panels. The LCD panel is made the major displaying and interactive media from human to those electronic devices. So you may know, since we have so many devices, so the global production of this LCD panel must be a giant number. You're right. So just in 2018, this is a bit old data, but in 2018, there are almost 200 million square meters of LCD panel has been built, synthesized globally, and the number is continuously growing, not surprisingly. So in 2019, in total, 53.6 million tons of e-waste was generated. So maybe roughly at least 1/4 of them contains some sort of form of LCD panels. And particularly after 2019, after the outbreak of COVID-19, the demands of study, work, and even entertainment at home has already greatly boosted the sales of consumer e-devices. You may have heard about currently the supply chain problem in the United States. And part of it is due to the shortage of all these electronic devices. If you recently order a laptop, you may suffer from a delayed shipment. That's because the greatly boosted sales, the total sales. So you may from this information, you may predict that the production of the LCD panel will continuously increase even during the COVID-19 pandemic. So now let's move to the substrate, the LCM. So LCD panels can actually emit many organic compounds during usage and disposal. Some of them are VOCs, and these will include the LCM, liquid crystal monomers, which is the essential substances used in LCD panel as the displaying material. So up to now, according to our survey, there are more than 360 LCM have been synthesized for commercial application. More than half of them are considered persistent and bioaccumulative. And about 1/4 of them are very persistent and very bioaccumulative based on their physiochemical properties. So actually, for one single model of panel, there are usually more than 10 to 20 different kind of LCM to form a mixture. So the manufacturer will apply this mixture to form the cocktail of liquid crystal and then fill in to the panel to form this LCD panel. And this is a typical LCM used in a lot of model of laptops. So a typical LCM consists of a hydrocarbon backbone with several aromatic rings. The length of the molecule is generally larger than its diameter, and the multiple bonds along the long axis or a system of conjugated double bonds will make the molecule rigid and planar. So it's a planar molecules, and modification of functional groups are normally used to enhance the performance of the molecule, such as introducing a side chain, replacing phenyl groups with cyclohexyl groups, substituting phenyl ring hydrogen with other functional groups, particularly the fluorine. Yeah, you can see the fluorine here. So according to our study, there are, and some other group study, there are a couple of different classification of LCMs. You can see it's here. So the first one is by cyclohexyls and its analogs. You can see there are two cyclohexyl ring here. The second group will be biphenyls. So we will have two phenyl rings here. Cyanobiphenyls and analogs, and the last group is by cyclohexyl phenyls analogs. So these are the four major groups utilized in the common LCD panels. So these functional groups will contain, you can see the R here. So these functional group will usually contain cyclohexyl ring, additional cyclohexyl ring, or phenyl ring, or diphenyl acetylene, phenyl nitro group, or fluorophenyl group. So all these terminal functional groups makes the molecules persistent, bioaccumulative, and biologically toxic. So we call these substances PBT substances. So the concentration of LCMs were comparable, according to the current study, very limited. They are comparable to those of legacy and emergent organic contaminants related to electronic devices or e-waste. They're including PCB, PBDEs, OPFR, that's organophosphate flame retardant, and NBFR, the novel brominated flame retardants. So they are all at comparable level, although the application of LCMs are relatively new. So a recent study studied 17 LCMs, which has been quantified in the indoor surface dust. So they collect the surface dust like on the table, on a wall. So the total LCMs range from 0.13 nanogram per gram to 2,210 nanogram per gram, with a very clear correlation with the usage extent of e-devices, which means if a room has more LCD panel or other related devices, they will have higher contamination level of LCMs. In another study, in total 14 LCMs were determined in sediment samples collected near one e-waste recycling facility in the Yangtze River Delta region, with total concentration up to 26.3 nanogram per gram. This is not a very high concentration, but mainly because that e-waste facilities is very young. It's very young. In the Sweden study, this group detected a wide range of indoor organic contaminants. 6 of them are LCMs actually. So the predominant species, I didn't show up the structure here, the MEO3BCH, with the concentration ranging from a very high, actually 68.6 to over 1,500 nanogram per gram. So this particular species actually are also determined in this first study, the sigma 17 LCMs, but with very low concentration. So you can see the dust in China and dust in Sweden are different regarding the LCM profile. So it's a very interesting study. And very recently, another group determined 34 fluorinated LCMs in a dust sample collected from e-waste dismantling center, which is in Guangzhou. So as you may know, the Pearl River Delta is the center of e-waste recycling in China, maybe even all over the world. So that's the very high density of LCM's contamination in this area. So in that study, the total LCM concentration will be up to, oh my God, 976,000 nanogram per gram. So it's pretty high. That's in the recycling facility. So recently, the same group actually determined 39 fluorinated biphenyls, 19 biphenyls, and 6 cyanobiphenyl LCMs in waste computer television LCD panel. You may know when you break up LCD panel, you will see a lot of dust in the panel, right? So actually, those dusts are seriously polluted by a variety of chemicals, including LCMs. So the total LCM concentration range from 50 something to 2,000 microgram per cm square, and more than 36 of them were fluorinated LCMs. So the toxicity, let's move to the toxicity. So the current knowledge of the toxicity of LCM is very minimal, almost minimal, nothing. So liquid crystal manufacturers, so those manufacturers claims that LCM are non-irritating to skin and eyes based on mammalian models, so the rat and mouse. However, a very early study, it's published on liquid crystal, so not in any environmental science study. So compared to the leotropic promonic LCM, the thermotropic LCMs, and surfactant-based leotropic LCM, so the 3 different kind of liquid crystal. So the later two show significant cytotoxicity based on the bacterial viability acids. And another early study, so this Korean group used a wash-off mixture from broken laptops. This mixture contains a high level of LCM. By that time, they didn't know what kind of chemicals cause the toxic effect. But this mixture actually caused significant adverse effect in fish, including impaired antioxidant enzyme activity. So in the first systematic study of LCMs, Professor Su's discovered exposure to LCM extracts from cell phone screens significantly impair a battery of key genes. He used the chicken embryo. So particularly the upregulation of CYP1A4 up to about 14-fold indicated dioxin-like toxicity of LCMs. So LCMs, particularly the fluorinated LCM, are potential persistent, bomb cumulative, and toxic contaminant, PBT contaminant. However, similar, you may recall the story of PFC, like PFOA and PFOS, that's in early 2000s. The current extent and magnitude of the environmental distribution, toxicity, and health impact of LCM are largely unknown. That's the very similar case compared to PFC in early 2000s. A lack of standards analytical method, right now there's only a couple of studies try to quantify this kind of chemicals. So there's lack of standardized method and data for sure to detect their environmental level, especially in the aquatic environments. You can see I present several literatures about dust, but there's no, particularly no marine data. Currently, we are doing one, and hopefully we can submit the manuscript very soon. And some LCMs analog do not even have reference standards. There's no standards for it. So we have to use suspect and even non-target analysis approach to determine these chemicals. The source of LCMs in the environment is not well understood. So it might be from industrial effluent or the domestic emission. As I said, during the normal usage, these screens can also emit LCMs and may be contributed to the environmental pollutions. So LCM, particularly the fluorinated LCMs, may display similar toxicity as PFC compounds. However, their ecotoxicity and human health risk data are absent. So given the potential enormity of the contamination issue, like the e-waste issue, everyone knows the entire Earth is surrounded by those electronic devices from domestic usage and laboratory usage and entertainment usage, you name it. So filling the above knowledge gap is urgently needed for a comprehensive risk assessment of LCMs as an emerging group of contaminants. Now let's move to case study that we are currently, we just published it. So we put our focus first on the landfill leachate. So you may know, due to inadequacy of proper management, particularly in the early time, massive amounts of e-waste have actually been discarded into normal municipal solid waste landfill. They're like ordinary municipal landfills. So many studies, actually a lot of studies have revealed that e-waste related pollutants, you name it, like PFOS or PBD, all this stuff can be detected, in the landfill leachate and the soil, groundwater, surface around the landfill sites. So there's clearly a migration of these contaminants from leachate to the environment. Therefore, landfill leachate is not only a potential sink of the e-waste pollutant, but may also act as a potential point source to promote these migration of these pollutants into the local environment. So it's since everywhere has landfill sites. So as far as I know, Hong Kong currently has 3 active landfill sites, and more than a dozen of landfill sites has already been decommissioned. So currently we are working on a large survey on the including a lot of landfill sites in Hong Kong, China, mainland China, Korea, and other places to see the distribution of these chemicals. But this is definitely not a local problem. It could be global. So the aim of this project is to develop a robust analytical method to determine 38 target LCMs in leachate sample, which is actually very difficult. It's very difficult job to do. And second is to determine the concentration and distribution of LCMs in leachate sample collected from 2 typical landfill sites in South China, which is one in Shenzhen and one in Hong Kong. And third is to raise the concern of these issues, like the landfill is at the potential point source of this emergent e-waste pollutant, and urge further research on their occurrence, migration, toxicity, environmental risk, particularly in the aquatic environment. Because if we don't know whether these chemicals exist in the landfill leachates, we will not put specific effort to remediate it, right? So it will be likely discharged directly into the environment, and particularly the marine environment, to cause problems. So we have 38 LCM chemical, 1 internal standards. Most of them can be purchased, some of them can be purchased from Tokyo Chemicals, but a lot of them we have to directly contact the liquid crystal manufacturers. So there are 2 leachate samples from Hong Kong and Shenzhen. So you may know Shenzhen is just alongside Hong Kong. 2 cities are very close to each other. So a combination of ultrasonic enhanced liquid extraction coupled with saponification and silica gel packed column purification was developed for the pretreatment of the samples. And we use GCMS to analyze the samples. So here are some results. I want to keep it as simple as possible. So the first one, the A, is the recovery. You can see we did 2 nanogram, 10 nanogram, and 15 nanogram spiking. So we spike non-level of these compound into leachate, and then we will measure the real concentration. So at the end, we chose 10 nanogram for the spiking. So we can generate a standard curve to measure, to determine the real concentration. B shows the UV 254, which is equivalent to like total organic compound measurement. So you can see after the ULLE means ultrasonic liquid extraction. So you can see it significantly decreased the overall background of interference of other organic compounds and keep our target compound, the LCM, in the extras. So this figure showed the different performance of cleanup procedure. There are saponification, acid wash, silica gel column, whatever. It is also shown that all these clearance procedure can significantly remove a large proportion of we call matrix effect, ME. So combine them together, we develop this ultrasonic enhanced liquid extraction coupled with saponification and silica gel purification. So we, after all, we receive around 80% to 100% recovery for all the target compounds. So here are the results. I don't want to go to detail, but there are some key points. So in Hong Kong leachate sample, the total LCN concentration are more than double of the Shenzhen compound. You can see one is 1,120 nanogram per liter. Shenzhen is about 4. Oh, let me use this pointers. Yeah, Shenzhen is about 409 nanogram per liter. And among them, there are more fluorinated LCMs in Hong Kong's samples, more than 600, and in Shenzhen is less than 50%. So there's a significantly different profile between Hong Kong sample and Shenzhen sample. So two cities are different. So more interestingly, for example, for this A figure, we detected 9 fluorinated LCM concurrently in Hong Kong sample and Shenzhen samples. So these are the 9 LCM, fluorinated LCM, and they show a very nice correlation, which means whatever fluorinated LCM detected higher in Shenzhen sample, it's also higher in Hong Kong samples. Okay, so that strongly indicated that the mixture of these 9 fluorinated LCMs are widely used in certain popular model of devices. I'm not saying it's from some phone, but it's likely from those very popular brand of devices that result in the same combination of these compounds in the landfill leachate. And the right figure just show the concentration range that we detected in our current study. So, which is almost comparable. It's comparable to other study targeting the landfill leachate. You can see this is the current study for total LCM. These are PBDE, so the LCM is already higher than the total PBDE. So the problem is real, I have to say. So the magnitude and impact of LCM pollution is expected to be significant. So there are a couple of future work that we are trying to do, and we also want to promote this topic to many other groups so we can work together. So the first thing is that we want to expand the LCM standard bank for target analysis. Currently, we have secured a bank of 64 different species, and I believe the number can be increased further. And second is to develop methods for suspect and non-target analysis. So there's, as I said, there are more than 300 different chemicals for LCMs, and even more without standards. So we need a non-target approach for sure. Determine LCMs and analytics in various environmental matrix and even biota samples, like fish, for example, even marine mammals. Currently, we are working on, one of our students working on dolphin. So the LCMs in dolphin levels. So even for some basic toxicological data, it's missing, like LC50. the basic LC or EC50 for most of the LCM. There's nothing. And the computational toxicology can also help for prediction of those basic toxicity data. And moreover, the sublethal toxicity and toxic mechanisms are not very well understood, including biotransformation, genotoxicity, lipid metabolism, neurotoxicity, reproductive toxicity. The list could be a very long list. So definitely there are a lot of works can be done in the near future. So at the end of my presentation, I would like to thank my team and the support from School of Energy and Environment and the Stakey Laboratory of Marine Pollution. So this work has already been published on JHM. If you have any further questions, you are welcome to email me. Thank you, everyone. Thanks, James.
Yeah, thank you, Dr. He. We are slightly behind the schedule. We have one very quick question by Professor Wu Wu.
Yeah.
Hi, good talk, Henry. And could you tell me what's the approximate KOW value of all this compound?
It's comparable to PBDE. Yeah.
To PBDE,
the range is very similar. Yeah.
Okay.
Range,
Okay. Okay. Thank you. I presume that you're also working on the toxic equivalents and things like that.
Yeah. We are currently working on it.
Okay. Thank you.
Thank you. So James, I will give you the podium back.
Yeah, sure, please.
Sure.
The next speaker in this session is Dr. Venkatasen Tyagarajan. Sorry for my bad pronunciation. Dr. Rajan for short. Dr. Rajan is an associate professor in the School of Biological Science at the University of Hong Kong. Dr. Rajan has developed a broad research expertise, for example, in environmental proteomics, biomineralization, biofouling, and oyster aquaculture. Today, Dr. Rajan will share with us his important findings about the Chinese oyster and their responses to ocean acidification. Dr. Rajan, please. Hello, is Dr. Rajan here? Hello. Dr. Rajan may be upset about my pronunciation of his name. Is Dr. Rajan here, please?
If you won't mind, should we move the thoughts forward or we take a longer break? Which way better?
If you ask me, I would suggest perhaps we have the next speaker presenting first to see if Dr. Rajan will come back on time.
That is me. So
I know.
That's good.
Okay. The next section will be chaired by Professor Kenneth Leung. Professor Kenneth Leung. Or do you have the break first?
No, I can give the talk now.
Okay. Yes, please. Professor Kenneth Leung.
So I'm going to talk about eco-engineering for enhancing marine biodiversity. So let me share my slide with you. Can you see my slide?
Yes, I can.
Good. So in the coming 20 minutes, I'm going to tell you about the principle of this eco-engineering, how this can help to increase marine biodiversity on artificial seawalls. So as you know that seawall is very important to protect us from the wave action and also flooding and soil erosion. They play a very important role in urban structure. So however, if you look at the typical seawall here, we have many vertical seawall is for the boat parking. And also if the area is mainly for open area for irrigation without boat parking, then you can use this sloping whip-web seawall. If you take a closer look on those seawall, you cannot find too many marine life. Most barnacles, oysters. Sometimes you may see some leakier, those are isopods. Then you may ask why the biodiversity is low in those artificial seawall area. So if you look at this thermal imaging, the upper one is the vertical seawall. When there was a low tide in the midday with strong sun, then the temperature can go about 50 degrees Celsius. That will kill many organisms. On the contrary, if you go to the very nearby lateral rocky shore, then you can see the rocky shore with lateral erosion. It has some indentation, crevices, and up and down structure. Then turn out the temperature in these features could be 10 to 15 degrees Celsius lower than the top, those highly exposed area. So take example from this lateral shore, and you can see all these white color patch are the dead barnacles scarred on the rock. This due to the heat stress and also the desiccation stress. If they live here at the low time in the midday in summer, they will be killed by thermal stress. But you can see many mussels here and also barnacle here. They go really well because this area can trap the water, allowing them to have a much better environment, cooler area for them to survive. As you can see, many animals actually like the crevices area as well. So another aspect is important to talk about. When you go out to the seaside with your girlfriends or boyfriend, you walk on this rock surface, then you can see the color. Sometimes it's brown color, sometimes it's green color, some are red color. If you scratch on the surface and then using a microscope, you will find them, they are plant species, they are microscopic plant, like a diatom and also the cyanobacteria. They are very important food for the grazer, those who eat vegetable, like many of us. As you can see, this chitin and limpet and this periwinkle, they all feed on this biofilm. You can see after they graze on them, the biofilm gone. So this is a very essential element for the rocky shore. If the temperature too high, this biofilm will die also. So take a look at this diagram. First of all, the seawall, you want a seawall to harbor more species. First, the seawall have to be warped so that it can anchor the nutrients, protein, carbohydrate, and so forth, and then attract the bacteria to go the biofilm, and then subsequently the plankton, zooplankton will come into place, and then finally the whole community will be formed and attract fish and crabs to use this habitat. So we try to develop different types of eco-engineering design product. As you can see here, if you have just a very, very smooth surface and very warped surface, then you can see even the same tidal levels, same environment, not much life on this smooth surface. But you can see many life, they like this warped surface. In actual fact, this is not new because when Professor Wu was young, when he did his master's degree at the University of Hong Kong with Professor Brian Morton, he worked on barnacles. He told me that he can make a heart shape so that the barnacle will follow the heart shape to attach. If you make the concrete surface rough, they will just attach to it. The same idea we use here, we make groove and up and down structure, then the barnacles really love it. Not just barnacle, but also the algae and also the oyster. They love this rough structure because it can retain the organic matter and then allowing the formation of biofilm. And then the animals love to live on this kind of nutritional rich area. And we also find another problem is the use of concrete. Most of these cement for building purpose, they have high pH around 13 or 12. Why? Because they need to protect the reinforcement rebar, the metal bar inside, so they have to retain the pH as high as possible to reduce corrosion. And also when they do this manufacturing process, the surface in most cases are very smooth. It's not very good for the marine organism to settle on. So to rectify this problem, then we have to modify the surface to be very rough. And also we have to reduce the pH to closer to the marine environment. The seawater pH is normally 8. So we have to reduce the pH to that level, if possible, using a carbonization technique or using different concrete mix. At the end, we cannot use a metal rebar, so we have to use a glass fiber as the reinforcement structure. After that, then we can build the Lego, say, with the shading area to cut down the temperature, with crevices and water retention elements so that the animal can have a more benign or better environment to live. One example is quite interesting. In Hong Kong, in Sai Kung, this is called Yin Jiam, the place. There was one accident, a barge sank at the bottom due to a traffic accident. It carried a lot of bags of cement. And then after more than 35 years, you can see that not much life on that because it is smooth and also high pH, no those microhabitat for animal to use. So then if we want to make the concrete to be livable, we have to mix the above mentioned changes. So after we design this kind of new eco-engineer system, then you can see if we have a whole, like a tube structure, the penny fish, they love it. Then you can see the crevices we create, the cowrie and the periwinkles, they love it. They also, the dog well lay their eggs inside those burrow area. Then the whole structure after half year to 1 year, you can see a very rich with mussels and spongy, and also sea anemone. The crabs really like those crevices. This technology actually have been booming over the past decade all over the world, from UK to Seattle in US, Sydney in Australia, Florida in USA, and also in Israel. They realized this problem of the desert in seawall. Now we can improve the situation to enhance not just biodiversity, but also fishery resources in urbanized coastal area. So if we do it properly, we can allow 3 major services for the seawall. First, shoreline protection. Second one, we can enhance the ecosystem. Finally, if the area big enough, we can allow people to enjoy the waterfront, okay? So if we do well, then we can enhance biodiversity, reduce biological invasion. Some studies showing that with the engineered ecosystem, then there will be reduction of invasive species because there will be more predators to control them. And then if we build the eco-engineered shore here in between 2 lateral shore, then we can increase the connectivities and allowing better gene flows in the entire ecosystem. So the settled down organism like mussels, bivalve, oyster, tube-worms, they can also filter the water to clean up the water, eventually improve water quality. So if we make the eco shoreline longer enough, it can help to improve fishery resources and also fix carbon as well. So if the area big enough, like this one in Sydney, they use this lateral sandy soil, this sandstone. You can see after millions years of erosion, then you can see many holes and structure there, allowing the marine organism to settle. Those are very lateral type of rock. Then people love this park because people can sit there, enjoy the waterfront. During the low tide, the kids can go down to see the marine organism. Now the Hong Kong government just copied this design into Hong Kong now. In North Pond and also in Causeway Bay, we are building similar type of steps in Hong Kong. So now come back to the science. We all know that if we increase the capacity of the ecosystem, we're going to have more different species to live in. Just for example, if you have a farm field, just those grassland, then you have only few species. If you start planting different trees with different fruit and different leaf structure, and then you will harbor more different mammals, birds, and insects. And then you keep doing this and then make it like a mosaic, then you can enhance the biodiversity, overall biodiversity greatly. So then in this study, we try to test this hypothesis. Then we have a very simple structure. This is a control, nothing, just a concrete tile. And then the next one, we have this ridges coming up 2.5 cm. And then the next one is more complex, 5 cm. Is a much deeper ridges. And then we clean the seawall as the reference site or control site. And then side by side, we also want to test this hypothesis called, it's called lateral assemblage. So we just stick the rock oyster on top of these 3 different designs to enhance the capacity. So the capacity increased from here to here and also from here to here. Then we try to compare to 2 different sites as well. Then we install them on the vertical seawall and compare also with a reference site in the lateral rocky shore next to the seawall. The entire study lasts for 1 year. Then we have monthly data and also the final study. We took out the tile and then do a very detailed study on everything. So it was a bit of surprise that we found many species we never seen on the control seawall before, like these crabs and also these amphipods and also these bivalve. So it means the new habitat working really well to harbor new species. Then it answers our question that the new structure can increase biodiversity. So after 1 year, when we look at the results, we can see from the fact 1, 2.5 to 5 cm ridges, we can see increase of number of species. If we put the rock oyster on top, then the increase even higher. Same as the other side, Lo'onpai, same trend, okay? However, sorry, for the abundance, for the same site, Lo'onpai. But some 水果 we only see in the one without the oyster. The one with oyster, we see increase, but without those pattern linked up to the tile capacity. So we find that in some 水果, because it's a low west facing, during the low tide summertime, it's facing to the west. It's always have strong sunlight. Then the capacity couldn't really cool down well. So that means we won't see this higher increase when we compare to this site. So the orientation of the device also need to take into account. So we would learn from this. Now we design something that will use the back side of the tower rather than in front. The front side is like an umbrella to cut off the light and heat. Now we try to use this new system. So if you look at those with the seeding of the oyster, the rock oyster, you can see they can harbor more oyster. The babies will join them at the end. So this is a cheap and good method. If you can't afford to buy this engineered product, you can actually stick those oyster on the seawall. Then guarantee you will have more oyster babies come by and also more other species to join the ecosystem. Then we also find most species, they like to sit in the bottom of the crevices rather than on top of the ridges. They try to hide away from predator. At the same time, it's a much cooler places with more food in the crevices. So overall, we find that with this, just the capacity made by the tower itself, then the number of species can increase by around 20 to 50%. And with seeding, then the increase would be around 40 to 75%. Then for the abundance, the increase even more massive, can increase 4 to 5 times, depending whether we add the oysters. So then this experiment demonstrate that this simple technology can enhance the biodiversity greatly. Then the European Commission also adopt our result as part of their Science for Environmental Policy to inform their leaders to also try to adopt this new technology in urban infrastructure. So our project actually joining with the World Harbor Project. In these 14 cities, we're doing the same experiment together. Then the result almost very identical, except that we found in tropical area, the recovery of mobile species is higher, but in the temperate region, then the recovery with the engineering tower, we see more sessile species like barnacles and seaweed. So some differences, but we still can see the enhancement of biodiversity due to the ecosystem capacity constructed by the modified towers. So now I would like to share more little bit complicated concept. So in this diagram showing that x-axis, when we have more development disturbance, then the beta diversity means number of new species will be declined. So then we try to reverse this using eco-engineering by pushing this curve backward. Imagine if we create more different types of habitats, then we should see this reverse curve. We can see enhancement of biodiversity. So we try to test this hypothesis using more different devices this time. We implement really big panel, 1 meter by 2 meter. And this eco tower more complex decided by our team and Polytechnic University. And the oyster shell basket, plus the tidal pool, and also this armoring unit. They presenting different type of new habitats we create on the artificial shoreline. So you can see all the structure is really big, 1 meter by 1 meter by 1 meter for the tidal pool and also the armoring unit. These are the basket, this 1 by 1 meter with ½ meter depth, okay? Then we installed them for 2 years. The project just finished. Then I just show you some of the results. This is the oyster shell basket. We put a batch of oyster shell into the cage, forming a more natural habitat. Then you can see many birds like our artificial seawall here on our structure, because sometimes they can have easy meal to take the fish from our structure. This is the overview aerial photo of our devices. Then we do the field measurement every season, once every 4 months. We are doing the survey. Then this just a 9 months data, but you already can see the trend. The pool and armoring unit better than the control seawall, and the panels also perform better in terms of species richness. Now the results are even more pronounced after 24 months. But we do find the winner actually is not those concrete structure. The winner is this oyster basket. We just put live oyster or just empty shell, the dead oyster shell. They can harbor more than 70 species in one site. The result is amazing. And then because the cage itself as a protection, protect the animals from predation, then we can find many different species inside the cage just after 1 year. So as you can see, just 9 months, you can find on the live oyster, we found 20-some species, same number of species found on just shell. So what we suggest here, we don't need to put live oyster, we just need to put the shell. You will find a similar species harbor on the shell. Then the cage is important as a protective environment. Then you can see another 20+ species joining. So come back to my earlier mentioning this reversal of this curve. Then we just use one site as example. Each column represent, this is just the ordinary seawall. And then we add one device, the pool and then armoring unit. Then we find new species we add on the curve. Then you can see the curve actually pushing backward this way. That means if we create more and more different habitat, we can harbor more new species to enrich the overall biodiversity. We are doing more complex analysis at the moment with the 24 months data from 3 different locations. So hopefully, maybe in a few months' time, we can share the final result with all of you. So the key driver why we are interested in this project, because the mainland China, Chinese government, our nation, want to do big-scale ecosystem restoration across the whole country, from rainforest to deserts to the marine environment. There will be many major projects for protection and restoration of coastal ecosystem, especially in our region, Greater Bay Area. The government encouraged us to implement this eco-shoreline or eco-engineering of shoreline implementation. Then Hong Kong government have been implementing this 3.8 kilometer long eco-shoreline. I was invited to be one of the designers of this new eco-shoreline in Dongchong, next to our airport. So in the area closest to the river mouth, the estuary, we have the mango plantation ecosystem. And then in the exposed shore area, we use this eco-concrete to build these fun sizes of pools and with different structure I showed you before. We already implementing a trial. The trial result really good as what we predicted. So then we also won important design prize for the sustainability award. So we actually we start from nothing. I was new to this business. When the government thought about eco-shoreline engineering, they have no idea. So they asked me whether I can help. So I told them that I need some help from outside. Then I invite all the leaders in the field. In 2016, we organized the first workshop to learn about the concept and the challenge. And also they give us advice on the design of the Dongchong eco-shoreline. And then in 2018, I even invited more people, 28 people, 25 experts to join us. Then I locked them up for 4 days. Then we wrote these very, very important papers together. And this paper summarized all the theories and practices and consideration and also assessment criteria all in one. Then if you want to know more about these topics, you can use this QR code to get a paper from us. Then in coming January, we will have another workshop. Then we will focus on the develop the specification when we develop this kind of eco-engineered product. So then you all are welcome to join us. The conference will be in hybrid mode. Then from overseas people, you can join through the online system. And also since the conference is also part of the United Nations Decade of Ocean Science Action. So then we offer some free registration pages. Please contact our conference secretary and we welcome you to join us. So finally, I would like to thank all my partners and also most important, our sponsor, CEDD. Thank you very much.
Yeah, thank you, Professor Leung, for his wonderful talk. Is there any question from the floor, please? If not, let me ask a quick question. Eco-shoreline is a very good concept to restore marine ecosystem on man-made seawalls. How do you compare the effectiveness of this artificial structure with other more natural restoration means, for example, planting mangrove, a strategy used by some audience here in the Philippines?
So if you try to restore a natural shoreline, that is easy to do the mango plantation, it will work well. However, we're talking about this kind of reclamation is different ball game. In a natural shore, you have the inclination from land to water gradually. But when we talk about reclamation, you like this. It's drop that. In fact, what the seawall you can see is just like the hanging out like this, and then it go all the way straight down 20 meters. So if you want to plant mangrove, that means you have to sacrifice some land footprint so that you have a extension like a gradual slope. We did it. We are going to do it in Dongchong, but still we're talking about 3 to 5 meter stretch area. Is not too big. If we want to have a very nice mangrove system with outwelling and succession, this is not feasible because you only have a limited space. So then in that case, we have to use a combination with those concrete-based engineering techniques joined together. But as I show you in our result, we find that the oyster shell inside a cage is the best. When we compare to engineering product, they may harbor up to 30, 40 species, but the oyster shell cage can harbor up to over 70 species. So that means in the way forward, we will try to use the oyster shell more, put them inside some natural fiber rather than using the metal cage. Hopefully they look like more natural in a sense. So we just got a grant to deploy oyster shell bags in Lemma Island. So we will try this out.
I see. Thank you, Professor Leung. Wonderful talk. This is an announcement. We have a slight change in the schedule. We will have a break now. We have a break now, let's say 5 minutes. 5 minutes. After the break, Dr. Apple Choi will give her talk on coral restoration, followed by Dr. Rajan on oyster aquaculture under ocean acidification.
Okay, thank you, very good.
Okay, 5 minutes break. Thank you. See you. Good afternoon. Welcome back to the symposium. Dr. Apple Choi is a research assistant professor in the School of Life Sciences, the Chinese University of Hong Kong. Her research interests include reproductive and larval ecology of coral, coral recruitment dynamics, and coral restoration using sexually recruited corals. Today, Dr. Abel Choi will present on the topic on degradation and restoration of coral communities in Hong Kong. Dr. Choi, please.
Right, so thanks, Chair. So hi, everyone. My name is Apple. I'm a research assistant professor from the School of Life Sciences of the Chinese University of Hong Kong. Today, I'm very happy to be here to be able to share with you this topic on degradation and restoration of coral communities. And this is actually a case study and a kind of an experience sharing from Hong Kong. Now, believe it or not, this video is actually taken in Hong Kong. This is not even in our marine protected area. It's actually a site in Sai Kung. And here in Hong Kong, we basically do not have extensive reef structure, as you can see, but we do have corals that usually attach directly onto boulders, bedrocks, and form these scattered coral communities along the coast. As what you see, our coral coverage can also be very good. It can even go up to like 80% coverage in some area. So people usually describe Hong Kong as a marginal coral environment. It basically means that coral communities here are living close to one or more of their survival thresholds. For example, corals in Hong Kong, they experience very large fluctuation in temperature year-round. Winter temperature, it can actually go down to 14 degrees Celsius. And because of the turbidity, of course, this photo, of course, is being taken in one of the best days of the years, which only happens, lasts for several days in a year. But on average, the site actually looked like this. So because of this turbidity, corals are usually found in quite shallow waters, and that's why they are also frequently exposed to salinity fluctuation due to heavy rain or runoff. And because of all these suboptimal conditions that they are experiencing, the coral population are currently experiencing, they might have already been well adapted or acclimatized to more stressful environment through generation of selection or experience from past exposure. Now, from previous talk, like earlier this morning from Professor Chu and Walter, you've already heard a lot about different stress responses of Hong Kong corals. So indeed, there are actually growing evidence suggesting that our corals in Hong Kong might be potentially tolerant to global stressors, such as rising sea surface temperature. And for myself, my research actually focused more on the early stages of corals. So here's one of the examples that I like to share with you as well. This one is on Platygyra coral, one of the most dominant coral genus in Hong Kong. And we exposed the competent coral larvae to elevated temperature and also lower salinity, and see if it will actually affect the settlement and post-settlement survival. I'm not going to get into details here, but I just like to let you know that even up to 5 degree elevated seawater temperature, it did not have any, as you can see, significant effects on both larval settlement and post-settlement survival. And this is actually kind of a long-term studies. It lasts for 56 days. So it's kind of like a prolonged exposure as well, which basically the result makes it the most tolerant coral species ever reported in the literature. And similar tolerance also applies to other aquaculture corals in Hong Kong as well. So overall, they might actually be the ones who can survive global climate change. And if there's, of course, a big if it's really true, if it's really the case, then they do have much higher conservation value. And I really love to collaborate and explore more in this area. Now, very quickly, we'll shift our focus from a more global threats to local stressors, because as you know, over the world, reefs are impacted by bleaching event, which cause a decline in percent coverage. While isle corals, if you can again recall earlier from Professor Chu's talk, if our coral bleach, they do have quite good recovery. However, Hong Kong corals are also under threats. Okay, and in Hong Kong, it's actually one of those places where coral decline are mostly due to local factors. Okay, like for example, coastal development, pollution, overfishing, etc. Now, back in 1980, there's two sites called Bush Reef and North Reef, two islands located in this called this Toloharan Channel. There has been record of very high coral coverage to up to 70 and 80% coverage. Just if you can recall, just like the video I just showed you, very high coral coverage. And worth mentioning is that this number is even higher than some of our marine parks nowadays. So what happened was due to pollution and sedimentation caused by urbanization, coastal development in surrounding areas. The coral coverage just dropped dramatically to below 2 and even 0 in coverage. So coral was once here, but because of pollution, they were gone. Unfortunately, even after our government has put in lots and lots of efforts to improve water quality, the coral just never come back. And it's truly disappointing, but this is the fact. The fact is that our data actually shows that the whole coral system was basically damaged beyond repair. Okay, first, there weren't not many remaining corals to grow through asexual reproduction. And secondly, there's just very low coral recruitment coming from replenishing from sexual coral reproduction. So the lesson learned here is that coral communities could so easily be destroyed, and yet the natural recovery can be so difficult and even unlikely, like in this case. So now, with the improvement of water quality, we decided that perhaps it's time for us to do something about it. And that's where we came in. I'm not sure if you've been here before. So this is our marine lab, marine science laboratory in CRHK. What we do here in the lab is that we actually culture corals for restoration studies. And to be honest, coral restoration is still in its infancy in both China and in Hong Kong. And most of the studies we have so far we have used asexual fragments only. Simply because it's easier, this is also so-called the asexually propagated corals. These asexually propagated corals, they're actually broken portion of a coral colony in the field. So these coral fragments, once they're broken, we collect them. People actually give this fragments a very beautiful name, that is the corals of opportunity, meaning that if you don't save them, they may probably die in the natural environment. So me and my team will go out and collect them. We then further fragment them into smaller pieces, because sometimes when you bring it back to the lab, it's really large, or it's not that healthy, like the whole fragment. So sometimes we'll further fragment them into smaller pieces, and then we'll grow them in our coral nursery, allow them to recover. And eventually we'll take them out to outplant onto our previously degraded sites, our target restoration sites. So this is some of our ongoing project in Tolo Channel, the Bush Reef and North Reef, and of course it's one of the site, the picture in one of the site. And we actually have different species outplanted, and after 2 years, we actually achieve around 90% of survival, which is really a very good number in terms of the survival after outplanting. And as for the growth, these are two very quick examples. We have 5 genus outplant actually, so these are 2 quick examples. This picture actually shows you that after a year, you can see that the corals in general, they grow very well, especially for this branch form, a copra coral. It's really amazing because it gave us like almost 20-fold increase in size per year for this copra coral, roughly 8 centimeters in diameter. And of course, we also have other corals outplant, like this Platygyra coral, a massive form one, around 1 fold per year, roughly 2 centimeters in diameter per year of growth. Which is as expected because different growth form, their growth are kind of different. So with our coral outplants, we can already begin to see them being able to bring in a bit of biodiversity. You see, there are seahorse, butterfly fish comes in and live around or use our coral as their anchor. Okay, live around corals. So all these actually indicate that once the problem is solved, I mean the problem of degradation is solved, say water quality is improved, there's actually great potential for restoration. And of course, in this case, branching corals are really a great starting point because they grow really, really fast. Great candidate for restoration. But of course, when we talk about restoration in general, we need also to put in efforts to restore other massive form corals as well. So on top of that, with the technique in larval culturing, my team also do massive coral culturing using sexually propagated propagation approach for restoration in Hong Kong. So the approach is actually involves the collection of gametes from coral during spawning. We culture them into larvae, settle them as baby corals on the substrate that we provide. So these baby corals, we then cap them in our land-based nursery, and then we outplant them to the field at a much larger size for restoration. It has to be at larger size because in Hong Kong, we already have studies showing that coral recruitment itself in Hong Kong in general is very, very low, both on settlement tiles and on natural substratum. So one of the major problems we have nowadays is low post-sediment survival of recruits in the field. Basically, if you just outplant them in very, very early stage, after a month or 3 months, all of them will be gone. Okay, so which basically means that if we try to use direct seeding of mass cultured coral larvae on natural substrate, it seems not quite possible in Hong Kong, but we like to give it a try, but it may not be very promising. And this actually reveals a huge problem if we try to use sexual propagation approach for restoration. So I've listed some of the major challenges here. For example, in our waters marginal environment, we have kind of very high sediment year-round, and also in some sites, there are actually very intense competition of space with other benthos. For example, those fouling organisms like oysters, barnacles, browsons, algae, and coral being slow growth is one very major problem contributing to the local sediment survival. For example, this one is a 4-year-old juvenile corals, a cockroach species. We grow them from baby corals in the field, so we know that it's 4 years old already. But it's 4 years and it's still only 8 centimeters in diameter, so it actually indicates that these corals just grow very, very slowly, and that's one of the problems we face in Hong Kong when we talk about restoration. So, and of course, when they grow that slowly, other organisms comes in and they will easily overgrow the corals, outcompete the coral. So marginal coral environment in general will have all these limitations, challenges, make restoration even more challenging than it already is. So you can see or imagine life is really hard for little coral recruit in general in the field. So in order for the whole idea of sexual propagation approach to work, we actually have to grow them to a much larger size before we do any outplanting. And I'll give you an example like this one. These little ones, we actually have been culturing them in our lab nursery for 2 years, and it's around 2 to 3 centimeters in diameter. And in 2019, we have our first ever outplanting of this 2-year-old sexually propagated juvenile corals in Choral channel for restoration purpose. We were like gambling, you know, like show hand. It either work or it doesn't. So it's kind of risky. But thankfully, the result is actually quite promising. We achieved 95% of survival in the first year. So the hard work kind of pay off, and they are growing quite well as well. So this species is a Copra pruinosa. And we know that the sexual technique is going to work in Hong Kong because we already have example of successful outplant of corals being reproductive viable in the field. It took us 5 years for this Copra tumida corals, but still it's possible. So now what we need to do is a bit of upscaling, and this is what we're trying to do in future. So now just a quick summary in here. people use asexual and sexual coral propagation approach. So asexual coral reproduction for restoration, this technique has proven to be quite successful in Hong Kong so far because survival is quite good and it's very easy to get these fragments. Just go out and collect them. The fragment size is much larger to begin with, so that's why the survival in general is better. However, there's also a problem associated with these technique, asexual propagation technique. First is the source is limited because it actually rely a lot, or it depends on the existing coral colonies to provide these kind of natural coral fragments. So it seems not to be a very, very long-lasting solution if we want a lot of coral source for our planting. And we also have to be aware of the genetic diversity. For example, if we collect the coral fragment, we break the coral fragment into smaller pieces. Although it looks as if we have lots of corals for our planting, but they are actually clones to each other, which means that genetically they're actually the same, meaning that if one of them died due to unfavorable environment, probably all of them will die at the same time because they're cloned to each other, they are the same. So this is where sexual coral reproduction comes in. Unlike those from fragments, baby corals from sexual reproduction, we mix the gametes from all parents together in the very first place to create as much diversity as possible. So they're more genetically diverse, and simply because every single individuals are different in their genetic makeup. So hopefully they are even more adaptable to environmental change than those created from fragments only. So this is very important because we want our coral outplant to not only survive just now, but also in the future. And we know that the environment is changing, and we hope by enhancing the biodiversity, we just let our mother nature to determine which one to survive in future. So our ultimate goal, of course, is not only to restore the coral, but the whole ecosystem. So this is just a beginning. The best approach, of course, is to use both at the same time. All right, so I'm just going to close by saying that Hong Kong, Hong Kong is a very interesting place for coral research. It's a marginal coral environment. These corals, they may have enormous potential for expanding our knowledge and understanding of the limits of coral adaptation or acclimation in this era of global climate change. However, the most immediate threats for our corals are local stressors, coastal development, pollution, sedimentation, overfishing. So restoration is a long-term process, it's not a very quick fix. And of course, we should always, I think the rule of thumb is always we try to prevent damage occurring in the first place, because it's much less expensive to protect reefs or mitigate impacts than to try to do all sorts of attempts to restore damaged ones. And it's also not a guaranteed success as well, after all. And here, our ultimate goal is to help our corals survive changing climate. And with that, we're trying to do, or our lab, my team is trying to do, our future 5-year plan is actually we continue to optimize techniques for using sexually propagated corals for restoration. For marginal environment, say we have to optimize on how to minimize the post-sediment mortality, develop methods for upscaling in a more cost-effective way. We also aim to enhance the resilience of coral stock, integrate more proactive approach to ensure long-term viability of restored corals in this changing climate, such as, for example, there are lots of initiatives happening or being proposed, such as identifying resilient corals, managed selection, breeding, chimerism, a lot of different strategies. And finally, my team, of course, we do a lot of community outreach in Hong Kong, trying to raise more community awareness in this issue as well. So with that, we aim to develop a coral restoration protocol that would be applicable not just in Hong Kong, and also with a much wider application, such as restoring coral communities in other parts of southern China as well. So finally, I would like to take this opportunity to thank Professor Put An, my former supervisor, also my lifelong mentor, and all members from my lab. So because all these definitely is a team effort, it wouldn't have been possible without all the support and assistance. And of course, I also like to thank the below funding support that I received from AFCD and also the Pew Fellowship in Marine Conservation. Yep, thank you.
Yeah, thank you, Dr. Abuchoi. We are so happy to have you here in Hong Kong working on coral restoration, wonderful research and conservation effort. We have received 2 questions from the audience from the Philippines. The first question is asking which species you have used for sexual propagation.
Yep.
The second question is, how are the growth rate compared between the sexual and asexual out plant?
Okay. Thanks a lot for the questions. I really welcome this kind of sharing, knowledge exchange. I really want to, know more about the reefs restoration happening elsewhere as well. So in my case, the sexual reproduction, because of the manpower, there are a lot of limitation in the very first place. So right now, up till now, we are able to work on 3 different coral species. One is Platygyra, but they just grow so slowly. So we're still working on the Platygyra coral because it's the most dominant coral species we have in Hong Kong. But at the same time, we also explore the Acropora because they just grow much faster, give us higher survival. I so far have been working on 2 Acropora species as well, together with the Platygyra, that is the Acropora prenosa and tumita. Yeah, so these are mainly the 3 coral species, and we're actually exploring to see if we can try or explore to work on more coral species in the future. Yeah. And as for the second question, is about the coral growth, comparing the fragments and the those baby corals from sexual reproduction. I would say what really surprised me is the growth. I will go back to the slide showing the growth of asexual fragments, because in general we know that Hong Kong is not an optimal environment for coral growth. So we will think that the growth rate will be in general slower than when compared to the tropics. And I think that's true, but in this case, what really amazed us is the growth of these Acropora fragment. Just 1 year time, 20-fold in size change. But I think we have to be careful when we interpret this kind of growth, because those are very kind of small fragments, and nowadays people always talk about even micro fragmentation as well, because we tend to think that if they're small, they tend not to devote that energy into sexual reproduction. So perhaps it's one of the reasons why they have all their energy for asexual reproduction. So that's why they grow much faster. So I think we have to be a little bit careful when we interpret this kind of early growth, early outplant, early growth, because their size is really small. So perhaps once they reach sexual stage, their growth will not be that quick, in marginal environment. And in general, if we measure our adult coral growth outside the field, it's not that quick, actually, to be honest. And as for the juvenile corals, this is, I would say in general, of course, Acropora coral is the fastest growing coral species. In Hong Kong, this one is like, if we grow it from baby corals, it would be like 2 centimeters per year. Yeah. So it's not really fast, but it's still the fastest one so far. So thanks again for the questions, and I really will welcome more. Perhaps save it to later discussion, or you can feel free to email me, actually.
Yeah, thank you, Dr. Abuchoi. The next speaker is Dr. Rajan. Dr. Rajan should be here with us. Would you please upload your PowerPoint slide?
Yeah, I will do it.
Today, Dr. Rajan will share with us his important findings about the Chinese oyster and their responses to ocean acidification. Dr. Rajan, please.
Yeah. Hi, you all hear me? Hello?
Yes, I do. Yes,
yeah,
I do.
okay, great. Thanks for the great opportunity. Sorry for mixing up the timing, and also thanks for giving me this opportunity to talk in later time slot today. Very nice. Enjoyed the talks so far. Thanks for all of you staying this long in this Saturday afternoon. So I'm going to be relatively giving you a relatively simple talk and give you rather a story instead of more technical data and the talks like that. So basically, I'm a Rajan. I work from Hong Kong U, and also work in the Surrey Institute of Marine Science. And many of participants in this meeting probably may aware of this institute and my work in oysters. So probably I'll give you a story about it. So that's what we have, a brand new Marine Institute now. You see the picture. And also we have set up recently Hong Kong Oyster Hatchery and in a innovation research unit. And we'll be talking on some of those things. So the three points I'm going to share with you today and discuss with you in this 20 minutes or so. The first point about aquaculture, and particularly oyster aquaculture in southern China, more focused on southern China than in other part of the China. So oyster aquaculture in southern China, that's the first point. And second thing, I will tell you what are the challenges, problems that oysters in southern China are facing due to various anthropogenic stressors, including climate change, and then who will be some sort of winner and loser under this scenario. So how to identify the winner, because aquaculture is all about winners. They don't care much about the losers. So we try to I try to tell you how we could identify those winners under this scenario, and then what is how to use those winners for sustainable aquaculture in later on. That's the third part. So those are 3 points I will be talking today with you and discuss with you. Oysters, I think many of you very familiar, and particularly Professor Wu here. He has pioneered the work in Hong Kong many years ago, intensively worked on aquaculture in La Fusan, the Deep Bay area, and figured out the problems that the species is facing in this region and things like that. But nevertheless, many of you still love oysters, including Hong Kong oysters, including those cultivated in the Deep Bay, and they are all very happy because we cook them in multiple ways and in typical Chinese ways, in multiple cuisines. They are great. Remember, oysters is omega rich, relatively less cholesterol, huge protein turnover, and is an amazing food in that respect. So oyster aquaculture is no doubt is #1 seafood in this region. In terms of in many other things. And also, I think many of you maybe remember, maybe know, or if not, this would be interesting information to all of you. Although oyster aquaculture oysters are cultivated around the world, over 80% of oysters, aquaculture oysters, comes from China, which is huge. Which is a huge in terms of the thing. And among those 80–90% cultivated species in China, there are 3 species that are predominant. The #1 contributor for this production is C. ungulata. We call the Portuguese oysters. Oh, Professor Yu? Hello? You all hear me?
Yes, I do. We hear you.
Okay, yeah, because I saw Professor Wu was trying to say something. So that's fine. Let me continue. So the Portuguese oysters is it's really the #1, which is cultivated in Taiwan and Xiamen and in the middle part of China. And then if you come down further, you see another major species, which is called the Hong Kong oyster, which is also another second most contributing species. It's cultivated from all the way in all Guangdong province in this region and all the way to Hainan Island. The Pacific oyster, which is well known to many of you and in the cultivated globally because it's invaded in other part of the world for commercial purpose, is mostly cultivated in northern part of China, which is the third major player among the three species. So this is how the scenario is. So the Portuguese oyster is the #1, Hong Kong oyster is the #2. These two oysters together contributing almost 60% of the global oyster production. So this is a huge and significantly important one. And among these two species, the Hong Kong oyster is somehow is cultivated mostly by the local growers, indigenous industry in southern China, compared to the Portuguese oysters. If you go to Xiamen, if you go to the other side, it's relatively rich oyster growers and more industries involved in the cultivation of the Portuguese oysters. And Hong Kong oyster is still controlled by local growers and more indigenous growers and things. But compared to these two species, Portuguese oyster and Hong Kong oyster is relatively rich in omega-3, natural antioxidants, high protein. We have lots of data coming up between these two species. So we have set up a food quality analysis testing facility also. This is all the data is clearly telling us this Hong Kong oyster is rich and taste-wise and is a super great in terms of that. But these two oysters in generally in southern China are facing several problems in recent times, particularly in the past 5 to 6 years. In these years, at least I'm involved with this industry and I come across all these things. There are 8 problems they are facing now, but I'm not going to talk about all of them. And particularly at the number 7, which is very relevant, very interesting to this meeting as well, is the climate change. Again, climate change is very, very well known to every one of you, and nothing super new in this case. But what is interesting here, the climate change, what I mean mostly nowadays is the changing in the seasonality. So the winter is the perfect golden time for oyster aquaculture, where they are not developing goners, putting all their energy for the meat, and the growers harvest during the Chinese New Year. That's the winter time. And so this is the winter time is a critical time for aquaculture, oyster aquaculture. And this winter time, because of the climate change, is either shrunken or it's much warmer winter. This is what the scenario is in the first few years. So in the warmer winter, what's happening, oysters started reproducing much earlier than it's supposed to be. They're supposed to reproduce in March, April, May, but they have started developing goners in February. So just at the time of their harvesting, that's killing oysters. When they started reproducing in the winter time, it's an unusual period. They are experiencing unusual pathogens, I guess. Then their mortality is huge. And almost 80–90% of oysters in southern China die before the growers harvest them. This is we call winter mortality, which is the major issue now. This whole southern China aquaculture people are facing. So we need to work on these issues. We need some innovation to sustain this oyster aquaculture. That's the 2 points I'm going to talk in the later on the slides. So it's the winter mortality is not about just the dying of oysters in the winter time, because the aquaculture in southern China also depend on those natural wild oysters and the seeds that produced by the wild oysters. If 90% die, you can't get enough seed from the wild. That's the issue as well. We don't have much hatcheries, oyster hatcheries in the entire southern China to supply for the whole aquaculture industry. It's coming up now, but is not enough yet. So the healthy seed production is also a question mark here, because oysters dying in the winter condition, or they are not healthy. If they are not healthy, if they are dying, they may not be producing healthy seeds for the next generation. These are the 2 issues. They're dying and they're not producing healthy wild seeds. If these 2 things are happening, if the oysters is dying, and probably the food safety and the food quality also matters, because they are struggling to survive. So you can't expect them to have a very tasty oysters with plenty of glycogen, which is question mark. So the quality matters, quantity is seriously affected, and the healthy seed production for the next generation is also affected. So these are all the issues I assume these southern Chinese oyster aquaculture people going to face in next few years' time if climate change continues to affect this industry. And so we have to find a solution for this thing. This is an oyster aquaculture area in southern China, where I go very frequently do the experiment. You see here, the growers keep telling me 90% gone, 100% gone, completely disappears in February, March, April time before they harvest. So this is a big issue in mortality. All these are happening. So why this is happening? Again, I'm not only one looking at this problem. There is my colleague, my good friend, Professor Sini Yu from Guangzhou, has been working in this field for 10, 20 years of time. It's hard to figure it out why they are dying in winter time, at least in recent times. But as I explained to you before, is obviously they have started reproducing earlier in time. Temperature is much higher in warmer winter areas in winter time. Then it's pathogens attacking them and killing them. We have to verify this hypothesis, but very likely this is going to be the scenario. But at the same time, there's another issue is happening. Oyster aquaculture, particularly the Hong Kong oyster, compared to the Portuguese oyster. I always be comparing these two species. Okay, the Portuguese oyster, more coastal species, live in high salinity areas. Hong Kong oysters live in estuarine areas, low salinity areas. So there is a drastic difference between these two species in terms of their survival skills. So the Hong Kong oyster, since there is no space for the cultivation in the estuarine areas in the entire China, is all well developed. So aquaculture people try to go a little bit far away from the estuarine areas to more coastal areas. Coastal areas is a high salinity areas. So these species die there, but they don't have option. They have to go. So they're trying to push it in that way. That's the one issue they're facing. At the same time, when you go to the more coastal areas, the coastal areas is in the winter time is much lower pH. So it's anthropogenic pH, we can call as acidification, either ocean acidification or acidification, whatever the reason, the pH is lower in the coastal areas where this species is now cultivated intensively. So these species are experiencing this acidifications, low pH scenarios quite often than ever before. So that's how the things that are happening is there. So now the pH is declining because they are cultivating an offshore area, and then is the warmer temperature in the winter conditions. So who will be the winner and loser under these scenarios? So either oyster is going to adapt within few generations to tackle this warmer winter, and also at the same time their lower pH at the same time. That is the biggest question we are asking now, and in this field. Again, I think many of you, I'll be going through quickly these slides because it's more technical. But the species adapt to the new environmental condition on any species in two different ways. One is acclimation, using existing genome, existing genetics, existing power. They just plastic. They try to adapt to this condition using acclimation. We call it acclimation. And then another one is genetic adaptation, changes in the gene. So genetic adaptation is not possible in a shorter period of time, so forget about it. So only the acclimation is possible. So the acclimation is just a natural acclimation using existing genes without much involvement in the environment. And if the acclimation happens only within the generation, is not much helpful. But if the acclimation, what is translated into the genes somehow, and then that acclimation capacity translated or adapted are imprinted in the genome, and that information transferred to the next generation, the next generation will be much better in survival skills under the same scenario. This is we call epigenetic inheritance. So this is possible in many human species or in many higher animals because the genome is highly methylated. 70% of the genome, they have a methylation mark. and it's possible. Environment influence the genome, and then it's inherited to the next generation. Now we have done enough work with me and my colleagues, and including Gen 1 here. I've been intensively working in this area. It's brand new area, epigenetics. So environment, like for example, the warmer winter, a low pH environment, telling or changing the DNA methylation patterns, and as a part of adaptation, as a part of acclimation capacity, and then this is inherited to the next generation. If that happened, the next generation offsprings may have much better chance to survive under similar warmer winter and lower pH environment. If this continues for a few more generations, then this particular population may have a higher adaptive capacity to live in this altered environmental conditions compared to others, because it's dictated, dictated by epigenetic mechanism, which is what we are looking forward. If this is there in a particular species, in a particular population, then we have a high confidence of using this particular population for aquaculture to tackle this issue. That's the whole core idea of the next few slides I'm going to talk to you. I hope it's clear to many of you. So we have been working in this area for past 2-3 years intensively, thanks to two of our graduate students, Kanmani and James. So they have done an enormous number of works in this species, in both the species, and particularly the Hong Kong oysters. So basically, I'm not going to explain this slide. They're cultivating these oysters under natural conditions with various stresses, also under laboratory conditions. And then they take the genome or DNA of these animals and see whether there is any changes in the DNA by epigenetic mechanisms, particularly methylation pattern, and how well these changes transferred into the next generation. And then this changed DNA is persistent in nature, how far is persistent, and whether they have any added advantage because of this changed DNA methylation patterns in aquaculture site. So this is what we have been looking for and working on different stress response pathways, and particularly. But one thing is clear so far. This biomineralization pattern, stress response pattern, is in the Hong Kong oysters in particular, is very well adapted, acclimated. And those acclimations is driven by DNA methylation pattern. As you see here, the number of genes that's altered by methylation patterns in response to warmer winter or low pH conditions is, you see here, there are a huge number of hypermethylated genes. And this genes is giving a power for these oysters to survive under this condition. And also now we have shown that this number of genes, the methylated genes, is translated the offspring. So offspring is also doing much better under these scenarios, other conditions. So the major conclusion, I mean, in a large number of the studies is basically Hong Kong oyster in a particular population, not all the population, we can't that way. A population that we have been using for our studies this looks like well adapted to survive under low pH conditions, in particular because their genes, their genome is capable of capable of learning from environment through DNA methylation patterns, and that methylation pattern is persistent, transferred to the offspring. So that mechanism exists in this species, and that helped them to survive quite well. And particularly the biomineralization mechanisms. That's what we are interested in. When the pH is lower, I think many of you well aware, the pH is related to the biomineralization pattern of the species, because the resources. So this particular mechanism is more capable of acclimating through epigenetic mechanisms. So this is the good news in terms of Hong Kong oysters with the things. Now we have been working with Lee Kum Kee as an industry. Remember, aquaculture is a big field. It's impossible to do this work in the laboratory for a longer period of time. So we have been working with Lee Kum Kee company They have big oyster farms and so several oyster hatcheries as well. So we have been doing this experiment in collaboration with the industry quite successfully for many years now. So that helps us to do the things. So the next important question, we see these Hong Kong oysters in the laboratory conditions are capable of adapting to this environmental stresses within the generation, and also this adapted mechanism is transferred to the offspring. This is true. But what is happened, whether this offspring can retain or persist with this adapted mechanism in nature. Then we outplanted these offspring into different aquaculture sites in southern China. You can see site 1, site 2, site 3. And then we looked at how they perform in the wild aquaculture conditions. Again, the James did a lot of work, and we have clearly seen a positive carryover effect. These individuals with this adapted genome, adapted methylation parents, and they have a good capability in the laboratory condition. And then we put them in the field, and they're doing much better compared to their counterparts, which they, this is compared to control and all other things. You can see here this the 7.4, 7.4 individuals. That's with the acclimated mechanisms, acclimated methylated mechanisms. They're doing much better in the field conditions, which is really, really good in terms of many things. In terms of, but so far they're not highly tolerable to the winter mortality yet. We're still waiting for a few more years to confirm this pattern, whether it's going to happen or not. But that's what the scenario is. So I hope this is very clear to you, the Hong Kong oyster situation. But there is another interesting species, Portuguese oyster, which I compared with you all. Just give me 2-3 minutes, I'll be done. It's another interesting species. This Portuguese oyster, I told you, is a more high salinity species. Is the #1 producer of aquaculture meat in the entire world. As you see here, this species compared to Hong Kong oyster is more vulnerable because it's a coastal species. They grow much slower under low pH conditions. This species is not suffering winter mortality, so forget about that. They are fine so far. But they are suffering because of the other anthropogenic condition, particularly hypoxia and the OA, this combination, all these things. So we have been interested only with OA with respect to this species. So if you see here, they are definitely, if you reduce the pH even very small amount, this species is definitely suffering more, and the shells is impaired. Which we have shown many years ago. But what we see here now, as Professor Wu has found many years ago with respect to hypoxia, which is same thing, sex change. You see the sex ratio is dramatically changed in this species, which is very interesting now to us, how they change the sex and whether these have any implications, which is again, I see it's really, now I'm reading more Professor Wu's past papers to see, how he looked at with respect to hypoxia and all these things. It's very interesting. We never knew that OA can change sex. Which is very exciting. We'll be again looking at, this is our hot topic now. We'll be very interested working in this particular species. So the last message, how to select this species for aquaculture. We know now this epigenetic mechanism, all these things is interesting, is fine, but aquaculture industry is expecting to speed up this selective breeding process using the knowledge we have gained. They don't have time to wait for 3 years, 5 years, 10 years for the natural selective breeding mechanisms to work. So now we are working with the big data, machine learning. I also indicated to James, I'll be meeting next week to talk about more on these things. So we have to use this artificial intelligence, big data to speed up the selective breeding mechanisms to help our aquaculture oyster species. But we are very highly hopeful of doing that in coming few years because we Lee Kum Kee company, which is a huge supporter for us, not only providing the technical support and other thing, they are gave us multi-million dollars to look at these problems. And a big thanks to Lee Kum Kee and all my colleagues. I haven't shown them, and most importantly, the students who have been intensively working in this area for us. and all the colleagues, Kenny Lang, and many others in the group as well, Jen Wen, all these things. Thank you very much to all of you for listening.
Yeah, thank you, Dr. Rajan, for the beautiful talk. Any question from the floor, please? If not, I would like to ask a very quick question. As you have pointed out, and as Professor Wu is here too, I would like to ask a question about hypoxia. Chronic hypoxia is common in the estuary where oyster coexist. Would you please provide your insight into the potential combined impact of hypoxia and ocean acidification on oyster aquaculture?
Yeah, excellent question, James. It's definitely excellent question. Even when we see the big change in the sex ratio, I thought it's Professor Wu is coming back again with the same his hypoxia trick. It's definitely true. Hypoxia is exacerbating OA situation, which has been very well established now, even in the Hong Kong waters. If the hypoxia is magnifying the effect of OA, physically as well as biologically. But luckily, so far, this oyster seems not cultivated very much in a low hypoxic waters because it will affect their growth. So I don't see the growers try to move away from hypoxic areas. I don't think so. These two factors will combinedly affect severely aquaculture of these species. But naturally, some populations experience these two. That's a more wild population. But aquaculture populations often goes to the more coastal areas where hypoxia is not a big factor. So I hope these two factors won't be interacting and affecting aquaculture. Yeah, Jenwen asked another question. Oysters are proteinary. Yes, the Jenwen is 100% true. Thanks for this question. Oysters is interesting sexual change mechanisms, unlike many other species. So they born, the first year they as a newborn individuals, they try to be a male, 90% of the male. And then the second year they change it to female, same individuals. On the third year, they continue as a female, and the fourth year they become a male. This is a scenario. But what we see here under OA condition, 90% in the first year become female, which is, I think, is just preliminary results. We have been doing a lot of work now, uncertain at this moment. Hong Kong species that's what we have observed. Hong Kong species is maybe different. We don't know yet. But what we have seen is only the Portuguese oyster. The Portuguese oyster is easy to work because they breed within 6 months. Quite fast breeding species compared to Hong Kong oysters. Good question. No idea, genuine for Hong Kong oysters, but they born as a male. Yes, you are right.
Okay, thank you, Dr. Rajan. The next session will be chaired by Professor Kenneth Leung. Professor Kenneth Leung, please.
Yeah,
okay.
Thank you, guys. Thank you.
Thank you, James, for wonderful chairmanship. So now I would like to introduce you the next speaker, Dr. Sa Ying Sen. She is our newly appointed research assistant professor at State Key Laboratory of Marine Pollution at City University of Hong Kong. She came from Canada, where she did her postdoc on algal ecotoxicology. Her main research focus quite diverse, including ecotoxicology, environmental risk assessment, emerging chemicals of concern, climate change impact, and water and wastewater treatment. So today she will share her latest discovery on emerging chemicals of concern, stress in microalgae. Sa Ying, please, over to you.
Well, thank you very much for the introduction. And I'm happy to give the presentation today. And my topic is about the toxicity of emerging contaminants on algae. And there are many emerging contaminants in the world. So my focus is on care products, PPCPs for short. The reason I want to study PPCPs is they are commonly used by humans and animals. After use, they will enter the wastewater treatment plant through drainage system. but most of them have not been regulated properly. And also because of the limited removal technology, most of them will enter the environment. And if they enter the environment, they may just have some uncertain effects and also cause unexpected problem for the living organisms in the water. Then my work is to study how those emerging contaminants affect the living organisms in water. But now I just use algae as a model sample, as a model, biological model. The reason I want to study algae is it's very important to the whole ecosystem. It's the link between the living organisms and the non-living things. For example, algae may absorb carbon dioxide and water and convert them into energy and carbohydrates to feed themselves. And at the same time, algae may produce oxygen for other organisms to consume. And as primary producers, algae could be considered as food to feed other higher trophic level organisms. And algae can produce many value-added products, such as biodiesel, food, pharmaceuticals for industry use and human use. Then if there is something wrong with the algae, that may seriously affect the whole ecosystem. But to be honest, the algae is not that weak, because algae is the earliest form of life on Earth. And no matter how many years pass, and no matter how many disasters the Earth went through, algae still lives well in the water and in the air and also in the soil. So algae is not that weak as we imagine. Then I just want to figure out how those emerging contaminants would affect algae. Are they safe or not? Then I'll give a specific topic to solve the problem. And it's the long-term impact of a post-disturbance of triclosan. And I used a different algal species to do the test. Triclosan is a kind of antimicrobial, and it has been containing many products such as soap, toothpaste, and body wash. You may use it almost every day. But triclosan is used to kill bacteria. But if it enter the environment, if it will target some non-target organisms like algae, zooplankton, and fish, that's a problem. But many people study that. But still, there are some problems unsolved. Like, I want to do this study because many different countries have different attitude towards triclosan application, like the United States, European Union, Japan, and Australia. They have negative attitude towards triclosan. They thought triclosan may target multiple harm the human health. But for Canada, they have a positive attitude towards triclosan, and they don't think triclosan would cause that serious problem. But I just want would cause other effects when it enters the environment, and I just use algae as the test model. In reality, triclosan would stay in an environment in days or months. So I just choose a longer exposure time, like the longest time is 29 days. And I used multiple algal species because some people just use model species, and I also add some other species and want to compare their sensitivity. And the last one is I used environmentally relevant concentrations. I just want to mimic the real situation. And those are the algal species I used. There are 5 species, and CPCC 12 and CPCC 243. They are the algal model, but they are very small, just to have 10 micrometers in diameter. And for CPCC 12, it has no cell wall. The other algae have larger size, and for chlorococcum species, it's a colonial cell, and that means the cells are love to grow together. And for E. viridis, it is the largest one I used. It has size as large as 1 meters in diameter. And for the tricycle concentrations, of evaluation, then I used multiple endpoints such as cell density, cell viability, pH, chlorophyll pigment concentration, and chlorophyll fluorescence, as well as biochemical operation. Those information can be just use synchrotron light, not the normal light source. So what is a synchrotron? I just want to give you a brief introduction. Its size is as large as a football field. It can accelerate electrons to almost the speed of light. So you can see the light speed is very fast, and also it has very high purity. That means because it has very high vacuum to make it has a very high purity and has very high brightness. That would make it can get a very higher resolution and a higher signal to noise ratios to make the results accurate. And we have a lot of, it has a lot of magnets around the circle that the magnets are used to change the light directions. When the light directions change, there are excess energy to go through the tunnel light to reach many workstations. Then
I'm sorry, there's some technical problem with the connection. She will join us again. We are welcome any comments or questions.
Wait, yeah. Hello, can you hear me?
Yes, welcome
Hello?
back. Perhaps since
sorry,
your Wi-Fi connection really weak, maybe you can you can close your camera so that you can transmit the sound back.
Okay.
Or you can change venue if you are in the camera.
So is that better now?
Yeah, but without your, can you upload your slide? We couldn't see.
Okay, I'm just
Okay.
Can you see the slide now?
No, now okay. Yeah. You
okay. Can. Oh, sorry about that. So I will just continue from this slide.
Yes. Yes.
Okay. And synchrotron has very wide applications in bioscience, medical research, and materials and environmental science. And some researchers have used synchrotron-based technologies to study toxicity, just to use animal tissues or human tissues. They want to see how those animal tissues have been affected, like in their proteins, lipids, those molecule components. But few people use single cell, like to do the toxicity test. And so my work was the first to apply synchrotron-based technology to study the toxicity, but use a single cell. So that's another reason that I want to use algal cell, because it's the single cell. And please look at those images. The first row is about the Canadian light source. It provides synchrotron-based technologies in Canada. And the second row is about like synchrotron-based FTIR spectral microscopy. It can be used to do in vivo tests. That means it can see the single cell, but live cell. You can see the right-hand side image. That is a sample holder, and all the light have been covered. Just the light is focused on a single cell, and it can extract the useful information from the individual cell. Then let's go to see some results. First is about cell density. The results showed the species-dependent effects. For example, CpCC12 is the most sensitive species, and the E. viridis is the least sensitive species. But for A. superbus and E. viridis, when they exposed it to environmentally relevant concentrations of triclosan, they show stimulated cell growth. That means their cell number increased, not decreased. For the cell size, you still can see A. superbus has the largest cell size changes, but for the other two species, CpCC243 and CpCC12, they have the mild or almost no cell size changes. They are the smallest cell, and it also meets the species-dependent effects. For photopigment variation, you can see 3 species have a good dose-response relationship. They are Qualcomm species, CpCC243 and CpCC12. And for the others, like E. viridis and A. superbus, they also have stimulated chlorophyll pigment concentration when both species are exposed to environmentally relevant concentrations. These results are consistent with the cell density. And for chlorophyll fluorescence, we did a lot of tests, but we found FVFM and QN are the 2 most sensitive species, sensitive parameters. And FVFM indicate the effectiveness in photosynthesis system. And QN is the non-photochemical quenching. And the increase of both parameters indicate the cell have the protection mechanism to protect the cells against the attack from the triclosan. But the inhibition in 2 parameters indicate, well, the photo damage from the triclosan. So you can see triclosan would attack the photosynthesis system and to affect the photosynthesis process. And also the results have species-dependent effects, like the CpCC243 is the most sensitive species to QN. And from synchrotron-based FTIR, we can see the molecular changes. Like we choose lipids, proteins, DNA, and carbohydrates. And different species have different responses in the molecular components. And in order to see some details, I use second derivative spectra, like just to take lipids and proteins as an example. 300 to 2800 are the area to give information about saturated lipids. And you can see CpCC12, A. superbus, and E. viridis are sensitive to the higher levels of triclosan exposure. And you can see the spectral shape, intensity, and spectral position changed. And for chloroqualcum species and E. viridis, when they exposed it to environmentally relevant concentrations, the intensity in saturated lipids increased. While for protein, we use amine-1 to see the second structure changes. And we can get a quantitative and a qualitative information from the spectra. And the results showed a CpCC12 or 4 algal species, except for the CpCC243, are showed sensitivity in protein secondary structure. And then I used the PCA principal component analysis to see the sensitivity difference among the species, and different species have different sensitivity. Like from the figure, you can see CpCC12 is the most sensitive species because the responses have been separated according to different concentrations. And also chloroqualcum is the less sensitive one. But for A. superbus and E. viridis, they are the least sensitive species. But according to the previous results, then we can conclude that E. viridis is the least sensitive one. And this study just tell us different species have different sensitivity, and just under long-term triclosan exposure, which indicate long-term triclosan exposure can change algal community composition, which means the long-term triclosan exposure may increase the cell growth for some species, but inhibit some the cell growth. And even though for the one species, different endpoints have different sensitivity. So in order to give a comprehensive evaluation, it's better to use multiple endpoints to compare. But those endpoints are different, but sometimes they are related with each other because we get the information just from the one single cell, individual cell. We can see the correlation from the spectra and the other results. And the last one is the most important one, is the environmentally relevant concentrations show different or multiple long-term effects, such as inhibition, resilience, resistance, and stimulation. The stimulation one needs to further attention because it's the opposite from inhibition, but it has wider effects, just like when environment has enough nitrogens or phosphorus, they may cause algal bloom or red tide. But the environmentally relevant concentrations, when the chemicals just to have those very low concentrations, they may also cause the stimulation for the algal growth. So, but the inner mechanism has not been studied. Then I think it needs further exploration about the hormesis phenomenon. And that's what I'm presenting. That's all about my presentation. Well, thank you for listening.
Okay, thank you, Sai. I would like to start a question that what can you do with the synchrotron? I mean, rather than using the traditional electron microscope, what additional things you can see using the synchrotron?
By using the FTIR, or
yeah.
okay, I can take FTIR as an example. Because it can measure the single cell and it can measure the live cells, so the information just is more accurate. Because from the traditional light source, if you want to use the FTIR, all of the algae have to be just freeze-dried, and all of the algae are dead. So there's a little difference just between the results. The second is about what can we get from the results. We can get the molecular component, some information about those molecules, like lipids, proteins, DNA, and carbohydrates. Well, there are some specific wavelengths that can give us information about those molecules. And also we can use second derivative spectra to see some details about those molecules. We also can see the secondary structure changes of protein, like the alpha structure or helix structure. We can see the changes, the ratio changes of two structures, and to see how the protein degraded. So it can give a lot of information about the molecules, but it's the general information, not the specific one. If we can get the specific protein or lipids, we can do the specific analyze further.
I see. There is a question from Dr. James Ban to you.
Okay.
Whether your data obtained from single cell or from cell culture with multiple cells?
From a single cell or from cell culture. Okay. Actually, the every time when I just measure one treatment, I will take more than 10 cells. So, but the spectra is from single cell, one cell, just one cell. But I will measure more than 10 cells to get a average, the spectral average. Yeah.
I see. So Sa-Ying demonstrate one tips for scientists now. If you use really high-tech machine, you can publish in good journal easily. So and also this synchrotron can tell us a lot of different parameters we seldom able to measure in the past. So very nice work.
Hello, Kenny. Can I ask a question?
Jenny,
Yeah. Yeah. So thanks, Xia-Ying, for a very interesting talk. And may I know whether there are significant now if you now you have the tools to analyze at a single cell level, right? So are there any significant variations between individual cells?
Like the individual between individual cells and the biomass.
Individual cells, between individual cells, the individual cell responses, because this is a very important actually a question in ecotoxicology, whether the individual variations, okay, and would how should I say that? So I mean individual sensitivity to the even the same concentration of a same toxicant.
Yeah, you're right. It's a very interesting topic. And when we do the single cell analysis, there is, of course, there is a difference just between like among cells. But the problem is if I use the same treatment, like when a cell is exposed to a specific concentration, then when I measure the cells in this treatment, there is a little changes. But when I get the average spectra and I compare this spectra to the another spectra just from another treatment, that means the cell exposed to another concentration, like the results are different. So just in the same treatment, in the same treatment group, the cells has difference, but compared to another treatment, the difference is just bigger. Okay. Well, yeah, and I think when I do the quantitative analysis, I would use a reference, like I would choose the spectra area of MI2, that is functional group of protein. And then I would use a ratio, like use the spectral area of lipid to divide it by the spectral ratio of MI2 and to reduce cell-to-cell difference. So we need to do something to reduce cell-to-cell difference if we want to get the quantitative information. But if we want to just reduce the qualitative difference, I think we may just control the cell circle. It's better to control the cell life circle and to make it in the same state, in same phase.
So why I ask this question is because we challenge the concept of EC50 or EC. Now, why 50% of things can die or can be responsive, or why another 50% can be still alive? You know, so we want to see if there's individual sensitivity plays a significant role in defining so-called EC50 classical ecotoxicology. I think maybe single cell analysis may shed a light on this issue. Okay.
Oh, thank you. That's
thank you
very
much.
to think about. Yeah, thank you.
Very interesting discussion. Given the time limitation, may I now hand over to Professor Rudolph Wu to lead us to have the final question and answer and also a roundtable discussion. May I invite all the speakers of today switch on your camera so that we can have the roundtable discussion. Now over to Professor Wu, please.
Yeah, okay, thank you. Now, everybody, this one-day symposium actually highlight a wide range of impending problem on our marine environment in the coming decades. And some of the problems that we mentioned are actually imminent global problems such as hypoxia, ocean acidification, emerging chemical concern, especially EDCs and LCMS, nanoplastics, antibiotics, etc. And some others are particularly relevant to our tropical and subtropical environment in the region, and such as ciguatera and coral bleaching and so on and so forth. At the same time, and a number of workers and also introduce novel technology and or novel approaches to address or to solve some of these problems, such as the biosensor population monitoring and the metagenomic approaches, and for identifying ARGs and risk assessment, and then coral restoration and eco-engineering of seawall and so on and so forth. Now, what I suggest is that we call on the panel members, Professor Kenneth Leung and Professor Jiwan Yao, and then Dr. Apple Ju, and then Dr. Henry He, and then John and Dr. Leo Chang to join us together for this panel discussion and lead the discussion. And of course, and all participants are welcome to throw in their ideas and in the discussion. And in view of the shortage of time, I suggest we actually primarily focus on two areas and in our discussion. The first question is that what is the grand challenge of these marine environmental issues in the East Asia region, which is much more relevant and close to us. And the second is that the state key lab of marine pollution has considerable expertise and strength in marine pollution. In our capacity as the leader of the Regional Center of Excellence in Marine Pollution, or PAMC, in what way that we can share, help, and contribute to these and their consortium so that we can have a better future. Now, the topic is open for discussion, and I'd like to have your comments. Thank you.
Maybe I start first. I still think those are common issues like eutrophication, hypoxia, still present the greatest challenge in our region. Because we have a really high density and population in coastal cities, and also many of countries in the region without the financial and also the infrastructure support to treat the wastewater to a more acceptable level. So I think basically the organic pollution and also the nutrients discharge remain one of the key challenges in the region. If you talk about chemical pollution, they have impact, but rather in a gradient. That means in the discharge location, you may have a more adverse effect. But once they reach out with dilution, the effect will be lessened. But on the contrary, if there is a eutrophication hypoxia, the impact area will be very huge, and then we'll have a long-lasting impact. I personally, I think this remain one of the biggest challenge we need to look at and tackle.
Okay. Another view. You guys are very shy. No? So how about the other panel member, Rajan?
Yes, Professor Wu. Yes, what Kenny says is correct. Hypoxia, eutrophication is going to be the key. But also, I think we can't ignore the global change, particularly changes in the seasonality, which is, we can call as a climate change, but I think is even the regional climate change, heavy rain, heavy, unusual weather pattern. These are all going to work with the hypoxia. It's all going to work with the eutrophication, which is I think the impact of this interaction between the seasonal change with the local pollution or local, whatever the pollutant we have studied so far is going to be in a different scenario. I don't know whether it's going to be the same or it's going to be totally different. I think this which the society wanted to know. If a huge rain in southern China for a particular year, will be the impact on all the stresses we have measured is on their impacts. I think that's I think is something interesting to me.
Mhm. So how about Gen 1? Oh, sorry, Henry.
Yeah, Professor.
Henry.
Yeah, I agree with Kenny and Rajan's view on the current contamination issue. But in my view, perhaps a more profound and emergent issue would be the Fukushima nuclear water. That's even compared to endocrine disruption or hypoxia study, the potential environmental impact or ecotoxicity of those nuclei on the ecosystem are actually under study. So no one actually know a lot of detail about what the consequence that these water, if these water with such a large amount being released into the ocean, and what could be the consequence to the East Asia Sea. So and the sample is very difficult to get, right, under the current circumstances. So we may think of a strategy that to deal with this potential problem. I don't know how, but that's really concern. And there's really a public concern as well, right? People are afraid of consuming seafood after the news been released that the nuclear water might be discharged directly into the ocean. And I don't know, maybe some other panel member can share their ideas.
I think I studied this issue quite in details. I think Southeast Asia and Hong Kong are pretty safe because the current won't bring those nuclear pollutants down here. I think the impacted area will be Yellow Sea, Eastern Sea, Korea, and Japan themselves. And also the current will bring the pollutants all the way go to USA. So then these are the facts. So we are quite lucky in Southeast Asia. We have no problem because the China government, especially the Southern Oceanology Institutes, have been monitoring this since the incident. So far so good in our region. So Professor Wu, please.
In actual fact, you're right, and I share the similar concern. And when they announced this, and in fact, and that's what Kenny pointed out. If you just based on the hydrography, so basically the water and discharge from the Hiroshima, and there would Fukushima, sorry. And then we then go around Japan and go up. And then by the time it will be pretty diluted. And then the most affected would be the sort of the northern part of China, Yellow Sea, and so on and so forth. And then the water, the current won't come down. But however, I got fairly, I can't think of a better word, pissed off. And then because, and then the Japanese saying that it's not, well, no problem, and same as the Americans. And then, in fact, they are, if you discharge so much, and then they said the dilution is the solution because I diluted so much, and that, so in that case, you don't need any treatment at all if in there based on the way of thinking. And then, so at several months ago, and we tried to use an artificial muscle, explore the possibility of using the artificial muscle that we developed to monitor the sort of radioactivity, because they actually take up uranium, cesium, and strontium. And then, so we are underway doing this with some of the colleagues in the CTU. And then hopefully we can provide a monitoring device, but I'm not sure because due to the political situation whether the American and Japanese will use this, and they know about this, but they refrain from doing this. In fact, and when the sort of Fukushima incidents occurred several years ago. I was serving on the group of experts on marine pollution of the UN, and they hire away a lot of these facts from us, in fact. But anyway, that's the side issue. So,
it's Professor, it's very good to know at least the technique you develop. The artificial muscle can be used in monitoring this kind of environmental pollutants. My thinking on the two issues Professor raised. One is grand challenge. One is the development of sticky lab marine pollution. For the first one, I think you guys have raised a lot of points. But for the second one, I think this meeting with a lot of colleagues from Southeast Asia would be a very good would be a very good platform for us to build up some collaborations. And from today's talk, I know a Sticky Lab has a lot of expertise and to analyze a lot of chemicals, to do very comprehensive toxicity tests. I think if we can do more this kind of meetings to develop some large-scale studies, we can contribute more to solve the problems in Southeast Asia. So I think this is a good beginning.
Yeah, I agree with you. In fact, and I hope members still remember and why we are bestowed with the title of the Regional Center of Excellence by PAMC is that they expect us to take the lead and then contribute and help each other out in the region of these and the 11 member country. So I think we should sort of pursue for that effort and then try to help each other out and then use our strength and then share with them. Say, for example, before that, we organized this technical training workshop on EIA and things like that. So now, say, for example, take examples, the things that we do, we are good at, something like the coral restoration, and then the eco-shore, and we can share at workshop with them so that they can follow and the same path and then improve the environment in the other country, particularly in the developing country.
Yeah, and also related to this coral restoration. In a lot of places, if we talk about coral restoration, we need to study their drink flow, the source and sink of larvae, and also other organisms. When we talk about restoration, we have to consider this kind of, large-scale geographic areas, populations in different places. So if we can build up this kind of collaboration, we can do more practical, more meaningful restoration projects.
Yeah.
So.
Yes, in fact, I think that's a good idea. Say, take example, if you organize a workshop, regional training workshop, and for coral restoration and for shore restoration, that will attract a lot of attention. And then this is also doable. And then the other thing is that the other members of the State Key Lab, if they organize a training workshop on the chemical analysis of these emerging chemicals, which are difficult, and then we share with them the SOPs and things like that that will help them to jumpstart to tackle the problem and very quickly.
Yes, I totally agree with what Professor Chu mentioned about restoration. We talk about connectivity. You know, the sea so large, the dispersal of larvae, where do they go actually? Where are our source of larvae? Where is the sink, etc.? I think because today we talk about big picture, and I really appreciate this workshop opportunity so that we can, you know, lay out everything and discuss about our plans in future. But if you ask my opinion, I think Hong Kong is really special because I always because especially for corals, I think it also applies to other organisms in general, because you know, Hong Kong is a very interesting place. It's so highly developed, highly urbanized, but at the same time, we have so amazing, awesome marine biodiversity. So we can always make good use to our very unique environment context, very extreme environment. For example, in terms of temperature, hypoxia, sediment, everything's just happen at the same time in such a small place. So by understanding these kind of impacts and study the organisms that survive in this environment, to understand whether or not they're really that resilient, and if they are, then we understand the mechanisms. We do comparative studies. So that's why I really appreciate what Professor Chu just bring out, whether or not we can strengthen collaboration across different countries. You know, these are very important for collaborative studies to, you know, try to see if there's any difference and the mechanism behind. But I think the very important is, after we understand the mechanisms, molecular pathway, the adaptation, very important is we have to bring our knowledge forward, you know, to really help in conservation in this phase of climate change. So personally, I love what all the presentation I've listened. I think they're really awesome, like oyster, you know, hypoxia, like eco shoreline. These are, I think, a very frontline study, and I think it set a very good examples to others as well. So I think a workshop, etc. I really like that. Yeah. Myself and contribute as well. Thank you.
Yeah, that's good. And how about Jin Ling?
Yes. So because we actually talked part of us, okay, I talked a lot of laboratory experiments, right, and the demonstration of those hazardous things. Now, what I'm curious about is, do we really see, okay, do we really see apical, ecologically relevant and those hazardous impacts really occurring in the regional marine environment, so that we can really tackle it as a priority? Otherwise, we may just play a lot of kitchen chemistry or whatever in the laboratory. So yeah, this is my 2 cents. I think we really need to think about the protection goal based investigation and future research to really target what has really been occurring in the environment. Yeah.
Okay.
Thanks.
I would like to invite Daisy Penayo to answer the second question. What can we help with the PAMC and the region's partners? Thank you, Daisy.
Okay, yeah, thank you, everyone. Thank you for all the speakers. It was a very interesting day, actually. The topics were very good. Yeah, in terms of how SKLMP can help the countries in the region, as mentioned by Professor Wu, SKLMP is one of PEMC's regional centers for excellence. And as one of the regional centers of excellence, we actually hope that SKLMP can provide capacity building to most of our partners in the region, particularly the universities and even the local governments. But then again, of course, most of the universities and local governments in the region don't have the capacity. I mean, they don't have the technologies that SKLMP have. So we can take that in consideration in terms of possible collaboration and partnerships, because most of them, most of the laboratories in the local governments, and at least in the universities, it's not the same as what you have in the University of Hong Kong. So maybe we can take that as a consideration. And then currently, we are actually developing the PEMC roadmap and the SDSC implementation plan for 2023 to 2028. So in that plan, we are actually identifying the priority programs or priority management programs in the region. So that covers a range of issues like climate change, marine pollution, habitat protection. and food security, fisheries management. So we can actually identify what are, in developing the plan, we identify the issues and what are the needs in the region. So in developing that, maybe we can also consult with SKLMP on what are some of the aspects in the plan that you can help in implementing it, and how we can share your knowledge and expertise to countries in the, at least in the PEMC countries. So I think we can put that forward. The development of the plan will start. It's actually starting. So in putting that together, we will take that into consideration. So hopefully it's all right if in the next few months we will also communicate with SKLMP, probably through Professor Wu or Professor Kenny. And while going through that, we'll be able to identify what areas SKLMP can contribute to. So and then part of it, of course, in the plan is we also have the capacity development plan. So I think that's where SKLMP can particularly help, and also maybe in terms of identifying targeted research for marine pollution assessment. Yesterday and this morning, there were possible opportunities for collaboration on research, like on Ciguatera research, and I think some, what's the other? There are other opportunities for possible collaboration. So we can actually do that in the future. And I also noted that this symposium and the training workshop yesterday was very helpful, actually. It was a good way of sharing your expertise, and it also provides opportunities for possible networking. So as you mentioned, we can also organize similar workshops in the future, but maybe more targeted or more subject specific. Like if we do microplastics, then we can organize one on microplastics, one on coral restoration. And then we can target specific participants who would benefit for those types of workshops and seminars. And also we take note that there are also opportunities for universities can send samples to the University of Hong Kong for cross comparison among labs and all that. So we take note of all those opportunities, and we can have future communication to build this with SKLMP, and link you with our PEMC network of learning centers and the PEMC network of local governments. Thank you.
Because I think that's I think that's a good idea. And then because the sort of the development is both ways, and then so we can offer something, but the most important thing is the PEMC to identify what's the thing that you need most urgently. And then as you said, I don't think there's any problem if you send somebody to come to Hong Kong, or to sort of affiliate with certain laboratory for a certain time, because we did this several times before. And I remember PEMC actually sponsor a couple of people to learn the artificial muscle techniques. And when I was in Hong Kong U, they attached with us for 3 months and something like that. And then the other thing is something like the toxicology is maybe one of the area, because something like, if I remember correctly, in the region, and there's no sort of self-developed or tailor-made and water quality objective. And that's one thing which is important. And also the toxicity test and that kind of thing. And that may be a possibility. And after these symposium, probably PEMC would have a much better understanding on the current expertise of the Stakey laboratory. And then I suppose, and the Kenny as the director of the Stakey Lab would be more than willing to help.
Yes.
Yeah.
Yes.
Yes, Professor Ken. So we will communicate with you.
Yes. I would also like to introduce to you our Dr. Leo Chang, our associate director.
Yeah.
He is expert on the coral ecology, coral reef survey, as well as secret terror algotoxin survey. Maybe Leo can say a few words, please.
I think we all need to thank Rudolph, because I think Rudolph asked us to organize this NC training workshop and symposium. At the beginning, we find there's a lot of documents need to prepare, right, Rudolph? So I think Rudolph is consistently, I think, one stage lab to participate in NC from the very beginning. But I think only we have the expertise to make it happen today. So in the past 15 years, I think several marine environmental research and innovative technology have been developed under the leadership of Rudolph. No matter Rudolph in CTU, Hong Kong U, FU, as James mentioned, that Rudolph is always our leader or our teacher. So I think in the next stage, I think also the China government are very concerned about is how we can develop all these SOPs into international standard of scientific method. They are pushing us to do this, including the Chinese Academy of Science. They already asked us to develop this kind of SOP, for example, artificial muscle, right? It's a very good example for pollution monitoring. And also, I think Daisy mentioned that the inter-country sample comparative, but those research is based on international standardization methods. So I already work with the Chinese Academy of Science and also I think WestPac to develop underwater habitat mapping. And also our chairman also mentioned that coral health monitoring, coral restoration. We also can develop different kinds of SOP at the beginning, but eventually develop into international standard. Also for the benefit data flagellate sampling, everyone using different methods. So it's very hard to compare the results. So I think it's also very important. And also I think we are Kenny asked us to establish the research training center for the UNESCO. So we think research collaboration together with training is very important. So in the future we will develop the RTRC with UNESCO, and also the Chinese Academy of Science. They propose to establish a joint laboratory for environmental ISO. So I think maybe probably Rudolph can help and take the lead for this initiative. And also we applied the ECF to organize ISO meeting for environmental methodologies. I can report to all of you. Thank you.
Thank you. Very good. And in particular, Leo has a wealth of experience in knowledge transfer and that sort of thing, but can also help and with the other PEMC members. But in view of the time, I was asked to stop at 5:30. Otherwise, I got killed or get blow on the head. So maybe I can start. So I should stop here. But again, thanks very much for all your contribution, and I hope you will continue to support and contribute to the Stakey Laboratory and the PEMC Regional Center of Excellence in the years to come, and that relies on the leadership of Kenny and Leo. Okay, thank you. Thank you.
I would also like to thank all the speakers for their time and effort and sharing of their latest discovery. I especially want to thank Saying and also Phoebe and Grace for organizing this event. They did lots of hard work for the event. I also thank Daisy for coordinating with us. Thank you very much.
Yeah, thank you. Okay.
Thank you, everyone.
Okay, goodbye.
Okay, bye-bye.
Thank you.
Okay, bye-bye. Thank you. Bye-bye.